Hi, everyone. I want to welcome you to MDA's first Insight and Research Investor Summit. I'm Sharon Hesterly. I'm MDA's Chief Research. Thank you for joining us this morning. I think if we have some panelists joining who are not on mute, please go ahead and mute yourself. Thank you. Before we kick off this morning, I just want to start first with some quick housekeeping items. Okay, so just so we all know, of course, online web activity has increased dramatically this year. Hopefully you will have a good connection. If you're having any problems, please let us know. You can let us know in the chat box. We can try to troubleshoot that. This is a one-time live program, but it'll be made available for viewing on demand for registered attendees only. So following the meeting, you'll get a link and that will take you directly to individual presentations. We're also going to update that compendium, the PDF compendium, and we'll include the links for each presentation within that compendium, and you'll get another copy. So for optimal viewing, under the options at the top of your screen, just go ahead and select side-by-side mode. That way you can make sure that the whole presentation fits in your screen. All attendees will be muted, so please submit your questions through the chat box. So, okay, here we go. So just a little bit about the Muscular Dystrophy Association. I know we have a lot of people joining who maybe aren't as familiar with MDA, but we're an umbrella organization. We've been around for about 70 years now. We cover not just the muscular dystrophies, but neuromuscular disease. And those are broad categories that you can see here, everything from, you know, the motor neuron diseases, for example, ALS. We have a pretty significant funding in ALS, and also mitochondrial disease, ion channel diseases, peripheral nerve disease like Charcot-Marie-Tooth disease, neuromuscular junction diseases. So as you can see, it's a pretty extensive portfolio, and not everyone is aware of the extent and the breadth of our program. Just a few highlights, as I mentioned before, this last year was MDA's 70th anniversary. We were founded in the 1950s. Since that time, we've invested over $1 billion in neuromuscular disease research. So that includes funding of 7,000 individual investigators. We've trained 2,200 new investigators and in both research and in clinical sciences. And highlights include the identification of the first gene that was ever discovered through positional cloning, and that was dystrophin. Responsible for Duchenne muscular dystrophy, and at least 8 of the new drugs that have been approved for neuromuscular disease had direct or indirect funding from MDA. And in fact, probably all of them had some indirect funding from MDA. Just as an example, over the years, we've spent about $125 million so far in gene therapy. So that includes you know, our first grant from the early 1990s, which I think went to Dr. Jeff Chamberlain, and continue to fund some of his research into figuring out how to shrink the dystrophin gene to fit into an AAV capsid. We've funded work early days in using adenovirus, then switched to adeno-associated virus. The first human trial in gene therapy was actually for alpha-sarcoglycan deficiency, and back in 1999, and MDA funded that study. We also funded the first Duchenne muscular dystrophy study for gene therapy back in 2004, and that was a collaboration between Jerry Mendel and Jude Samolsky. Since then, we've funded more, you know, we've been funding gene editing and RNA targeting therapies as well. So, actually, we in 2019 were awarded the Scarlatti Award for Achievement from the American Association of Cell and Gene Therapy. So just a little bit of the highlights for where we've invested our money over the years. If you don't know, we also maintain a network of care centers. So we have 150 care centers throughout the US. These conduct about 70,000 different medical visits over the year for 60,000 people living with neuromuscular disease. We serve as a hub of research, and we know that we have about 12,000 individuals at any given time participating in clinical trials and natural history studies. And then given that we have that large clinical network with people who are coming in regularly to be seen for care, we've also in the last several years launched the Mover Data Hub, where we are collecting clinical data at the point of care. So we have we're up to 40 participating centers now who are collecting data in 7 diseases. These include ALS, limb-girdle muscular dystrophy, Duchenne and Becker muscular dystrophy, Pompe disease, and FSH dystrophy. So those are just some highlights of some of the activities that MDA has, if you're not familiar with the organization. You'll hear a little bit later about our venture investments. But I guess a point that I wanted to make here is that, you know, as I'm sure people are aware, this space in neuromuscular disease has just been exploding compared to 10 years ago, when you could almost count on your fingers the number of companies involved in the space. We track the assets in this space, and we'll hear more about that later today at the Cello Health panel. But according to the numbers that we track, you know, we have approximately almost 120 assets in ALS. You can see Duchenne muscular dystrophy is close to 75, and the others beneath that. So clearly the field is exploding. There's been a lot of activity, and I think that was the genesis of doing this investment seminar now, is given the amount of activity that we've seen and the number of different applications that we received last year for our MDA Venture Philanthropy program. It seemed to make sense that we had hit a critical mass to hold a conference like this. And we'll of course be very interested in your input afterwards. And we will be asking people what you think. So just a few notes for today before we get into the main part of the session. So each speaker will have either 15 or 20 minutes. So this is time inclusive of time for questions. So if you are a speaker and you go over your time, you will be out of time and we will move on to the next person. So please try to stick to your allotted time, especially if you want time for questions. Also, the PDF compendium that was mailed to all speakers and registrants contains the agenda, along with a lot of additional information about the companies and the institutions that are presenting today, including their follow-up information and their contact information. If you don't have a copy of that compendium, you can request that from Liz Habib-Laux. If you send her an email now, she'll get right back to you and make sure that you have the final updated copy of that compendium. And finally, an important note, please remember to register for both days of the conference. So you should receive 2 registration links, one for today and one for tomorrow on Friday. That link is in the chat box right now. So if you haven't registered for Friday and you want to attend Friday's presentations, please do that as well. So we are slightly ahead of schedule, but I think that's usually okay. People don't typically complain about being ahead of schedule. So we are going to move, into licensing opportunities, where we're going to hear from a number of different universities and their academic investigators who have some interesting technology that they would like to share with us. So I'm going to turn it over now to my colleague, Jonathan Sabag, who is our research portfolio director, and he will introduce the next speaker. Jonathan?
Thank you, Sharon, and good morning, everybody. Welcome to the first session. This is Licensing Opportunities. Our first speaker this morning, who will have a little bit more time than he planned, so feel free to take your time, Dr. Lopez, is Luis Carlos Lopez Garcia from the University of Granada, and he will tell us about one molecule, 2 therapeutic applications. Dr. Lopez.
Do you hear me?
Yes, we can hear you.
Okay. So good morning for all of you that are in the US. Before I begin, I would like to thank the organizer for giving me the opportunity to present our work here. And this work is about an interesting molecule with 2 different therapeutic applications. The molecule is called, its name, beta-resorcelic acid. It's a natural phenolic compound that is present in different plants and different fruits like cranberries, olives, or coffee beans. And interestingly, it is used by the food industry as a taste enhancer. And the first therapeutic use that we have tested is in the treatment of coenzyme Q10 deficiency, which is a rare mitochondrial disease with a limited therapeutic option. For that, we use an animal model of CoQ deficiency that we name CoQ9R239X. These mutant mice have a fatal mitochondrial encephalopathy, and all of them die before 7 months of age. The treatment with beta-arteroxalic acid dramatically increased the survival of the animals, of the mutant mice, being this survival curve similar to the survival curve in the wild-type animals. And this is 5 times higher than the survival curve of the untreated animals. So this therapeutic effect is really extraordinary, and it's not usual in the lab, no? In the experimental model that we use in the labs. Also, the locomotor activity was increasing the mutant mice treated with beta-arteroxalic acid, as you can see here in this video of 2 mutant mice from the same littermate at 5 months of age. This increase in the survival and also the improvement of motor performance were due to a decrease in the astrogliosis and a decrease also in the spongiosis, which are morphological features of mitochondrial encephalopathy. However, the animal body weight did not increase in the mutant mice treated with beta-arteroxalic acid. And this is something that we will get back because it is related with the second therapeutic application. The main mechanism of this therapeutic effect was the decrease in the levels of demethoxyubiquinone, which is a metabolite that is abnormally accumulated in the mutant mice. In fact, when we recover the high levels of demethoxyubiquinone in the mutant mice, these mutant mice have again an early death. And importantly, the effect of beta-arteroxalic acid in decreasing the level of demethoxyubiquinone can be also reproduced in skin fibroblasts from patients with CoQ deficiency due to mutations in the same gene, the CoQ9 gene. So in short, we can say that beta-arteroxalic acid targets coenzyme Q biosynthesis, and that leads to a decrease in the levels of demethoxyubiquinone, providing a powerful therapeutic effect in the CoQ9R239X mice. And this is something that has been also reproduced in other mouse models of CoQ deficiency, such as the models with mutations in CoQ7, CoQ6, or CoQ8b. Well, the second therapeutic application of beta-arteroxalic acid is related with the prevention and treatment of overweight and obesity. When we treat wild-type mice with beta-arteroxalic acid, we observe a slight increase in the survival at the early stage of life. As you can see here better in this graph at 24 months of age. Also, the locomotor activity was slightly increased at 18 months of age, but the most important thing is that the animal body weight were clearly decreased in both male and females. And this decrease in the animal body weight is due to a reduction in the content of the white adipose tissue, while the other tissues, like skeletal muscle, for example, looked normal. This low content of fat lead to the prevention of the age-related hepatic steatosis, and also to a reduction in the adipocytes hypertrophy. And this effect on decreasing the fat lead to to mice that are thinner and also healthy. As you can see here in the second video of 2 mice, also from the same littermate, at 18 months of age. So to figure out how beta-hydroxylic acid can decrease the content of fat in the body, we culture pre-adipocytes with beta-hydroxylic acid. And what we observe is that the number of pre-adipocytes in the treated group were clearly decreased. And this is because the levels of protein that controls the cell cycles, such as P27 and CK2, were decreased under the treatment of beta-hydroxylic acid. And also, when we differentiate the pre-adipocytes to mature adipocytes, the fat that they can produce is less in the group with the treatment with beta-hydroxylic acid. And this, we believe that this is related with the reduction in the levels of PPAR gamma, which is a transcription factor involved in the control of adipogenesis. We also perform mitochondrial proteomic analysis in the kidneys of the wild-type mice treated with beta-hydroxylic acid, and we found a downregulation of beta oxidation, and also an upregulation of the acetyl-CoA biosynthesis and TCA cycle. Also, we observe a decrease in the level of glycerol 3-phosphate, as well as in the activities of two glycolytic enzymes, PFK and PK. So we believe that the glycerol from fat is entering the glycolytic pathway and then producing pyruvate, which is used to produce acetyl-CoA, and then this acetyl-CoA is used in the TCA cycle. So, and all these pathways are upregulated in the mice treated with beta-hydroxylic acid. So, in summary, with this second application, we think that beta-hydroxylic acid can inhibit the adipogenesis, but also stimulate glucose and acetyl-CoA metabolism and lipolysis. And this produce or lead to a decrease in the fat content, which is important for the prevention and treatment of overweight and obesity. And this has also implications for hepatic steatosis and to have an healthier aging. So for the first therapeutic application, we think that is important to apply for orphan drug designation, because this option can provide a fast track path at the EMA and the FDA. For that, it's also important to mention that we have here in Spain 2 patients that are using now the beta-hydroxylic acid as a compassionate use since February of this year. For the second application, a patent has been filed on December last year, and for the 2 options, we think that there are 2 potential commercialization options because of the nature of the molecule, and also because of the result that we have. So it can be sold as a drug and also as a nutraceutical. And in both cases, we think that we have competitive advantages because of the mechanisms that we are describing, the outcomes that we have in mice, and also because of the price of beta-hydroxylic acid, because it's a very cheap compound. And both applications are applicable to the market for rare diseases, overweight, obesity, hepatic steatosis, type 2 diabetes, and also metabolic syndrome. And if we introduce this treatment in the clinic, of course, we believe that it would have an important socioeconomic impact. We have a plan for further development, and this includes some ongoing experiments, such as the transcriptomics and metabolomic analysis in different tissues, and for that we are collaborating with a group in Utrecht University in the Netherlands. Also, we are trying to define the minimal effective dose in both cases for primary CoQ deficiency and for the prevention and treatment of overweight and obesity. We are also trying to validate these results related with the treatment of obesity in other mouse models of obesity, insulin resistant, and metabolic syndrome. And in that case, we have some preliminary data in animals fed on the high-fat diet. And again, the effect of beta-salicylic acid is very powerful. It reduces clearly the weight of the animals, and that is because it reduced the content of the fat. And also we need to define the preventive versus curative effects. And this is something that we already have also some preliminary data that show that in mice that are already obese, we can reduce the weight in a couple of weeks by 10%. There are some other aspects that we have also planned to do, that is the validation of the anti-adipogenesis effects in human cells. Of course, we need to search for potential partners and investors, and that's why we are here. And we are planning to apply also to private and public calls for research grant and also for proof of concept grants. We need to develop a business plan and an exploitation strategy with an evaluation of the market. And of course, the last step will be the clinical validation. Finally, I would like to show you the team that is working in this research line. We are here in the Health Campus in Granada, in Spain, in the Biomedical Research Center, which is close to the nursing and medical school, also to the university hospital, some other research centers in the biomedical field, and also some biotech companies. And we are also close to the mountain named Sierra Nevada. And finally, I would like to thank the financial support of the Spanish Research Agency and also the Muscular Dystrophy Association. I'm happy here to answer any question that you may have.
Thank you. It's very nice talk. While we wait for questions to come in, and if you do have any questions, please type them into the Q&A box. Seems like there's a lot more than 2 applications here that this compound could be useful for. Have you tested it in any other models in what you've shown? If not, could you speculate a bit on what other diseases it could be useful for? I mean, just off the top of my head, I think neurodegenerative diseases could be useful if it's really targeting mitochondrial oxidative stress to some extent.
Yeah, we have to, at the beginning, we thought that it could be also useful in other models of primary mitochondrial diseases. And we tested this treatment in the NDUFS4 mouse model of Leigh syndrome. This is a mouse model that is available in Jackson Lab, and many people in the world has, but the treatment didn't work. So it cannot be used in general for mitochondrial diseases. And related with the second application, as I said before, we have used also, we are using now in a model of diet-induced obesity. And we are planning also to use in some other genetic models of obesity. Usually these models are related with a problem in the leptin pathway, so they eat and they are not satiated. So they are good models for obesity. So mainly, I think the therapeutic mechanisms in CoQ deficiency is related with the accumulation of the methoxyubiquinone. So we can apply this treatment in the cases of CoQ deficiency with accumulation of the methoxyubiquinone. And that happens in CoQ9 mutations also in CoQ7 mutation. But there are two other reports in the literature, very recent reports, that show also therapeutic effects in mouse models with mutations in CoQ6 and CoQ8b. And these mouse models of CoQ deficiency develop nephrotic syndrome. So in that articles, they didn't really explain the mechanism, and we think, because of our results that I show here, we think that it could be related with all metabolic changes that we observe with beta-sarcoglycanic acid in the kidney.
Very interesting. And can you talk a little bit about the dose that you chose? Is there any toxicity at really high doses?
Yeah, that is a very good question. We have tested, the result that I show you here are with beta-sarcoglycanic acid at a concentration of 0.33% in the food. That makes a dose of 0.5, 0.7 grams per kilogram per day, which is a really high dose. And with this dose, there is no toxicity. And you can see in the survival curve that I show you, there is no toxicity in either mutant or wild-type mice. But we have tested also a dose 3 times higher, which is a really very high dose, and there is toxicity. There is a renal and hepatic toxicity. That's why we are also trying to go down in the dose.
That makes sense. And you mentioned that you're treating 2 patients in Spain right now. Can you comment a little bit about how they're doing? Does it seem to be having a beneficial effect?
Well, as I say, they started the treatment on February. The only news that I have from the clinician is that they check at 1 month of the treatment, and there is some mild improvement, but it's very early to say something, I think. Yeah, we have just some visit to the clinician after 1 month of the treatment, so I think it's early to say something.
Sure, that's fair. And then I think we have time for one last question. So you mentioned you're looking for partners, investors. Do you have any plans to potentially spin out a company from this, or do you think you'll just look for out licensing?
We are open to both options. We are starting just to think about it. We are in contact with the Technology Transfer Office at the University of Granada. We are trying to promote, to show the results to investors, to pharma companies, and depending of how we see the environment, we will decide if we can go to a spin-off company at the university, or we can license the product to some company, to some investor, and try to go faster in the development of the treatment.
Great. Okay. Well, thank you very much, Dr. Lopez. This seems like very compelling work.
Great. Thank you to all of you.
Okay, while we're waiting for our slides to come up for the next speaker. There we go. So our next talk is titled New Strategy to Treat Sarcoglycanopathies, CFTR Correctors for Recovering Misfolded Proteins. And the talk will be given by Doriana Sandona from University of Padua. Dr. Sandona?
Hello, can you hear me?
Yes, you're a little quiet, but we can hear you.
Okay, so now I would like to start. Okay. So good morning to everyone from Padua. First of all, let me thank the MDA for the organization of this meeting, giving me the opportunity to share with you our story concerning the development of a new strategy to treat sarcoglycanopathy by repurposing compounds developed to treat cystic cystic fibrosis, so CFTR correctors. Let me spend a few words on sarcoglycanopathy. Maybe most of you well known the disease. Sarcoglycanopathies are 4 diseases altogether with very similar features. They are due to mutation in 4 genes coding for sarcoglycans that normally form a tetramer that is included in the dystrophin-associated protein complex that is located at the sarcolemma of striated muscle. And in that place is playing a very important role in assuring stability to the sarcolemma against the stress that can be elicited during muscle contraction. The disease belongs to the family of limb girdle muscular dystrophy because the most affected muscle are those of the proximal musculature of the upper and lower limbs. Sarcoglycanopathies are very severe disease with an early onset. They are highly disabling, fast and progressive, with sarcoglycanopathy to the wheelchair since the adolescence. But they are also heterogeneous. In other words, we can have also mild phenotype with the late onset and slow progression. The most important point is that sarcoglycanopathies are orphan diseases. Until now, there is no cure for these dystrophies. And so we try to search for a novel opportunity of therapy for this disease. And in this slide, let me show you, first of all, the fact that the majority of the mutations, the most commonly recognized mutation in sarcoglycanopathy are missense mutation or in-frame small deletion of one nucleotide or insertion. What happens when we have this kind of mutation? Remember that we are interested on this type of mutation. You well know that sarcoglycans are membrane protein, and so their biosynthesis goes through the endoplasmic reticulum, where they are folded, they are post-translationally modified, and they assemble into the final complex that is a tetramer. And only when the tetramer is formed, it can leave the endoplasmic reticulum and move to the sarcolemma. But when there is a missense mutation, for example, the protein is unable to gain the proper native form, native structure. And so it is recognized by the endoplasmic reticulum quality control system that recognizes the protein and delivers back into the cytosol the protein. And during the dislocation, there is also the modification of the proteins by the addition of a polyubiquitin chain. This is a mark for the final degradation through the proteasome. Of course, you can understand that this can lead to a loss of function. We have a reduced amount of the mutated protein that cannot form a proper tetramer. And so we have a reduction amount of sarcoglycans at the sarcolemma. This can increase the fragility of the sarcolemma, can lead to damage, and at the end to the progressive muscle degeneration. Of course, you can imagine that this mechanism is not limited to sarcoglycanopathy, but there are many other diseases in which we have this kind of loss of function because the protein is prematurely eliminated by the cell. And among these, one very famous
Sorry about that, folks. Looks like we lost Dr. Sandona. Hopefully we can get her back in just a minute, so stand by. Sorry about that. Okay, in the interest of time, maybe we'll just try to move on to the next speaker. Dr. Wichterle, are you there? I think I see you on.
Yep, I'm ready.
Okay, wonderful. We will just regroup here and move on to your slides. Sorry, we'll be starting a few minutes early, but hopefully we can get Dr. Sandona back and move back into her talk after we finish with you. Okay, so our next speaker will be Dr. Henrik Wichterle from Columbia University, who will tell us about Prociton, a new investigational drug for ALS, which protects motor neurons from misfolded proteins. Dr. Wichterle.
Thank you, Jonathan. Good morning, everyone. And so I'll tell you about a new investigational drug that we developed at Columbia University with support from Project ALS Foundation. And this was effort that was a collaborative effort between my lab and the lab of Brian Stockwell, also at Columbia University. So I'm sure that I don't have to tell you about the ALS, uniformly fatal neurodegenerative disease, which is caused by motor neuron degeneration that leads to muscle atrophy and paralysis. And at this point, there are approximately 30,000 people living with ALS in the United States. And typically they face progressive deterioration and death within 2 to 5 years of diagnosis. The tragedy of ALS is that we have currently no effective treatment for the disease. There are only 2 FDA-approved drugs, Riluzole and Edaravone, that offer very modest effects. And I think this is in part due to complexity of this disease. Although it has been described 150 years ago, we only recently realized that the disease is, in most cases, it's sporadic disease, but in the 10 or 15% of familial cases, the disease is caused by mutations in more than 20 different genes. Moreover, the disease, although it starts by motor neuron degeneration, it's more complex, and at later stages of disease, it also involves other cells such as astrocytes and microglia and inflammatory reaction in the spinal cord that further exacerbate the disease. Now, in terms of the drug development for ALS, I think one of the challenges is, as I mentioned, the heterogeneity of the disease, and the other is that many drugs that are FDA approved are developed under conditions that do not really make them CNS penetrant. On the positive note, ALS, I think, is far advanced in among other neurodegenerative diseases because it was one of the first diseases that was effectively modeled using stem cell derived motor neurons in vitro. And so that's the advantage, or that's the system that I would like to describe to you now. Although I mentioned that ALS is a highly heterogeneous disease, there is actually a shared common pathological feature that can be detected in many patients, both with familial ALS, but also with sporadic ALS. And that's actually accumulation of misfolded proteins that leads to ER stress response. Now, ER stress response is initially protective. Basically, it's a response system that helps the cell to cope with unfolded proteins. But if the protein misfolding is not successfully resolved, eventually the cells undergo cell death. And so we took advantage of this pathway and asked whether motor neurons are selectively sensitive to accumulation of misfolded proteins by triggering protein misfolding with several small molecule pharmacological treatments, such as cyclopyrazonic acid, tapsigargin, or tunicamycin, all of which were shown to induce protein misfolding. And here on this image, you can see actually our stem cell derived motor neurons that were co-cultured with stem cell derived spinal interneurons. So motor neurons are green and interneurons are red. And you can see that when we induce ER stress, it's actually motor neurons, the green cells, that are preferentially dying in these conditions, suggesting that motor neurons are highly sensitive to activation of these ER stress pathways. Now we can recapitulate this very effectively, not only with healthy motor neurons that are sensitive to ER stress, but when on top of that, the motor neurons also carry a mutation in one of the genes that causes ALS, these cells are even more sensitive to motor neuron degeneration, as you see on the lower right panel, where very few of ALS motor neurons survive addition of this ER stress inducing compounds. So we took advantage of this ER stress motor neuron degeneration to identify compounds that would protect motor neurons from these stressors. We designed this screen in 96 well plate, where we treated motor neurons with cyclopyrazonic acid and then added increasing concentrations of small molecule library compounds. And in these studies, so here you see that this is the survival of stressed motor neurons that were treated with cyclopyrazonic acid, and these are the control cells treated just with DMSO. Now you can appreciate that there is a significant number of compounds that were able to completely prevent motor neuron degeneration under these stressful conditions. One of these compounds, Kenpulon, was recently identified in different stress screens for motor neurons as a protective compound. But this compound has very poor pharmacokinetic properties, and so was never developed for in vivo assays. So in our work, we followed up on a series of small molecules that we identified that belong to different class of chemical compounds. And that also turn out to be highly protective for motor neurons. And we focus on one of these molecules called URMC99 that was developed as MAP3 kinase inhibitor and showed a very protective or excellent ability to protect motor neurons from ER stress. Now this compound is very protective, but it's also highly unstable. And so we thought that it would not be a great drug to take for further development. And so we joined forces with Brent Stockwell's lab at Columbia, who synthesized now over 60 different analogs of the initial compound that we discovered. And we focused on one of these that we termed Prosetin, that has excellent pharmacokinetic properties. So first of all, in our survival assays, we showed that Prosetin is actually much more effective in protecting motor neurons than the parental drug. But then importantly, Prosetin is also highly stable. In microsomal, liver microsomal stability assay, we observed that Prosetin has more than 2-hour half-life, which is very strong stability. And when we dosed the drug actually in drinking water, so it's orally bioavailable, we observed that the drug actually accumulates at higher concentrations in brain compared to plasma. So you can see that the effective dose is already reached around 7 milligrams per kilogram per day doses, and exceeds the effective dose in brain, at least at 10 milligrams per kilogram dosing. Now, since this drug is highly penetrant and accesses CNS, we were concerned that the drug might be actually accumulating in the lipid or highly hydrophobic regions in the CNS, like white matter. And so we performed DESI-MS analysis that allows us to actually identify the locations of Procitin in the spinal cord. So this is the outline of the spinal cord of a mouse that was treated with Procitin. The white matter is here on the outside, and here inside is the gray matter with motor neurons shown here in the ventral horn. And as you can see, Procitin actually accumulates within gray matter, and we believe that we can actually detect it inside motor neurons, suggesting that the drug is reaching the target cell. So I mentioned that the initial drug that we started with was designated as MAP3 kinase inhibitor. And since we synthesized over 60 different compounds, 60 different analogs, we were in a great position to ask whether inhibition of MAP3 kinase is really the mechanism of action of Procitin. And so we plotted here the kinase activity. So the closer to the zero, the more inhibitory the compound is, versus motor neuron protection for MAP3 kinases. And we surprisingly found that actually the survival does not really strongly correlate with effectiveness of the compounds for that inhibition of MAP3 kinases. Instead, we found that the compound actually very strongly correlates, or the activity very strongly correlates with inhibition of MAP4 kinases, one step above MAP3 kinases. So effectively, we repurposed this MAP3 kinase inhibitor to inhibit different kinases within the pathway MAP4 kinases. And we believe that that's the therapeutic target in terms of protecting motor neurons in ER stress. So since this drug is stable and orally bioavailable, we were able also to start testing the drug in ALS mouse models. And so for this, we use the SOD1 mutant mice that usually die within 120 to 140 days. And here you can see that we were dosing the mice continuously from day 40 all the way to 120, or to the end stage, with Procitin in drinking water. The Procitin was non-toxic. We did not lose any mice. And importantly, we see that Procitin has actually very strong effects at the early stages of the disease. So we see that Procitin-treated mice in blue here actually do not lose weight as quickly as the control, the ALS mice. They sort of stay within the control range for 2 to 3 weeks longer than ALS mice. And finally, also, we tested the strength of these mice and show that, again, Procitin significantly delays the weakness of the mice measured by inverted grid assay. So with these encouraging results, we decided to continue on the development of this drug and apply for IND status to FDA. And for that, of course, we had to actually outsource a lot of the work to certified laboratories. So we worked with Charles River Laboratories on IND enabling safety and toxicity studies that were all already completed. We also partner with Patheon, which is a chemistry development company for synthesis of the drug. And we have now developed basically protocols for synthesizing pharmaceutical quality Procitin at large quantities in Patheon. And we are at the final stages of preparation of FDA application that should be submitted within the next 2 weeks. So the next steps for us is to design a clinical study. And so we worked together with FDA to develop the fastest way to establish safety, dose, and PK and PD for Procitin in people with ALS. And for that, actually, we decided to start with testing Procitin in healthy volunteers in phase 1A and 1B, which would be single ascending dose studies and multiple ascending dose studies. And then only in phase 1C, we will administer Procitin to ALS patients, where we will confirm the dosing and safety in patient population. So with this, our next step, obviously, is identification of a partner for clinical development of this drug. So we are looking actively for licensing partners who would be willing to participate in the phase 1 through 3 clinical trials. We believe that Procitin has many properties that make it a great candidate for development of the drug for ALS treatments. It is highly brain penetrant. It's potent, stable, orally bioavailable, and it's efficacious in both in vitro cellular assays that we developed, as well as mouse models of ALS. Importantly, the drug also was designated as orphan drug. It received orphan drug status from FDA. And so it has a potential for expedited clinical development, given the high unmet medical need in patients with ALS. And so with this, I just wanted to acknowledge the amazing team that we put together, the researchers that worked on the development of the drug, the consultants that helped us to prepare the IND application to FDA, and of course the Project ALS that funded this effort. I would be happy to take any questions.
Thank you, Dr. Wickerle. I think we have time for 1 or 2 questions before I have to turn it back over to Dr. Sandona. So we have one question here in the Q&A. So success in ALS clinical trials and drug development today mandates the need for a pharmacodynamic marker, some evidence, ideally brain-based like CSF or a plasma marker that accurately reflects target engagement in the patient's brain and not mice. There's too many failed trials in ALS that lack such critical tools. Do you have such a marker?
Yeah, so we are working also on marker development for Procitin. We have identified one protein which we see is targeted, or its levels are reduced in plasma in mice, and so we would definitely be interested to develop this as a marker for, or biomarker for, effectiveness of Procitin in ALS patients. So we are working on that, and we would like to include it as part of the clinical trials.
Great. And obviously, one other question, MAP kinases obviously have a lot of different targets. They phosphorylate a lot of proteins. Have you looked at off-target effects? Do you have any concerns about specificity?
Yeah, it was definitely on top of my mind when we started these animal studies, and I have to say that we had no idea whether the drug will turn out to be completely toxic. So it's we are encouraged by the fact that so I showed you that at 10 mg per kg we get already over the effective dose that we see in vitro in brain. We increase these doses all the way to 30 mg per kg in mice, and we have not seen any adverse effects in those animals either. So it is kind of surprising considering how kinases are important, but maybe it's because the drug is relatively quickly cleared from periphery and accumulates a higher concentration in CNS. Maybe that's why we don't see much of off-target or side effects.
Okay, and one last question. So you looked at Procitin in a very aggressive ALS mouse model, the SCD1 G93A, and you saw it looked like good effects, but they tended to taper off as the animals got to their usual typical survival end date. Do you have plans to test in another ALS model, maybe TDP-43 model?
Yeah, so we are now actually testing the drug also in a FAS model of ALS, which is much milder and does not show us aggressive, progressive nature. And so I don't have the data yet, but we are definitely looking into it. And I agree that the SOD model is probably not the best model because of its aggressive nature. And in the end, I think it just reflects the fact that Procitin is probably not going to be the only treatment that will be used in ALS patients, but ALS treatment will really require a combinatorial therapy where different drugs will be targeting different stages of the disease. So we view Procitin as the drug that is really attacking the early motor neuron vulnerability, but it's probably not helpful in the later stages of the inflammatory response.
Great. Thank you, Dr. Rickerly. That was a very nice presentation.
Welcome.
All right, Dr. Sandona, are you there? We're going to try to go back to the spot in your slides where you left off. So just give us a minute to get back.
Okay, I'm here.
Okay, great. We'll try to keep you on this time.
Yes, can you hear me now? Sorry for this problem.
No problem. Part of the technology.
I'm very sorry.
No problem at all. Just give us 1 minute. We'll get back to where you left off.
Yes, thanks. Okay, here we are. We were talking about the fact that sarcoglycanopathy shares with other diseases the same pathogenic mechanism when missense mutation cause the disease. And so we decided to focus on cystic fibrosis and of the many compounds that have been developed to treat cystic fibrosis. And we decided to check mainly the CFTR correctors that are acting on the CFTR protein that present folding problems and trafficking problem like our sarcoglycan. And therefore we decided to try them. And as you can see here in this part of the slide, that corresponded to the treatment of a cell model of sarcoglycanopathy in which we expressed a mutant form of the alpha sarcoglycan. You can see that without any treatment, we don't see much sarcoglycan on the surface of the cells. On the contrary, on what happens when we express the wild type alpha sarcoglycan. However, if we treat these cells with the CFTR correctors, and we use several of them, you can see that there is again the relocalization of the sarcoglycan on the surface of the cells. This is an immunofluorescence using an antibody recognizing an extracellular epitope of alpha sarcoglycan. And so we can say that by using this small molecule, we have the opportunity to relocalize, to improve again the traffic of the sarcoglycan mutant, in this case, on the surface. Of course, this is a model, and therefore we decided to use something more close to the physiology. And this was possible because we had the chance to use primary myogenic cells from a patient with alpha sarcoglycanopathy. And we were able to grow and to differentiate these cells. And as you can see here in these myotubes, that is the first step of muscle differentiation, we can see almost nothing at the sarcolemma of both alpha sarcoglycan, that is the mutated protein, but also on the wild type partner, delta sarcoglycan. On the contrary, when we treat with the different CFTR correctors, you can observe the relocalization of the protein on the surface. So, and the fact that we observe the mutated and also the wild type partner means that the entire sarcoglycan complex is there. By the way, I cannot see you for question of time, but we also tested the strength of the sarcolemma in vitro, and we indeed observe an improvement of the stability of the sarcolemma upon the treatment. This treatment were of about 4 days of treatment. At this point, the proof of concept of the invention was obtained, but we need to pass to in vivo validation. And this was the very big issue of our story, because the animal model available were not suitable to treat with this compound, because the knock-in animal model, remember that we need to have a protein with a missense mutation. So the knock-in available failed to develop a dystrophic phenotype. And therefore, we decided to move towards a different strategy, exploiting the use of adeno-associated viruses able to transduce the muscle, in this case, of a knockout animal model, so the alpha-sarcoglycan knockout mouse. That is our background. In this background, where only 3 subunits are present, we introduce the human sequence of the alpha-sarcoglycan, either in the wild type form, and this is our control of the process, and or the mutated form. And we perform the viral infection very early, at 1-2 days after birth. And after 10 weeks, we analyze muscle. And as you can see here, these are the positive controls, so healthy phenotype. This is the background alpha-sarcoglycan null mouse. And this is the fluorescence coming from the sarcolemma of muscle from the humanized mice with the wild type alpha-sarcoglycan sequence, human alpha-sarcoglycan sequence. And you can see that it recovered the protein. However, if we inject the viral vector expressing the mutated form of a human alpha-sarcoglycan, we are unable to see the expression or very low expression. And also at the level of histology, we can still observe the dystrophic phenotype. So we decided to use this animal for the treatment in vivo. And here is our protocol. So the process of infection is the same. Starting from 7 weeks, we start the treatment with the compound by intraperitoneal injection. So it's a systemic injection with the most promising CFTR corrector as identified by in vitro experiment. And after 3 or 5 weeks, we analyze the effect. And here you can observe in this part of the slide that 5 weeks of treatment results in the recovery of the sarcoglycan complex at the level of the sarcolemma of this of the muscle, hind limb muscle of this animal. And also there is an improvement in the histology, very well resembling the wild type, the healthy phenotype of the wild type. So now we are doing the muscle functional analysis. So we are trying to evaluate the muscle force of this animal in vivo. And I can tell you that we have very, very, very good results. And so here we are, because we can say that maybe the identification of the lead compounds and its optimization was already obtained. We want to conclude the preclinical experiment to start at the end with the clinical validation of these compounds. And to do this, we need help for partners interested in funding or collaborating with us, both for concluding the analysis of muscle force, but of course also for testing different regimens of administration, identifying the minimal dose, testing different compounds, and also the combined administration of the compounds. Of course, performing pharmacokinetic and pharmacodynamics experiment. As you can see, some of these experiments are already ongoing, other are just planned. We want also to test these compounds on different diseases, because as I told you before, the very important point is that our proteins, one rescued, maintain its functionality. And we have very good data concerning sarcoglycanopathy, but we have also data concerning some mutation of the SERCA1 pump that is mutated in Brody myopathy. Of course, those mutations that do not affect the activity of the pump can be recovered by this system. And the patents that we have cover also this disease. But we have also preliminary data with these different disease that is again a rare disease, is not concerned in muscle problem, that is the Allan-Erdo-Dudley syndrome.
And Dr. Sendo, I'm sorry, we have to cut you off right there.
I would like just to show you the patents, and I would like to thanks who funded this research. Thank you very much.
Thank you, and sorry again about the technical difficulties. If anyone is interested in contacting Dr. Sendona, feel free to reach out to her or to MDA, we can make contact.
Okay, thank you.
Thank you. All right, our next speaker is not Dr. Richterle. We already heard from him, so we'll advance to the next presenter, which is Dr. Abbas Abdallah. Here we go. So Dr. Abbas Abdallah is from the University of Massachusetts Medical School and will tell us about novel siRNA muscle targeting platform, a potential treatment for FSHD. Dr. Abdallah.
Thank you. Good morning, and I really want to thank MDA for giving us this opportunity to talk about our novel siRNA muscle targeting platform, which has the potential to treat not only FSHD, but many other skeletal diseases. But before I talk about our platform, I want to acknowledge our amazing faculty who made this platform a reality. First, I want to start with Anastasia Kovorova, who is a leading expert in oligonucleotide biochemistry, Charlie Emerson, who has extensive experience in neuromuscular diseases, Larry Hayward, our neuromuscular physician scientist, and Oliver King, our computational biologist. So what is small interfering RNA or siRNA? In biology, the central dogma revolves around the fact that, sorry, just let me get my laser pointer. So in the central dogma revolves around DNA being transcribed into RNA, which in turn will be translated to proteins. However, the cell has different methods to fine-tune this process. One of them is using siRNA, which can bind this messenger RNA and target it for degradation, and thus no proteins is being made. Researchers quickly realized that this can be utilized to create on-demand gene silencing systems. All you need to do is create an siRNA molecule that targets the specific sequence that you want, tag it with a conjugate. That conjugate will allow it to attach to specific receptor in the target organ or tissue that you would like to target. And so what happens, it goes internally, does the same function, and thus control gene expression. siRNA is not a recent phenomena. In fact, it has been discovered in 1998 by Craig Mello and Andrew Fire, and their work led them to share the Nobel Prize in 2006. However, it took us about 20 years between the discovery of siRNA and the ability to make it clinically relevant. It turned out that RNA by nature is very unstable, and what happens, it degrades very quickly. In addition, finding those conjugates that could deliver to specific organs turned out to be much harder than we thought. Nevertheless, over the past 20 years, scientists improved the stability of RNA, which now can last for several months, and a new conjugate called GalNAc, which allows for accumulation of siRNA in the liver, was discovered. This culminated in 2018 with the approval of the first siRNA drug by the FDA. And then it was followed by 2 others from now between 2018 and now. In addition, there are 7 drugs in phase 3 clinical trial right now, and the expectation is that this trend will continue and expand. And while most of these drugs are targeting either liver target or blood and focus on rare disease, there's a new one called inclisiran, which will target high cholesterol level in blood. And this one is poised to open the door for siRNA to become a mainstream modality in treating diseases. So in short, while the stability of siRNA and hepatic delivery has been solved, there remains a huge need in the extrahepatic delivery. I mean, pretty much everywhere else, we don't have access to any organs. And then that's why at UMass, we've been actively developing delivery platforms that will help us address that huge unmet need. One of our great success stories is developing a CNS delivery platform. And this was done by 3 of our distinguished RNA Therapeutic Institute, Anastasia Kuvarova, Craig Mello, and Neil Aronen. This platform provides uniform, long-lasting, and functional siRNA delivery to the brain and the spinal cord, including in deep regions like the caudate and the putamen. If you look at this figure, you don't even have to look under the microscope. You can see the difference between delivering siRNA using our platform and delivering siRNA without our platform. And if you look under the microscope, you can appreciate the uniform distribution in non-human primate animal models. And this platform has the ability to treat many diseases, any neurological disease of known origin and known genetic pathway that we know that we need to target can be targeted by this pathway. And this led to the creation of Atalanta Therapeutics, which to date has been successful in raising $110 million in Series A from F Prime and through collaboration with Biogen and Genentech. Now that we have conquered the the CNS delivery, our focus has shifted to create delivery platforms for other organs. Specifically, we were very interested in targeting the muscle, but how can we achieve that? If you look here, siRNA is basically made of its backbone, which determines the structure, sequence selection, stability, and toxicity. However, you can add a lot of things to it, like a PK modifier, which affects the rate of kidney clearance, a linker, which affects endosomal escape, and a conjugate, which affects delivery and accumulation, which is honestly the most important part when you want to target a new organ. And thus, we screened for conjugates that can accumulate in the muscle. And we found out that there's a molecule called DCA, when conjugated to siRNA, can accumulate up to 5% of the injected dose in heart and skeletal muscle. And while 5% may seem like a low number, it's actually pretty significant, considering that once you do systemic injection of siRNA, most of the dose will be excreted out by the kidneys, and the rest will be absorbed by the liver itself, just because of how the liver functions. And we know from previous studies that accumulation doesn't really always correlate with functional delivery. And thus we wanted to test whether our platform works biochemically and phenotypically. We knew that there's a mutation, and we know that myostatin puts a brake on muscle growth, and a mutation in myostatin can lead to loss of function and produces these enlarged muscle size and weight in affected animals, as you can see here. We wanted to see if we can do the same thing to our mice. So without going into too much details, we screened for several siRNAs sequences that can target myostatin, and we tested its impact on muscle growth. And as you can see here, by single dose, we see almost 20 to 25% increase in the muscle size. And that increase is dose dependent. So as we increase the dose, that muscle size increases as well. And we can even detect it after 1 month of injection. And so that tells us that our platform is functional, not only in being able to deliver siRNA, but it is functional siRNA. And so while targeting myostatin could be important in certain conditions like muscle wasting in cancer patients, or even potentially in acute muscle injury, we wanted to determine whether our system can work in muscular dystrophies, knowing how much of a huge unmet need there is there. And as I mentioned before, once you develop that scaffold, which is the conjugate and your siRNA, all you have to do is literally change a sequence of about 20 nucleotides, and then you can use it to deliver or target other genes without having to do all the work that you've already done. And so we shifted our focus to FSHD, which is a muscular dystrophy with no cure. FSHD is a genetic disorder caused by the misexpression of pathogenic DUX4. And here I'm specifically talking about FSHD1. It affects nearly 1 in 20,000 and as high as 1 in 8,000 people. It causes muscle weakness and atrophy, and specifically in the face, shoulder, upper arm, and legs, but also potentially in other muscles. The symptom onset typically starts in teenage years, but is also very variable, and the current supportive treatments do not address the root cause of this disease. So the question is, how does DUX4 cause FSHD? DUX4 is actually a transcription factor, and it's only expressed in early embryonic stages and in testes in males. It forms these, what we call these D4Z4 repeats, and in the general population, those D4Z4 repeats can reach somewhere between 11 to 150 repeats. And what happens is it causes the compaction, the DNA to become compact, and thus it silences DUX4 expression in mature muscle tissue. And as you can see here, if the DUX4 is not expressed, muscle cells survive normally. However, in patients, or specifically in FSHD patients, that D4Z4 repeat array is reduced to about 10 units or fewer. And thus, it increases the accessibility to the DUX4 and allow its pathogenic transcription. Once that happens, DUX4 as a transcription factor activates hundreds of other target genes, which in methods we don't completely understand, causes cell death. And so we asked ourselves, can we efficiently deliver our siRNA to reduce pathogenic DUX4 production? And in this case, to test our siRNA on FSHD in a clinically relevant setting, we created a xenograft mouse model where we kill chemically through BACL2 mice muscle cells, but we maintain the matrix, and then we inject xenograft primary myoblast from FSHD patients into the mice and allow them to grow for about 2 weeks. After which we can use our DCA conjugated non-template control or the DCA with our siRNA, which in this case we're going to label as DU01. We know that this works pretty well because if we look at the place of injury, we notice that all of the new muscle growth is human muscle, as indicated by lamin and collagen. And then when we looked at the level of DUX4, we noticed that we were able to reduce in single dose injection, about 45% of the expression of DUX4. But not only we reduced the expression of DUX4, but we also looked at other downstream targets of DUX4, and we saw that we can achieve reduction between 40 to 45%, again, with a single dose. And so to sum up what we have so far, is we have a muscle delivery platform that's ready to partner. That has been based on our proprietary backbone modifications and full chemical stabilization, which we have multiple IP dockets to protect, our proprietary cleavable linker technology, in addition to novel chemical conjugates that allow for muscle accumulation. And through creating a new siRNA muscle delivery platform, now we can target not only FSHD, but many other neuromuscular diseases that could be of interest. And we have tested this platform in 2 different ways. One, we tested its functional delivery in mice by targeting myostatin, and as you can see, we can get up to 60% increase in muscle size just in 1 week. In addition, we screened for novel siRNA sequences that could target DUX4, and we created a new model, which is a cell xenograft model for FSHD1, and that showed significant reduction of DUX4 and its biomarkers once we used our siRNA. So what are we looking for? We are looking for a partner to either license the FSHD asset and help move it into the clinic, or to an investor who's interested in creating a new company around muscle delivery platform to target FSHD and other muscular dystrophies moving forward. Thank you, and I'm happy to take any questions.
Thank you, Dr. Abdallah. We have time for one or two questions. We're a little over time, but it's through no fault of your own. So I'm wondering if you can give us a little hint of what other indications you're thinking of for this technology platform.
Right. So one of the other obvious indications is DM1 as well. So DM1 is another muscular dystrophy that can, we understand the molecular pathway and we can go after it. Another indication that we don't talk a lot about, but you know, cancer patients who are subjected to chemotherapy undergo muscle wasting. And this could be one thing that could help in restoring that aspect of, you know, of their body.
Great. I think in the interest of time, we'll have to move on to the next speaker, but thank you very much for the presentation. Right. While we're waiting for your slides to come up, I will introduce Dr. Thomas Sukna from SUNY Buffalo, who will tell us about GSM-TX4 treatment for muscular dystrophy. Dr. Sukna.
Hello, can you hear me?
Yes.
Okay. Let's see if this works here. It's not moving forward.
Go ahead and click on the screen first. That may help.
Oh.
And sometimes there is a bit of a delay.
Can I turn off the, I think that's what had given me a problem before, turning off the.
Yes, exactly. You can turn off the laser pointer. So if you just hover over your screen, so click back into the presentation. Yes, and just turn that off there. Perfect.
Ah, there we go. Okay, thanks. And good morning, everyone. And I'm glad you gave me the opportunity to talk about GSM-TX4, our novel treatment for muscular dystrophy, we believe. So this presentation is about first-in-class compound GSM-TX4 and its treatment for muscular dystrophy, and is possibly an adjuvant for gene therapies. Our strategy is based on the fact that mechanical dysfunction is at the root of many forms of muscular dystrophy, and this mechanically unprotected membranes lead to abnormal opening of mechanically sensitive channels, which are called MSCs for short. This creates a persistent pathological calcium leak that kills skeletal and cardiac muscle cells. And so calcium leak is the primary early contributor to MD pathology in many cases. And various calcium channel inhibitors have been tested on MD patients, but with little success. That's because while calcium is a ubiquitous signaling agent, it is not all calcium sources are the same, and many calcium sources affect others. So I will show you the evidence that supports targeting MSCs as the most effective therapeutic intervention, we believe, for and with the least side effects. So GSM-TX4 is a peptide inhibitor of MSCs. We found it was isolated from spiders, and we have solved its structure. It is a wedge-shaped structure. It is a wedge-shaped amphipath with a ring of positive charges, making it buoyant on the membrane surface. It associates with lipids surrounding the MSCs, like piezo and trip channels, and these are novel targets for MD. In normal cell membranes, tension sensors on the channel cause the channel to open only infrequently, allowing calcium to enter. In dystrophic muscle cells, this happens too often and toxic calcium levels enter the cell. GSM-TX4 shields these channels from excess membrane stress so that they remain closed and calcium levels remain manageable. And GSM-TX4's unusual shape and charge distribution make it difficult to duplicate. So there are currently no equals to this compound. Because GSM-TX4 binds to the local lipids around the channel and not directly to the channel in a lock and key fashion, we were able to synthesize it from D amino acids and make a mirror image of it. And its function is identical to the natural form. The advantage of the D amino acid peptides are that they are resistant to enzymatic digestion, giving them longer half-lives. So does targeting piezo channels protect dystrophic muscle? Well, we tested GSMTX4 in normal and dystrophic mice for 6 weeks, dosed 3 times per week, and we measured injury susceptibility and muscle growth. At 6 weeks, we exposed the skeletal muscle to stretch contractions, which are very damaging to the muscle. And GSMTX4 protected against loss of muscle contraction strength. And in addition, there was a 20% increase in muscle weight. In a second study, we examined GSMTX4 protection of cardiac muscle in a model of ischemia or loss of blood flow to the left ventricle. And GSMTX4 reduced infarct size by up to 25%, and it improved hemodynamic function, increased ejection volume and contraction force, and decreased ventricular tachycardia, leading to deadly arrhythmias. We showed that the deformed GSMTX4 has a 7-day half-life, which is quite long for small peptides, likely due to its resistance to enzymatic degradation. And no animal death or abnormal behavior was observed in either of these studies. Over the past 50 years, there's been many potential treatments for DMD that have been tested with little success, and correcting the mutation by gene therapy has become recognized as the only viable cure. This can be accomplished by either genetically modifying stem cells and placing them back in the patient, or by direct injection of gene therapies into the treatments into the patient. And several drugs in the direct injection method have been approved by the FDA and fall under the 2 main categories of AON, or antisense oligonucleotide mediated exon skipping, or adenovirus mediated gene delivery. But both of these, the problem is that these gene therapies are currently very inefficient and pose a variety of disadvantages shown here. Both therapies would benefit from muscle protection during the treatment period, especially cardiac muscle. And we have already demonstrated this protection for GSMTX4. But there is evidence that these therapies can be potentiated by reducing calcium, a calcium leak, and by reducing the elevated inflammatory response that occurs. In the next 2 slides, I will explain to you why GSMTX4 may improve all these limiting factors. First, recent studies show that excess calcium inhibits dystrophin expression induced by gene therapies. Dr. Miceli's group at UCLA, our collaborator, showed that improving calcium homeostasis leads to exon skipping in a human muscular dystrophy cell model. They used dantrolene, an inhibitor of ryanodine receptor channels, which mediate calcium release internally to promote calcium balance. And dantrolene plus antisense oligonucleotide produced a 1.5 to 2.5% increase in dystrophin RNA versus a 1% increase for AON alone. A company, Armgo Pharmaceuticals, makes a compound called Rycals, which is a drug that stabilizes ryanodine receptors. And these have received significant funding to test Rycals as adjuvants to gene therapy in DMD, demonstrating the interest in this avenue of treatment. However, calcium leak comes from multiple sources, as I was saying before, and MSCs are early contributors that affect downstream calcium leak to ryanodine from ryanodine receptors. GSMTX4 has shown to reduce the calcium overload and protect contraction strength in dystrophic muscle cells. It is more effective than any other inhibitor and likely reduces it is more effective than any other inhibitor and is as effective as removing all the calcium from the cell. Importantly, reducing external calcium leak into the cell also reduces calcium leak from internal stores. And I show this you can see the blinking green over here shows the internal calcium leaks by ryanodine receptors in cardiomyocytes treated by hypoxia is depleted and contraction strength is reduced. But in the presence of GSMTX4, purple blinking line, calcium release and contraction strength were protected, showing that blocking external calcium leak can stabilize ryanodine receptors. This supports our contention that GSMTX4 will improve calcium handling by multiple mechanisms and enhances exon skipping. And we currently have a pilot funding to test this hypothesis. Further, recent high-profile studies explain why GSMTX4 could be used as a standalone and as an effective adjuvant to genetic therapies for DMD. One study looked at the role of piezo channels in cardiomyopathy. Dilated cardiomyopathy is a primary cause of heart failure in DMD and Becker's patients. Piezo channel dysfunction leading to increased calcium is at the root of DCM pathology, or dilated cardiomyopathy. Our study in cardiac ischemia and this new evidence suggests that GSMTX4 could be a key to stemming DCM. In other studies, piezo channels were shown to be important in mediators of the immune response to mechanical stress. Inflammation is an important contributor factor to DMD pathology, and it is a significant side effect of the adenovirus-mediated gene therapies, which can induce inflammatory immune responses. Piezo is involved in activating the innate immune response, and we showed that GSMTX4 suppresses the inflammatory kinases released during during a heart being in stress. And this and other recent studies on piezo strongly supports the continued development of GSMTX4 for gene therapy and DMD. Development opportunities for GSMTX4 include, there are multiple opportunities. The first one is there's a pilot study that we're doing with the University of Buffalo. We are seeking funding for and testing GSMTX4 as a gene therapy adjuvant. We are using the in vitro human muscle model developed by Dr. Carrie Miceli's lab at UCLA, and she is collaborating with us to help us develop that in the lab. And we are also collaborating with Dr. Christopher Ward from the University of Maryland, whose company Myologica will be conducting the mouse demonstration study. We are hoping to gain funding for that. And the question will be, will calcium stabilization by GSMTX4 boost dystrophin expression by exon skipping therapies? And the second one here, we're looking at, we're using a pig model of heart failure, and we are testing to see if GSMTX4 can limit remodeling and improve vascular perfusion of cardiac tissue during pressure overload. And with additional funding, we can extend this study to include experiments designed to include induced dilated cardiomyopathy, which occurs in DMD patients. Both of these studies will lead to new IP and patent protection. Other potential therapeutic avenues we would like to pursue, and we have relationships with specialists in those fields to help us succeed, include pancreatic cancer with Roswell Park Cancer Institute, which is right here at UB, and we have people that we know right there who would help us with that. Joint inflammation and degradation with Dr. Wasil Lee at the University of Rochester, and stroke and astrogliosis with Drs. Lou Quinn and Mikhail Pleknikov at the University of Buffalo. In all these disease states, increased piezo ion channel activity contributes to the pathology. And lastly, I'd just like to talk to you a few highlights of the key elements of the current status of GSMTX4 development. The current patent is licensed through Tonus Therapeutics, our biopharmaceutical startup. And we also have orphan drug status, providing an additional 7 years of exclusivity for use in DMD treatments. We have a long-standing relationship with our peptide supplier CS Bio for producing GSMTX4 and new analogs. We met with the TACT NMD advisory panel, whose primary recommendation was that we test GSMTX4 on non-normal cardiac function in larger animal model. And we tested 3 concentrations of GSMTX4 on normal free-roaming ferrets and showed no change in heart function. The D form of GSMTX4 shows a 7-day half-life, likely due to its lower enzymatic susceptibility. And the primary target of GSMTX4 appears to be piezo ion channels, a high-profile novel target for therapeutics. Yeah, last one. And so I'd just like to introduce our collaborative team. Our lab at UB is composed of 3 PIs. Our expertise is in cellular biomechanics and investigating pathology stemming from dysregulation of mechanical sensors. We have co-founded an early stage biopharma company, Tonus Therapeutics, and as the lead in obtaining FDA approval and to develop a new IP, but we need a lot of help financially with this And we're considering all avenues of licensing or sub-licensing, whichever works best. We would be interested in talks with anyone. And our collaborative team also include, as I spoke before, Dr. Christopher Ward, Dr. Carrie Miceli, and Dr. Brian Weil at the University of Buffalo, who is a protege of Dr. John Canty here at UB, a world-renowned cardiologist. And so in summary, I'd just like to say piezo channel dysfunction is a critical and novel pathological target in multiple disease states, and in GSMTX4 is the most effective means of blunting piezo dysfunction in DMD and may be the best agent for boosting gene therapies. So thank you.
Thank you very much. I think we're right at the 15-minute mark here, but we have one question came in. So does TONUS only have DMD or all muscular dystrophies?
So right now it's I believe licensed for DMD, but it could be used also in other it could be used for all muscular dystrophies, is I think the way the patent is set up right now.
Okay, thank you, Dr. Suknia. I think we are out of time, unfortunately. Now we're going to switch gears to the next session titled Disease-Focused Venture Philanthropy and Venture Capital Funds. Our first speaker in the session is Dr. Liana Orlando from Cure Duchenne, who will tell us about their venture philanthropy program. Dr. Orlando?
Thanks. So I cannot show my video now. There we go. Thank you. So thank you, Jonathan. Thanks, everyone, for the invitation today. Okay, the slides. So Cure Duchenne is an organization that has programs and events for patients and families and medical education, advocacy, but today I'll just focus on Cure Duchenne Ventures, which is the way we help advance research and therapy development. Our investments are Duchenne and Becker specific, meaning we're not going to invest in something just to make money if it's not going to address a need in those diseases. But it does include platform technologies to be applied for DMD and BMD. And in fact, in that case, we think our funding can actually prioritize Duchenne and Becker as one of the lead indications. And with that, and often with these platform technologies, what we're doing is we're thinking about the drugs currently on the market, as well as what's in clinical trials, and where are going to be the gaps, or what communities of individuals are those treatments not going to serve. So we sort of have to take sort of a 3 or 5 year approach and think through where those gaps and where the needs are going to be, and start working on those solutions today. So Cure Duchenne was started in 2003 and started supporting research both through grants, shown with the yellow stars, and investments, shown with the red stars. And it wasn't just grants to academics and investments to companies. As I noted, some therapy development in companies which were supported by grants in the early days. But I'll draw your attention to this investment in Prosenza in 2004, which was to support their exon skipping program in Duchenne. And that ended up being very fruitful, because as the science progressed and the work went on, later BioMarin acquired Prosenza in 2014. And so with that, there were some proceeds which came back to Cure Duchenne, and Cure Duchenne established Cure Duchenne Ventures as a separate, distinct entity, part of Cure Duchenne. And that really also marked the switch from doing a mix of grants and investments to really focusing on investments. And so the funds from Prosenza were deployed for the next series of investments, and including one, Exonics, which I'll note here because I'll talk about it in a later slide. And proceeds from Exonics actually then are being deployed even still to this day. And you can see this uptick in activity in more recent years. And that's due to the Duchenne drug development accelerating in the community at large. And so our activity is accelerated as well, with us doing 4 investments for each of the last 3 years. And we're on pace to meet or exceed that this year as well. So I already talked about Prosenza. The first investment was in 2004 with 1.3 million in seed funding. I think it was 2 million over the entire course of CureDuchenne's investment. And then later when BioMarin acquired Prosenza, the funds from that came to CureDuchenne and supported the next series of investments, which included Exonics Therapeutics. So this was the first time CureDuchenne co-founded a company, which it did together with Eric Olson at UT Southwestern. This was using CRISPR-Cas9 to gene edit dystrophin. And as the work progressed, CureDuchenne provided all of the seed funding. There was later a Series A raise, but as the work progressed and Vertex became more interested, they eventually acquired Exonics in 2019. And we're excited, for the work to be progressing at Vertex. So all in all, since CureDuchenne was founded, there have been 23 investments, 20 of which are equity investments. We're open to all structures, but it just turns out most of them end up being equity investments. And as I showed on that slide with all the red stars at the end of the time arrow, the activity has really been increasing with 15 equity investments in the last 5 years. And that represents more than $11 million that CureDuchenne has put into Duchenne therapy development. The size of the investments can vary depending on the need, the stage of the company. Is it a proof of concept in an animal model so it's a couple $100,000, or larger investment to get them to an inflection point of 2 million. So it's really on a case-by-case basis. We're looking at over 100 leads every year, as well as meeting with companies and groups that we've met with in previous years to get updates and see how things are progressing. And that's because even when we don't provide funding, we're always providing strategic advice in terms of what would de-risk the project from our perspective, what might attract larger VC funding. The opportunities generated come from connections in both the investment and the scientific communities. And what has really characterized the last couple of years is the number of investments that come to us from traditional venture capital partners who are putting together syndicates. And if Duchenne is on sort of the list of potential lead indications, very often they're coming to us because they know that we are looking at everything in the space and we have really specialized knowledge to evaluate and de-risk those early DMD programs. In terms of science, the lack of dystrophin being the root cause of the disease, we're of course looking at therapies that can improve the amount of dystrophin being expressed. But we recognize that there's many components to the disease process, and there's unmet need there that we really need to be looking at treating the whole disease. So in terms of dystrophin restoring therapies, there are exon skipping therapies which have already been approved and are on the market, but we think that we can do better, or some of these guys can do better. So, part of the problem is getting enough of the exon skipping oligonucleotides into the muscle cells, and not just skeletal muscle, but we really want to get it into the heart too, to replace dystrophin expression there. So these 4 companies that we've invested in all are aiming to do just that, with Avidity and Dyne using antibodies or antibody fragments to the transferrin 1 receptor to be able to increase uptake into muscle. And PepGen and Entrada are both using cell-penetrating peptides. We're also very hopeful about gene therapy and gene editing. I already talked about Exonics Therapeutics, but I'll also mention that CureDuchenne was one of many investors in Bamboo Therapeutics while they were developing their microdystrophin gene therapy program, which had later got acquired by Pfizer. And Pfizer is gearing up for phase 3 clinical trials now. So, we're very hopeful, not just for Pfizer's program, but also for Solid and Sarepta. But we recognize that any gene therapy and gene editing approach is going to be limited in terms of who can get those treatments because of the use of AAV virus to deliver that machinery. So we're also very keen on next generation gene editing or gene therapy delivery mechanisms to get around some of the immune limitations. And 4D Molecular Therapeutics takes the approach of generating novel caspids that don't already exist in nature, so individuals presumably would not have neutralizing antibodies to those. Chameleon Biosciences takes a different approach and envelops the AAV in a lipid cloak, so to speak, and has some inhibitory immune molecules. And Myosana Therapeutics and Code Bio get rid of the virus altogether. And so in addition to overcoming some of the immune limitations, what these technology platforms may be able to do is deliver something larger than mini or microdystrophin, with the goal to be to deliver a full-length dystrophin. And then lastly, under the theme of treating the whole disease, I'll just point to two recent investments. Edgewise Therapeutics has a small molecule which stabilizes the muscle in the absence of dystrophin and really prevents the contraction from causing as much tear and damage to the muscle. And Mesentech is not focusing on muscle at all, but rather trying to regenerate bone, which is a huge unmet need in Duchenne because individuals have bone thinning, not just because of chronic inflammation and lack of weight-bearing exercise, but also because many individuals with Duchenne are on chronic corticosteroids. So that was a quick walk through our ideas and our portfolio, and just welcome anyone who has interesting new approaches to feel free to reach out at any time.
Thank you, Liana. It's very nice. Great to see that Cure Duchenne Ventures has been so active lately. So hopefully some of these bear some fruit.
Thanks, Jonathan.
Okay, we're going to move on now to our next speaker, who is Craig Boyce from the ALS Investment Fund. As soon as you can take control, Craig, feel free to fire away.
All right. There we go. Okay. Thank you, Jonathan. Hello, everybody. I'm Craig Boyce, and I'm a managing partner at the ALS Investment Fund. On behalf of my two partners, Felix and Melanie, I'm pleased to provide you with a brief overview today of the ALS Investment Fund. I think you'll hear a few similar themes as Alana just spoke about, but the ALS Investment Fund is a for-profit, single disease venture capital fund. It's born out of 3 Dutch patients that had a vision that the combination of capital and science could defeat ALS. That by investing in translational science, the ALS Investment Fund can accelerate ALS therapy development to patients, and in so doing, achieve a 2-pronged goal. First, generating attractive returns, and second, making impact to the ALS community. Now, while this is a unique to ALS, a unique platform to ALS, it's not a unique model, as we just heard around Cure Duchenne's, as well as many other both for-profit and non-profit single disease focused funds that are shown in this slide, and several others that aren't included. If you were to sum up our goal and visualize it in a relatively simple way, we're hoping to replicate the success of the Cystic Fibrosis Foundation, where over the course of 15 years, they invested over $400 million towards a disease at the time that didn't have any known treatment. Now, after that investment, they can treat over 90% of the cases and return $3.3 billion back to the foundation. It's our goal to set up a similar platform that can have that success. We're 5 years into it, and so hopefully another 10 will be in a similar spot. Our mission is simple. It's to invest to create a world without ALS. The fund was founded in 2016 as a for-profit VC fund in the Netherlands. We have 3 partners and a deep network of advisors and scientific advisors and a broad network to help us execute on our investments. In the first fund, we've invested $31 million in a target a $50 million first fund. We've invested across 6 portfolio companies and are looking to invest in an additional 2 to 3 more. We have over 25 limited investor partners, including 2 nonprofits, the Dutch ALS Foundation and the ALS Association here in the US. We believe that we can be a compelling platform both for for-profit investors as well as nonprofits who are looking to replicate similar outcomes of other nonprofit funds, where they can invest in their mission, but also, unlike a grant, have money that returns back. And so for the ALS community, ALS nonprofits, it could be the next Ice Bucket Challenge. The fund is led by a strong team. Felix von Kopper founded the fund and has a strong entrepreneurial background in neuroscience, as well as European ties. Melanie Leitner, who many on the phone recognize and know, is our Chief Science Officer. She is a PhD neuroscientist and deep expertise in ALS. I have an engineering background, but then transitioned into an extensive investment career at Bain Capital. I was also directly impacted by ALS. About 25 years ago, my father battled the disease, and ever since then, I've been involved philanthropically. The ALS Investment Fund, for me, combines my career experience and a mission to cure ALS. Rhys Brower and Alan Watts are also our advisors and round out our investment committee. The fund is supported by a who's who in ALS research scientists and clinicians, as you can see from this slide, both in the US and in Europe. We believe now is the right time and an exciting time to invest in ALS. A combination of scientific advances, new genes discovered, new therapeutic targets identified, combined with the influx of capital, primarily driven through the Ice Bucket Challenge, has spawned a growth and innovation in the ALS company formation. We're tracking 140 private companies in our pipeline with an ALS therapy in development, ranging from early R&D through preclinical studies and as well as into the clinic. This activity, as you can see from the slide and the growth in the number of companies, gives us the confidence that the disease-modifying therapeutics are on the horizon. In fact, our first investment in Amylyx is knocking on the FDA's door. The ALS Investment Fund seeks to fill a funding gap facing early-stage ALS companies, bridging philanthropic supported research and innovation, helping companies get to a proof of concept, ideally phase 1, phase 2 clinical trials, where large pharma and larger VCs can step in to take it through phase 3 development and out into patients. We have a 3-pronged investment strategy to create a diversified portfolio across technology. We primarily focus on disease-modifying therapies, but also are investing in AI and development tools like Emulate, organ on a chip, which can accelerate drug development. The second area of investment thesis is around the ALS pathway. We see ALS having a potential multitude of diseases and a potential need for a cocktail therapy. The idea of this is to invest in the best 1 or 2 approaches across a range of 6 to 8 of the most promising ALS targets, ranging from mitochondrial protection to protein misfolding, TDP-43 irregularities, neuroinflammation, and gene therapy. And lastly, we apply classic portfolio construction to our portfolio, looking to diversify our companies across age and stage of company, as well as invest alongside other well-funded and value-added investors. The ALS Investment Fund is uniquely positioned to gain access to investments and add value to our investments after we've made them. This is driven by our deep expertise and differentiated expertise in ALS, which allows us to actively be involved with our portfolio companies, both prior to investment and post. Our activity is then supplemented by our network of science advisors who provide us thoughts in advance of our investment, as well as supporting our companies after. Our network of other investors, and our contact and network with the variety of nonprofits involved in ALS. The combination of all of these makes us a very complementary investor to investing consortiums. We're able to help validate the science through upfront diligence, and post-investment add value and support to the regulatory and development pathways. We've had an active 5 years, highlighted by our 6 investments, the first in Amylyx, then in Orphazyme, Verge Genomics, Emulate, Apic Bio, and most recently Prolentia. We're excited by each of these companies' progress and development. We're also excited by Amylyx's positive phase 2 trial, and hopefully that will be a therapy that will be available in the not too distant future. There's a multitude of ongoing partnership and licensing discussions within the portfolio, which gets us excited, as well as a deep pipeline of opportunities that we see for future investments by the fund. With that, I'll pause for any questions. I would just highlight that if anybody is interested in partnering, investing, or has an interesting company, ideally in the phase 1, phase 2 clinical trials, we'd welcome the opportunity to connect. Thank you.
Thank you, Craig. Very nice presentation. It's great to see how many companies are really active in this space right now. It's absolutely booming. So,
sure.
Wonderful to see. I think in the interest of time, we'll have to move on to our final talk of the morning session. So this will be from Dr. Sharon Hesterly, who will tell us about the MDA Venture Philanthropy Program. Sharon?
Sure, great. Thanks, Jonathan. So you already heard a lot about MDA and our programs and what we cover, so I won't have to include that in my review of that description. But if you missed my early intro to MDA, we're an umbrella organization that funds research in neuromuscular disease over, we say 45, but really when you start parsing them genetically, there are hundreds of neuromuscular diseases. So I think unlike the 2 funds you just heard about, we're not a single disease focused funder, but we are restricted to the neuromuscular disease space. So MDA Venture Philanthropy seeks to invest in early stage research, primarily usually before a Series A funding. Our first investment was in 2004 in Asclepius Biopharmaceuticals, which was recently acquired by Bayer. And since we started in our sort of translational venture philanthropy investment strategies, we've invested a total of about $30 million to date. So we currently have 1 asset in phase 2 and 1 in phase 3, and we've got 7 preclinical assets active. So, you know, actually over those time, there have been a lot of lessons learned, I can say, and I'll mention a few of those as we go forward. Our average deal size is about $500,000, but the range is from 2.5 million to 150,000. We're currently accepting inquiries for 2021. So all the presenters in this forum are eligible to apply. You can feel free to contact us. And I will talk a little bit about what that process looks like. I will say that when we started back in 2004, we were just calling this translational research. And that was our first investment that we did in a company. At the time, we were noticing that in our grant funding program, we were seeing a lot of the same projects come in over and over, just slightly tweaked. And it was clear that although these projects had a very translational focus, that the investigator just wasn't sure how to move them forward. And so at that point, we decided there are sort of 2 ways to go. We could start funding people in the academic space who are trying to do drug development, or we could fund companies to do that development. And we decided at the time that it made sense to just start directly funding companies. So AskBio, we were angel investors in that company way back in the day, and then actually had the opportunity to work for AskBio for a while before I recently returned to MDA. So I've seen that company from a lot of different angles. So just a little bit about what our current diligence and deal negotiation pathway looks like. I think this has evolved quite a bit over time. So we typically ask for a pitch deck, and we review that pitch deck with our advisors, and then based on a number of potentially compelling attributes, we'll decide to invite the company to submit a full diligence package. So for us, that diligence covers a number of different areas that I think would sound familiar. So we look carefully at the product development plans, at the commercial plans, at the management review. Who are the key members? What is their experience? Who are the board members? Who are the investors? Again, since we're usually investing in pretty early stage companies, we want to make sure that they have access to the right people and the right experience to help them progress. And then we also review the corporate plan. If we're going to make an equity investment, we really need to understand the company at large, of course, including what their exit strategy is, the all-important operating cash horizon. So we take all of these things into account and then make a final recommendation. Usually we invite the company to do a final presentation for our MVP advisory committee, answer final questions, and then we recommend the project to the board for approval. So this is just a glimpse. So our advisory committee, we have a range of people who help us with these investments, and they include people with background in venture investing or who are current investors or previous investors. We also have eminent scientists in the field, Dr. Lou Kunkel, Dr. Beth McNally, and Dr. Jeff Rothstein. Plenty of people who have who also have extensive industry experience, like Dr. Bruce Wentworth. So, and some with just business experience. So we get a variety of input into these projects and look at them from a lot of different ways, and we find that to be a very useful combination. So typically in our investment terms, what we're looking for, we can do this in a number of ways: equity, convertible notes, or royalty sharing agreements. In the beginning, we largely did royalty sharing arrangements. And that was because at the time, nonprofits were being told that we should not do equity investments. And the reason was that an equity investment has to be booked as an asset. So it's not a program expense. And when you're a nonprofit and you're looking at your 990s, your program expenses count in your favor. But an asset is something that if it doesn't pan out, you have to write off. And it can actually really hurt your financials because people carefully scrutinize how much you're spending on programs as opposed to acquiring an asset. You also have to value those assets every year, which we're not always equipped to do. Over time, it seems like this has become less important, and certainly if it is not the majority of your funding, it won't have as large an impact on the 990s. So we have started to take more equity investments. And I will say that, one of the challenges, there's of course pros and cons to doing it both ways. With a royalty sharing arrangement, we're really investing in that case in a product. And if that product does not make it to market, then we lose that investment. So the example there is our early investment in AskBio, which was acquired for $4 billion. It's still a little painful. We did a royalty sharing arrangement around an early gene therapy vector for Duchenne, and that capsid ultimately didn't go forward. So our product did not go forward. Although, a separate product with a different capsid did go into Bamboo Therapeutics, which was eventually acquired by Pfizer. And then AskBio itself has recently, as I mentioned, been acquired. So there are pros and cons to this. Of course, when you invest in the whole company, you sort of rise and fall with that investment. The other challenge there is a lot of companies have pipelines that include things that are neuromuscular and things that aren't. So if a company ultimately ends up with a lot of things in its pipeline that aren't neuromuscular in focus, that can be a challenge for us because then it's hard to justify it being a mission-focused investment. And we won't have time, but I'd actually be interested in hearing, say from Liana, how you manage that with your board. So I won't go into detail. I think we're running out of time, but just to mention, we are showcasing some of our investments tomorrow. So if you look on the agenda tomorrow, we will hear from Avanti Bio, who has a gene therapy approach to Friedreich's ataxia, Myogene Bio, who are working in a gene editing approach for Duchenne, and our most recent investment in Incefa, who's a small startup company who has a novel CD38 approach to ALS. And you will hear from all three of those investments tomorrow. We have I think what's going to be a very interesting panel planned for this afternoon. And thank you very much to Cello Health for helping us to put this together. And I'm going to turn it over now to Michael Rice, who's the VP and Head of Advanced Therapeutics at Cello Health Bioconsulting, and who will host this session.
Great, thanks, Sharon. So I'm Mike Rice. I lead a practice area at Cello Health Bioconsulting. We're a knowledge-based consultancy that's really grounded in science and focused on helping advance early-stage platform companies in particular. We work directly with the entrepreneurs. We also work with potential pharma partners that are looking for disruptive technologies to invest in. And so it's really a pleasure to be here working with, presenting on this panel with the Muscular Dystrophy Association and some of the leading investors and accelerator programs that are helping advance therapies for neuromuscular diseases. It has become one of our biggest areas as well over the last several years, and it's been kind of striking because 10 years ago there was relatively little advancement in the space. We looked at the blood-brain barriers being an issue. We looked at being able to deliver high titer AAV to treat a large mass of skeletal muscles being a barrier, and there was significantly slow progress. So it's great to be here, and I think it shows the interest in the space. The fact that Sharon and I were just talking about, well, how can we help you? Can we put a panel together for your early stage investor conference? And lo and behold, we were able to get some of the biggest investors in the space in short notice. So thank you to the panelists. And I'll give an introduction to them in a minute. But first, I wanted to spend a little bit of time just heighten your awareness of the period of time that we're in as far as advancing therapies for neuromuscular diseases. And I'm not I want to get to the key issues because we have a very ambitious list of discussion topics. So I'll probably go through this quickly, but you're welcome to have a copy of it after the conference is over. So because we're always looking at across genetically defined patient populations for helping, advanced technology platforms, we've developed an internal database that tracks close to 10,000 genetically defined phenotypes. About 5,500 of them are single gene defects, and the others are polygenic and acquired disorders. And when we look at the ones that we track most closely, which are the ones that are greater than 1 in a million prevalence, about a quarter of them are neuromuscular diseases or related neurological disorders. And so there's a large representation of diseases in this area. They are very heterogeneous, going across a bunch of different sort of categories of pathophysiology that we look at. When we look at the therapeutic approaches that we track, they cross all modalities from small molecule, splice modifiers and nonsense mutation therapeutics, exon, a lot of oligonucleotide therapeutics, exon skipping, splice modifiers as well, and then a lot of work in the gene therapy space, mostly led by AAV therapeutics right now. And so we track, development in the space. We match across the categories to figure out, where are the neuromuscular diseases where there's a lot of activity, where's the white space to go, and what are the pros and cons? Why isn't anybody investing in these other areas? Is it because the biology is so complicated or not? But in this gene therapy space, including nucleic acid therapeutics, About a quarter of the pipeline, is related to neuromuscular disorders. We have several marketed products now, and we have a pretty robust phase 2, potentially registrational programs that can move very quickly. And so when we look across the indications that are being pursued, what we find is that, there's a handful of, given, even though we track 225, indication, there's really only a handful where there is robust development activity. There's a lot of one-off research programs, mostly mostly all pre-POC, mostly early preclinical or discovery yet. But so we'll see those sort of advance in the future. But, really leading the space, we have Duchenne's, Parkinson's, ALS, some CNS, broader CNS disorders, SMA, limb-girdle. But, it's a relatively short list where we have a lot of programs going. So there's a lot of white space opportunity to go after. When we look at the market, it was great to see the philanthropic venture groups present of their early investments and how they're paying off and everything. But it's really, this early translational investment that now is stimulated in this immense growth potentially in the market. So, on top of, the SMA and Duchenne products that are, approved and advancing towards the market, we see stimulation into the broader neuromuscular space. And we also see a lot of interest and stimulation of big pharma neurology franchises that have struggled through the years to advance many of their programs, but now they see these biologically tractable patient populations as another way to start advancing and stimulating their R&D programs. So, yeah, great. So just, as an eye opener, we look at the M&A that has really kind of driven, so it was a couple seminal things. We had the pleasure of working with the Avexis team to try to build a go-to-market strategy and a way to bring SMA products to market, but also to bring and advance additional neuromuscular diseases prior to the acquisition. But it really was a seminal event that really kind of drove a lot of, increased investment into the space. We've seen many other pharma partnerships and M&As through the years, but it's really just been in the last couple of years where there's been a tremendous spike. The 2021, I don't know how far we are last this was updated, but we expect a lot to come yet the year, even though it looks like it's low right now, but it really hasn't been materialized yet. So then beyond that, so because we're seeing a lot of high value exits, there's a lot of new companies being created. So we've also had a record year in Series A follow-on investments, some of them going very quickly last year through the COVID period from, initial startup, to go into crossover rounds and then IPOing. So, it's been a phenomenal and maybe outlying year. So we'll see what happens going forward, but we do foresee this momentum continuing, but the challenge is to see what what happens going out on the out years. So I think that's all I wanted to present today, and you're welcome to have it, but I would like to introduce the panelists and get into the discussion. So again, we have a great group of panelists. I'd like to invite them to each give a little bit of self-introduction, tell what they're doing, talk about their investment thesis, and whatnot. So why don't we start with Beth Seidenberg? Why don't you introduce yourself and hear about what you're doing in the neuromuscular disease space?
Great, thank you, Mike. Appreciate the introduction, and good afternoon, everyone. Delighted to be here. So I'm Beth Seidenberg, physician by training, spent about 20 years in the industry before becoming an investor. I've been investing in life science companies for the past 16 years. And 2.5 years ago, started my career at Kleiner Perkins and learned how to incubate companies and do really Series A startup and building from the ground up. About 2.5 years ago, I joined forces with my co-managing director and partner, Sean Harper, who used to run R&D at Amgen, and we started Westlake Village Biopartners. We're focused on investing in mostly seed and Series A companies, looking for breakthrough technologies. Gene therapy is clearly a space of great interest. We have over $820 million under management now across 3 funds, and we're very excited about this particular area, and I look forward to the discussion today.
Great, thanks, Beth. Chris, why don't you go next?
Sure, yeah, Chris Karabedian. My 30th year in industry. The first 25 years was in operating companies. Abbott, spent 8 years at Gilead, 4 years at Celgene, 5 years at Sarepta as a public company CEO. Left Sarepta in 2015, started an accelerator company incubator called Zontogeny, based in Boston, making seed investments, really partnering with scientific founders, first-time CEOs, entrepreneurs. My investor in that accelerator was Perceptive Advisors. So they asked me to join them to launch their first pure play venture fund. So I also manage the Perceptive Zontogeny Venture Fund. We are about to close out Fund 1, close to announcing Fund 2. And we've made 9 investments so far, including 2 in the gene therapy space. In fact, this morning we announced a Series B with Forge Biologics, led by RA Capital, that we led the Series A and participated in the Series B. So yeah, really excited to be here. And obviously from the Duchenne experience I gained at Sarepta, we see a lot in the neuromuscular space and the rare disease space as a result. And yeah, we're really excited to invest in some of the emerging technologies.
Great, thanks, Chris. Inbal, do you want to introduce yourself next?
Sure. So first of all, thank you for inviting me. It's a pleasure to be here and take part of this discussion. So I'm Inbal. I'm a developmental biologist by training. Did my PhD years ago at the Weizmann Institute, which is actually located just on our backyard. In FutureX, I'm leading the scouting and evaluation of novel disruptive technologies. Before that, I spent a few years in Madison Ventures and Israeli biotech BioLineRx. So maybe I'll give you some background on FutureX and our investment thesis. So FutureX is the leading biotech incubator in Israel, almost the only one that is solely focused on therapeutics. We were founded 7 years ago now as a partnership between Takeda, J&J, OrbiMed, and recently Takeda, and also recently aided by Empire. And we have a very unique model where we do venture formation. Namely, we create companies based in our facilities here in Israel, where each one of our portfolio company receive a $3 million R&D budget over 3 years of incubation. And we give a very holistic support, envelope support to each one of the companies so that they can solely focus on the R&D side. So we have here highly equipped lab space and offices and finance and HR, legal, VD, of course. And of course, we have also highly experienced management team that guide each one of our companies, you know, from designing the right killer experiment, the development plan, choosing the right indications, and more. So in a way, we structure more like a biotech company. And above all this, we get the guidance and support from our partners at J&J, Takeda, and Bayer. And we're really using both their R&D and disease expertise to really generate companies that will create meaningful value for the pharma industry and, you know, at the end of the road for the patient. We just mentioned that our partners do not have any preference or any right of first refusal. And when time comes to raise money, we talk with all VC, all pharma partners to enable our portfolio company to be independent after the 3 years of incubation. Sam.
Great, thank you. Leah, why don't you tell us about Longwood?
Yeah, thanks, Mike, for inviting me today. I'm thrilled to be here. My name is Leah. I'm an entrepreneur in residence at Longwood Fund, where I've spent the past 4 years building startups. So taking academic work directly kind of from institutions and figuring out what's the best way to translate some of these really awesome high science ideas into something that can, hopefully help a patient sooner rather than later. And that's in general what Longwood does. So, we invest in early stage companies, but spend probably most of our time building companies from scratch. We serve as early operators in the companies, and then hire a full-time management team to take over from us after the fact. So, at Longwood, we look at, like I said, large platform companies, ideally with near-term product opportunities. Typically, our thesis has been that we need to see some kind of major value inflection point between 3 to 5 years after we make that initial investment. And what a value inflection point is changing over the years, I would say. So we've started to invest even earlier over the last year and a half, especially, I think. And yep, we look at a lot of rare disease, a lot of neuromuscular conditions. I actually trained in neurodegenerative disorders at MIT in the Broad, so that's kind of where I got my scientific background. And yeah, I'm currently kind of half between Longwood and operating at one of our gene and cell therapy companies as we speak.
Great, thanks. Yeah, and it is amazing to see, the changing benchmarks for POC and what is, requirements for IPO, these days. So Sharon, why don't you, I know we heard, everybody probably knows you here and have just recently, heard your description of MDA, but why don't you, briefly, introduce yourselves and then kick us off on a discussion about, I'd just like to hear a little bit about your outlook. What do you think about what's, coming up in the next few years? Do you see this robust investment environment? Is this going to continue, or what are your thoughts about the next few years?
Sure. So, so I'm MDA's Chief Research Officer. My background's in neuroscience, and I've been in the space for about 20 years. And I started my career at MDA. I started MDA Venture Philanthropy in our translational research program while I was there, and left in 2010 and worked for a number of different nonprofit groups, including Parent Project Muscular Dystrophy and the Myotonic Dystrophy Foundation. And then went to industry, as one does, and there was with Bamboo Therapeutics and Pfizer, was CEO of a special purpose entity called Lion Therapeutics that was focused on gene therapy for limb-girdle muscular dystrophy. And then that got rolled back into AskBio, where I was the Director of Portfolio Development. And recently returned to MDA. So that's the full circle story. If you were here earlier, you heard my description of MDA Venture Philanthropy, but we've invested, if you're just joining us, we've invested about 30 million over the years in various areas in the neuromuscular disease space. And like I used to do these, so in the answer to your question, Mike, this topic, I used to do these graphs for our board where I would graph out the number of companies in the space, and it fit easily on one page. In bar graphs where you could actually read the names, unlike your bar charts that were microscopic. So it's clear to me, the field has just blown up extraordinarily in the last, 10 years, easily. And I guess, my sense is that it's pretty solid, that I think the interest in rare disease wasn't a passing fad. It seems to have stuck with us. And in the neuromuscular space, it feels like a lot of these areas and technologies have matured to the point where this is pretty stable and that growth will likely continue. I mean, what I see is companies branching out from Duchenne, where there's a clear path to development. So I think that's, triggered a lot of interest, a clear regulatory path. But people are now getting interested, I think, in limb-girdle muscular dystrophy for sure. And also some things like FSH dystrophy and myotonic dystrophy, which are major unmet medical needs with very large populations for rare diseases. Like myotonic dystrophy might be as many as 80,000, 90,000 based on a sort of population-based study that was done by Nick Johnson. So I think there's a lot of opportunity there. These are markets, and another one to consider is Charcot-Marie-Tooth disease, which is a peripheral neuropathy, but it happens to be the most common disease in our program. So the majority, the largest proportion of people registered with MDA have CMT. So I guess when you talk about the white space, there's some pretty big white space out there in large populations. So
yeah, and it was interesting when I was working in siRNA. It seemed like things were getting bumpy, and then all of a sudden GalNAc targeting, it became validated, and understanding of liver targets to go after, really stimulated that side of the field. In the neuromuscular space, what is it that has been so attractive in the last couple of years? Like what has driven, the investor interest in that? And field that initially, I guess, to Beth and Chris. You want to kick that off?
Sure, I'll go first. I think it's a few things. It's multi-pronged that has given people a lot more excitement of investing in the neuromuscular rare disease space. So first, we're seeing more and more technologies, genetic technologies that are maturing and even advancing, right, in terms of getting better at cell penetration, right, targeting. And I think that where those technologies typically go are monogenic diseases, and where we understand the target well enough, right, to get a good proof of concept early of whether it's protein expression or knockdown or what have you. And then the other piece of it is that we've seen a lot of clinical and regulatory precedents in understanding endpoints, capturing more natural history information. The regulators have seemingly, the first real guidance document they provided for rare disease in collaboration with the industry was around Duchenne muscular dystrophy and dystrophin as a biomarker endpoint. And we've seen a lot of clinical studies, right, that really help us understand the disease areas. So I think the regulatory clinical precedence is another factor where people have a roadmap for what is it going to take, right, to get a drug approved. And I think not just Duchenne, SMA, ALS has been, very active in the clinical setting, and a lot of feedback. And by the way, even when there's bad news, it's good for the industry, because then they have clear guidance from FDA, what they're looking for, what's not enough, what statistical methodologies are acceptable, what, biomarker endpoints are acceptable. And so I think all of this has fueled and supported the slide you showed with why there's so much activity.
Mm-hmm. Yeah, Beth, I don't know if you want to comment on that as well.
Yeah, thanks, Mike, and thanks, Chris. I completely agree with Chris's comments. And so in order to not be redundant, let me focus a little bit on tools and technology. And I think that, we often talk at Westlake about the golden age of biotechnology, and it's truly here, right? That explosion of growth and interest is really based on the explosion of new discoveries in biology, new tools, new access to genetic information. If you just think about how, a handful of years ago, how much it cost to sequence the human genome, it was in the millions of dollars, and now it's a couple $100. And so we can do a lot more genetic screening, which then opens up populations and knowledge of patients. You marry that with the tools that are available, whether it's things like gene editing or knowledge of being able to not only replace genes, but knock down, provide different expression of genes in different tissues, or in some cases, lower the expression of a resultant gene that is causing clinical genotypic and phenotypic changes. And then, of course, the work around you mentioned, Mike, in your comments, how to access the blood-brain barrier or how to overcome, what is designed evolutionary to keep us out of trouble with respect to virus and parasites and such. There is very good engineering of now delivery tools, which include AAV capsids, and that is allowing us to not only address and cross the blood-brain barrier, but also target specific cell types within the brain. You can do that beyond the brain as well in, muscle, heart, kidney, start to name the organ that you want to target. We now have tools and technology that really opens up the opportunity to address those organs in a much broader way than we could, honestly, 3 to 5 years ago. We didn't have these tools.
Yeah, and so I hear, less biological risk because they're genetically defined, patient populations, and the researchers, on this call today, have advanced the understanding of many of those diseases. I heard better delivery to neuronal and muscle cell to the full muscle unit. And, even though, we still hear, setbacks in both, toxicity as well as, still some failed programs, such as the Huntington's space that there's been some significant news about, in the last few weeks. So what risks exist, going forward? Maybe we could talk a little bit about, crowding of, I know Duchenne's is still a great unmet need, but there are many, many programs, and from an investment standpoint, how do you pick winners and losers, and how do you think about being able to allocate funds to some of those white space opportunities? And we do a lot of that. We do a lot of helping identify, unique opportunities, beyond where everybody's developing, but there's, there are areas where we're going on, Chris described that there's regulatory guidance in those areas, there's a lot of reasons to go there. So I don't know if Leah or anybody wants to kick off maybe some thoughts around that.
Yeah, I can comment a little bit on that. So the way that we view it, right, is when we're looking at spaces that are starting to get increasingly crowded, there's kind of a few ways you can approach it. One is if we see a company that has a broad enough platform and you can fund to that, not, to pass that first program getting to a value inflection point, but to that second program getting far enough along that you have some meaty data, I think that helps us mitigate. The other nice thing, like a comment on the previous question too, is when we look at some of these muscular dystrophies, I mean, the things that you need to do there, the challenges that you need to overcome are, once you do that in some of these test cases, they're applicable to things way beyond muscular dystrophy. So the question of delivery and tropism and cell penetration, all of these are things that other fields struggle with. So to us, we don't mind entering a space that's crowded if we think there's a really well-defined path to get to that proof of concept, and then that unlocks additional value that we can then point at some of these other diseases that have been intractable for other reasons. Usually it's that, the regulatory path is either long or unclear or both. And so that's a little bit of in terms of how we think about mitigating risk in these, gene therapy in general. Some of these spaces are so crowded and it's driven by a lot of the M&A. And so we still want to be a part of it, but we have to see that, that secondary piece of some other platform or some unlocking feature when we make that investment.
Yeah, certainly. Chris, do you want to comment on?
Yeah, I think this is one of those things where the default for a lot of early stage biotechs is to follow those who have led in a certain space. And you saw that with Duchenne. There were, you could count on one hand the number of programs 10 years ago, and now there's dozens, right? And I think that in some ways you get too crowded in a certain area. You have too many followers. You have real challenges with trying to break through, show incremental benefit, et cetera. And there's a lot more risk when you're competing against, 3 dozen programs to show a benefit. And then you've got issues with pricing and reimbursement when there might be 5 or 6 drugs approved in a given disease. We're not there yet, but I think that the real innovative companies and investors will go where there's not a lot of precedent, where, and you know, Sharon mentioned this with, you know, increasingly we're seeing those kind of, you know, look into FSHD or myotonic dystrophy, or, you know, limb-girdle has gotten a lot of activity. There's a whole host of other neuromuscular diseases that nobody's really going after. And I think part of that is the concern that it might be too difficult. We don't know the natural history. We don't know the endpoints. Nobody's really shown the biomarker data that's compelling. And I think that the rewards for those who blaze the trail, if you will, in some of these new areas would be quite significant if people had more of the will to take some risk and to be innovative. And by the way, the reason I am most encouraged about that today is the FDA has really shown you know, a willingness to meet companies halfway. Not only, you know, have they given great, you know, policy presentations, but it seems that some of their actions in recent years has really shown that, you know, they are flexible, they are supportive of accelerated approval, they are, you know, not requiring 2 well-controlled, placebo-controlled trials to get approval, especially in rare disease. So I think, you know, companies that are willing to negotiate and work through those FDA issues, I think we'll find the rewards of that on the other end.
Great.
Mike, if it's helpful to add just a quick comment, because I'm tracking very much with what Leah and Chris are saying. One of the key areas that's helped advance a number of these diseases is working with the disease foundations. So some of the work that Sharon is doing and the foundations have the opportunity to do a number of things. One is it's way beyond funding. It's important to identify and create a network of where the patients and where the families exist, and then have, through either company sponsor or academic sponsor, do natural history studies. So to Chris's point around endpoints and how can you get traction in a disease that has never had a therapeutic intervention, you need to understand the natural history of the disease, validate endpoints, so that then when a therapeutic is available, you're ready to go. Patients are there, the endpoints, the knowledge of how the disease progresses. So I'd encourage everybody who's interested in this area to, you know, lock arms and do that work that will be highly complementary to the therapeutic work.
Great, thanks. And Sharon, I think that's a segue to you.
Yeah, no, thank you. I think a lot of what we've tried to do are these de-risking strategies, and some of it is funding early stage companies, but you're absolutely right. A whole host of things come with that. So, you know, we have a whole category of grants that we fund called infrastructure, which are what we call tools, techniques, and services. And that includes things like antibody repositories, cell line repositories. We also fund clinical trial networks, and we'll fund natural history studies. So these aren't things that necessarily have a high return on investment financially for the organization, but it's a high return for building the field and building the tools that will attract people into these spaces. And I think a lot of the nonprofit groups in this space really deserve a lot of credit for doing some of that hard background work that's sort of unseen, but can really help bolster various areas in the field. So thank you for mentioning that. We try.
Thank you.
Yeah. So what are the areas that you think that, so if you're developing proof of concept in a crowded area, and maybe pharma has interest in broad applications for neurodegeneration, what do you think the areas that are going to be fruitful in the near, not so distant future from studying drug mechanisms or pathophysiology in neuromuscular diseases like neurodegeneration, inflammation, things like protein aggregation, RNAopathies. You mentioned Duchenne, myotonic dystrophy. I know that that's a very hot area where we have many very early stage programs that are all going after DMPK and other things. what are the areas that you think are going to drive innovation, beyond those initial disease populations?
I'd love to hear from Inbal. Yeah, just from your vantage point, looking at early research. Yeah.
So I can address this question maybe more broadly, not just for neuromuscular disease. So in a way, FutureX is more pathway-centric and looking for platform technology for multiple indications. However, if we're thinking about the first indication, the proof of concept indication, we typically search for any human genetic links and start with a smaller indication, like rare disease indication, where the scientific rationale is more straightforward. And of course, the clinical path is more feasible. So this is one area. With regard to some wide space area, I think there are multiple reasons for that, including commercial and patient access, etc. But in my view, one of the reasons for that is that usually when thinking about drug development, it's usually driven by a disease or by target of interest, and less, using the unbiased approach. You mentioned, exon skipping for DMD. So in a way, if you think about it, there are limited known target indication for exon skipping, and a more unbiased top-down approach, I think, is needed in order to identify novel potential target or indication for exon skipping and for other, approaches. So this is just in a nutshell.
Great.
So when I think about that question, Mike, and it gets back at what Beth, you were saying earlier, is there's always this push and pull when we look at different fields between where is it that we need to advance on the tool side versus the underlying biology side. And so, in certain fields, it's still the case that targets are the bottleneck. And in other fields, it's actually, there are some interesting good targets to work on, but we just haven't quite figured out how to crack those nuts. And so I think we're getting closer to that end of things. I think when I look at a lot of the different muscular dystrophies, I think there are some good targets that just have not been prosecuted in the way that we'd like them to across all these diseases. But then you get things like, Huntington's. It seemed pretty straightforward to lower mutant Huntington, and yet, you see what happens there. So there's still risk, but I do think, when we try and look at different spaces, we try and, either determine whether that target bottleneck is really a thing that's limiting the innovation. And so that's where we need to, take advantage of lowering costs of DNA synthesis and sequencing and do unbiased screens and all of that. Versus actually just focus on, what tends to be the less sexy science, but how do we get, some of the science that's already been somewhat well validated to be tested in a patient.
Yeah. Chris, did you want to touch on that?
No, just to say that it's a very encouraging time right now. We have multiple modalities that can go after a given target. And I think the level of healthy competition to figure out which of these technologies, right, can address given diseases. We've seen an era over the last 5 to 10 years of just the platform technology that once you have the right tool, you can really, alter that to target a myriad of diseases. And one of the questioners in the Q&A box talked about, looking at these not as just a group of diseases, but they're all individual kind of subsets of a disease category. And I think we're seeing more and more of the technologies that Beth pointed out early on about the excitement of these, new tools is really where I think I love to see that we've got, different genetic technologies, DNA, gene therapy, different RNA modalities going after rare disease. We've got, newer antibody platforms. We've got engineered cell therapy. You've got protein degradation technologies that I think that we are in an era where you see a lot of healthy competition across these different technologies, and, good for the disease area because, a lot of people are going after this and we're not sure which ones are going to emerge as the real leaders here.
Great. Any more comments on that? Otherwise, we can move on to hearing a little bit of advice for the entrepreneurs that are tuning in today. All right. So maybe Chris and Inbal, maybe you could talk a little bit more about your incubator programs and how you actually work with the entrepreneurs to advance their technologies.
Inbal, you want to go first?
Yeah, sure. So, thinking about what is our role as an incubator, it's in a way, the goal of our partners, in establishing FutureRx. So first of all is, to create platform that can go out and look for early stage technology that are probably too early for classic VC and cooperation to directly invest in, but highly innovative and have the potential to become, those technology that would be likely be invested by those pharma companies. So this is the first goal. Additional goal is to, I think, to overcome the inefficiency that's usually associated with early stage drug discovery. And the way that we do it is by providing a very holistic support to our portfolio companies, as I mentioned earlier. Maybe the last role in my view is to, to secure follow-up investment or out-licensing opportunity to enable our portfolio company to be independent after the 3 years of incubation and not, limited only for these 3 years. Chris, the floor is yours.
Yeah, no, and actually Beth is doing a lot of seed investments with early stage technology as well, so she should comment. But I think what we're trying to do, of all of the sourcing we do, I'd say about half are not ready for a Series A. It's not de-risked enough. They haven't taken any capital in, and they need some real support and hand-holding, if you will, active management to make sure when they get from $500,000 to a few million of seed capital, they're using it wisely and they're trying to de-risk it, and they're trying to turn that concept into an investable company where you can pull down a Series A of 20 million or 30 million or 40 million. And so Zontogeny, we really partner with that early researcher and really think about how to efficiently deploy that capital. We bring a lot of industry experience to bear of what are people going to want to see, what are they going to be looking for, picking the right translational models, having the right positive controls. How do we really get that preclinical proof of concept that is going to kind of win the day? And then the good news is we have, I think like Beth's strategy as well, we have a Series A fund where we can pull down that capital as it's needed to continue to advance the company.
Great.
Yeah, thanks, Chris and Mike and Inbal. So I would highlight, 3 important things that are really necessary in order to take a great idea and make it into a therapeutic, a medicine that hits the market. The first is really important to industrialize the technology. It may seem trivial, and you say, well, I've studied it in 3 monkeys or a handful of mice, and that often is a wonderful starting place, but not insufficient to really allow a technology to scale and be able to be produced at a level that's necessary for preclinical safety studies, clinical trials, and ultimately the market. The second is you need people to complement the academic founders who have real industry experience. And that can be a range of people from the technical area of expertise in a particular disease to things like understanding, what's needed from a process development perspective and scale up, what's needed from a regulatory perspective, clinical, et cetera. So partner yourselves, make sure that whomever you're working with is bringing to the table real industry expertise on the people side. And then the third is strategy. It's often the case that we talk to people who have a great idea, but they don't have a real strategy of how to take that idea and actually build it either into a company or a product. And it could be things like, Mike, we've been talking about, which is understanding the competitive landscape and understanding how you're going to address that. Or it could be picking the right disease. You may have a great technology, but you picked a disease that's going to be either too hard to study, or Chris was talking about, you don't have an endpoint that is tractable, or the disease progresses over decades and it'll take decades to get a drug to market. So I'd marry those three things: industrialization, people who have industry experience, and a real robust strategy.
Great, thank you. That's very well said. And outside of the genetic disease space, I do a lot of work in the adoptive cellular therapy space, and given the competition in that space and the limited experience, human resources and talent to get the job done, it's amazing how I see so much turnover, and it's hard to contain continuity on a development program. And any advice in how do you put that dream team together and retain them?
So for us, one of the things we pay attention to really early on is getting some thought leaders around the table from square one. So whoever the KOLs are in the space, both clinically, but also, even on the basic biology side, when you have those folks and you're trying to recruit in an area, then you can, you're much more easily able to attract talent if, someone great from the field has already kind of given you their stamp of approval. And that's another case where all these different advocacy organizations can help to the extent that they have these lists, and it's really well known who the leaders are in particular fields. So I think that's just, one that's not often talked about, but that we found to be quite helpful in terms of kind of creating and attracting the team.
Great, thanks. All right, I don't know, anybody have anything else to add to that topic?
I mean, on the talent thing, I think we're in a good place because there's been so much activity in the rare disease space, neuromuscular. You're getting more experienced executives who've been able to navigate that clinical regulatory pathway. And I think that's a good thing. And there's more folks to choose. Gene therapy is still an area where, we don't have as much talent that we need for the demand. But I think when it comes to understanding clinical pathways, endpoints, right, regulatory, navigating those, I think we're in a good spot. But yeah, the earlier you can engage, I would agree with Leah, the better you are as a company.
Yeah.
And Mike, the only other comment I would make, and I've learned this over now, almost 2 decades of investing, find somebody who is a missionary. You want missionaries versus mercenaries. That's how you retain people. It's oftentimes when, we're starting companies or we're building teams that there's a personal story, a personal connection, and people have to be on a mission together to solve hard problems. If they're in it for a quick flip or, money on the mercenary side, don't back them. They won't be with you for the long run, and they're in it for the wrong reason, in my opinion.
Great. So changing topics. So Sharon, this is a great, format, to get to meet and know, these emerging, innovators in the space. And elsewhere, sometimes it's hard to, identify up and coming new technologies that is ready for investment. I've been a member of ASGCT, I think, since the first year in like 1996 or so. And you go around and you meet, inventors and entrepreneurs and everything in space, but there's not really a format for, doing partnering and pitches as much, So how do you investors, how do you identify, these early stage, companies that could be, future breakthrough therapies?
Well, I can say for MDA, we have a great advantage because we have this large research funding program as well. So we see these things from a really early stage. you know, when it's just an idea and we can watch them grow into fruition. That was part of the reason for this conference, that we reached out to all of the tech transfer offices in our database and kind of asked, what do you have that might be sitting on the shelf that could be interesting IP? So we can see back to pretty early stages what's coming out down the pike. So that's sort of our primary way of identifying new projects, in addition to attending meetings and keeping an ear to the ground, but that certainly helps.
Great.
Yeah, Mike, we used to call it, or I still call it occasionally, lab crawling. So, you know, be present, make sure, you know, you're connected with the academic institutions that are on the leading edge. Spend time, sometimes, as I think Leah and Chris said, may not be ready for prime time, but we can also provide advice and input to the academic founder and discoverers for what are the right next experiments that may make it ready. And often that's just our willingness to spend time with investigators. And of course, you know, we have to spend a lot of time reading the literature, looking at publications, whether it's ASGCT or other meetings, and try and stay on top of the field.
Great. Chris, how proactive are you in you know, searching versus, you know, or do things come to you?
Yeah, for the first couple of years that I started this, it was all unsolicited. So just, you know, a broad network, and people were aware of what we were doing. I hired a couple of years ago someone to just really focus exclusively on reaching out. And that's not just in the US, but around the world, to, you know, tech transfer groups and disease foundations and, you know, seed funds and other incubators, accelerators, to create a pipeline of opportunities. I'll be honest, I wish we had more bandwidth to do what Beth suggested, which is really just dig into the literature and keep on top of the research. That's where, you know, being early and being quick in engaging some researchers sometimes gets you some really good opportunities before everybody else realizes there's the potential there. So we'd like to get better at that. But look, I can tell you, just for the industry writ large, I don't think anybody's really struggling with seeing opportunities. You know, for the most part, those of us who have been in the industry for a while, people know how to find us. You know, they know where to go. And we're doing things like this. So I appreciate, you know, MDA. I'm sure all of us have been sitting on panels, right, this past year during COVID on Zoom, and it's really been allowing us to expand our reach instead of getting on a plane and, you know, flying into a, you know, city to go into a conference room and hopefully get the right people in the seats. So this, you know, virtual panel has really helped.
Yeah. Inbal, can you comment? You know, what is your visibility into, you know, U.S. inventions or European inventions?
Yeah, sure. So we are, of course, in a close relation with, you know, all PTOs around the world, hospital, research centers, etc. But if we're looking on, you know, actively pursuing some indication of interest, so in the last year, we launched 2 dedicated call for proposal together with J&J, which is one of our partners. So this is one option. Other option that we use is, for instance, we are using companies like Cello to identify for us new fields that we want to, you know, get into, to identify what are the, you know, potential project out there that we should, you know, pay attention to. And also we recruit some dedicated scouter, both in Europe, where is, you know, we have less visibility, even in Eastern Europe. And so I think that's it in the broader picture. I can get into more details if needed. Yeah.
Sure. Leah, do you want to comment?
Yeah, sure. So we're kind of in between Chris and Beth. Like, we like to do deep dives into different fields or areas that we think are interesting. So I just actually wrapped up one on kind of chemical inducers of proximity. So what's beyond PROTACs and modulating other PTMs. And then, so usually we're disease agnostic and we look more at technologies and then kind of determine the applications from there. But it's an incredibly bandwidth intensive opportunity and series of weeks and months even to do that kind of work. What usually ends up happening and why we still are willing to invest the time is because we meet, people on the academic side who we end up working with in other capacities, even if that exploration doesn't result in a NUCO, for example. So for us, we love doing it. We stay close to all of our former SAB members of the NUCOs we've started to, and just periodically check in with them and ask them, what's something that's new on your radar, and kind of go from there. So we try to be as close to the bench science as possible.
Yeah. Yeah, there's a lot of interest in artificial intelligence, AI companies or formats. I mean, what is your interest in the potential of AI finding a new drug to repurpose for a neuromuscular disease, or and can anybody comment on that?
Yeah, I'm happy to comment, Mike. So AI ML is a foundational part of most companies that have a platform around research. If you look at, for example, today we launched one of our gene therapy companies called Capsida, and we are profiling billions at a time of modified capsids. And that's not possible to have a human process through that. And so AI ML is critical. Many of our companies that have platform technology are hiring and have, a half or sometimes more of their teams who are computer scientists, data analysts. It's really changed. And so it's a foundational piece of many, many companies. With respect to, repurposing of drugs, that was a fad. Chris will probably remember as well as I do, a good 10 years ago, a little bit more, and not much, if anything, came out of those efforts. So I'm not convinced, even with more robust computer technology, that that's the right direction. I think it maybe give you a starting point of some knowledge, but at least from my perspective, having been in the industry for a long time now, I just haven't seen that play out to, success.
Yeah, I'll just add one comment to that. By the way, it's cool that you have two panelists that had news this morning. So congratulations, Ben, on the
Thanks, same to you.
Yeah. But yeah, AI machine learning, I think that's right. Like Derek Lowe, who blogs as a chemist, he says it's not that, he's paraphrased a well-known quote, it's not that computers are going to replace chemists, but chemists with computers will replace chemists without computers. And essentially what I think this means is that you're going to have better thoroughbreds in the gate. Okay. It's not going to help you run the race. It's not going to help you win the race, but you're going to have a better starting point, and hopefully over time will increase the probability of success. But you still need good drug developers, to figure out how to apply that, how to do the regulatory strategy, how to, get the right statistical methodology, all of that. It's really hard to see how AI machine learning is going to answer that contextual information around good, drug development.
Right. Okay. I think we have a couple minutes left. So why don't we just turn it to providing a little bit of advice to the companies that are pitching today. Just some little advice of what sort of, evidence that you're looking for, what sort of exits are you looking for, from these novel programs, or any other advice that you think would be just helpful to them. So Leah, do you want to kick that off?
Yeah, so I guess I already previewed a little bit of this, but talk to KOLs early and often. I think that's a huge one, so that you can be really sure that you're solving a meaningful problem. I think one of the things that we often see is there is some benefit to just moving into an in vivo system, oftentimes a lot earlier than, you know, traditional academic scientists are comfortable with, where you get some useful data. And obviously we'll redo that in the context of the company, but everything doesn't need to be solved before you go into an animal. And in fact, animals can be useful in terms of, you know, focusing some more of that early research. So that's just, those are two very specific things that we found, you know, are true time and time again. So I'd offer those two pieces of specific advice.
Great.
I can add some flavor, yeah. So generally speaking, we are focused on early stage therapeutics, you know, across all indications and modalities. We have technologies from small molecule antibodies, cell and gene therapy, even synthetic biology, RNA therapeutics, and more. And our strong focus is on the novelty of the program, you know, namely on novel target, novel mechanism of action. And as you can imagine, we start very, very early. So it is important for us to at least have the potential to be first in class when time comes. Another aspect when, you know, evaluating potential program is programs that have strong translational potential to human. So we will look for any oil sign from human disease, any human genetic links, or any data from patient-derived cell, etc. Additional aspect in the selection process of project is project that, you know, have the capability of reaching an inflection point within the time and money that we offer, namely, you know, the $3 million over the 3 years of incubation. And I must say that we see more and more platform, you know, as I mentioned before, it's less, you know, one compound to one target and more platform technology. And just, you know, in short, when I'm looking on, you know, new opportunity, I imagine some kind of, you know, triangle. One angle is the project itself, namely how novel it is, how clear is the mechanism of action, how translatable is it to human disease. So this is one aspect. The other aspect is the people behind it, which is crucial for us in the due diligence process. So we'll look for people, you know, PI that is reliable and cooperative, and someone that is leading in this field, etc. And the third angle is the market. So we can look about, you know, perfect project, very novel, exciting project, but if there is no need and there are multiple alternatives in the market, so we probably won't go into it. So
great. And I know, Beth, you gave a lot of strong advice to the companies. Anything else that you want to add to that?
Yeah, thanks, Mike. Basically, I make a couple points. One is, remember, which is often hard when we're doing basic research, that mice are not men or women. Mice are not primates. So focus and find funding to do non-human primate experiments. It can't be underestimated how many mice we've cured of diseases that haven't translated. The second advice I would give is focus on delivery. We have seen so many really interesting technologies that can't be delivered. And, you know, the breakthrough, if you think about mRNA, you know, becoming vaccine candidates and basically saving the planet. Thank you to the companies and scientists who worked on that. It was all, you know, old delivery technology with LNPs, lipid nanoparticles, siRNA with GalNAc, but it's only addressing the liver. So think about how are you going to deliver to the right cell in the right organ your technology. And the third I would say is think about, you know, again, the mission you're on, patients, treating patients, build a company, don't focus on the exit. The exit will come if you build a great company.
Thanks, Chris.
Now, my fellow panelists have covered everything really well. I've really enjoyed listening to all of their inputs, and yeah, I just really enjoyed participating. Really great panel, Mike.
Great. Well, thank you all. I leave it to Sharon to give her last bit of advice and wrap up our panel.
Sure. So since I'm moderating the next session and I'm aware that we're a little over time, I'll still say just, you know, the one thing I will say is having worked for several companies, every company I've worked for had a blind spot somewhere. There's some assumption or something that people are hanging on to and not testing. And I would say whatever you're most sensitive about, that's probably that blind spot. So instead of protecting it, you need to really pull it apart and make sure that you understand, is this an assumption? Is this real? Are you basing your strategy on something that may or may not pan out, and really probe on that a bit instead of just accepting it. So that's my parting words.
All right, well, thank you very much. And that concludes our panel.
Thank you, Mike. Great to work with everybody today.
Thank you, all. Mike. Thanks, everybody.
Yeah, bye-bye.
And I will say thank you all as well. And we're going to move on into our next session. So now I would like to introduce Chris Barden, who's the CEO of Triventis, who is going to tell us about anti-misfolding small molecules targeting TDP isoforms in ALS. So I'll turn it over to Chris.
Thank you, Sharon, and thank you to the panel. Very interesting insights. Okay, so today I'm going to tell you about Triventis. Triventis is a company that I co-founded with a number of other scientists working in the area of computer-aided drug design, particularly in neuro, and specifically our approach to protein misfolding disease in ALS. So Triventis has a platform called Common Conformational Morphology, or CCM, which enables the in silico screening for and design of compounds that can be tailored to impact on one or more misfolded proteins. And then we leverage that platform using carefully designed experiments, cell-free, cell-based, ex vivo, in vivo, that feed back to our internal medchem discovery efforts in these diseases. Now, when I talk about the idea of precisely targeting misfolding, I'd like to spend some time explaining Triventis's thinking on this subject, because misfolding is something that has a long pedigree in neuro, certainly. When we think about misfolding, we should think about it in terms of what it contrasts to. When we have a normally folded protein, it is typically expressed, it's post-translationally modified, lives out its natural life, and is ultimately targeted for deletion by the proteasome. But when you have an intrinsically disordered protein, even when it so-called normally folds, there's some region of it which is not folded. And so, as indicated by this lightning bolt on this figure here, there's some small population at any given moment of a feedstock of an intrinsically disordered protein that is in an epitope that might be critical path to further misfolding and oligomerization. So when it collides with another of its ilk, it can potentially begin seeding, which, as this builds out in sort of a natural polymerization process, can result in what is called often in the neuro literature as soluble oligomers. And then once these things are large enough to no longer be aqueous soluble, they crash out to become fibrils, and in various diseases are referred to as plaques and tangles. I'm sure many of you are familiar with many of the controversies in Alzheimer's disease, particularly around these particular species, what to do with the plaques, what to do with specific proteins that are present. Tau in particular is a major point of discussion. And I would like to say that at Triventis, the way we think about targeting here is to say that it's not the protein that misfolds per se that is the target, nor is it to say that we're just looking at monomeric protein. We actually want to target just that portion of the monomeric protein which happens to be in the epitope, which is critical path for this misfolding event. And historically, the problem here has been that it is an intrinsically disordered protein. And so you don't have crystallographic information. You don't have the kind of structural information you would ordinarily use to do computer-aided design, rational design of compounds. And that's really where Triventis' CCM approach comes in. CCM is a in silico approach that utilizes molecular mechanics and dynamics and various computer simulations and high performance computing to model the various different ways that these kinds of proteins misfold, with a view to finding active sites and identifying epitopes that can then be drugged. And I have a video here which will hopefully play. I guess it's playing without the audio. I can narrate. As you can see, the misfolded protein is a difficult thing to get a really good notion of the conformation of, but by utilizing simulation, we can interrogate all the different regions of these proteins, identify the segments which are most likely to be important for misfolding, and then create candidate structures that are potential points of epitopes. So this allows for the identification of shape and active sites associated with different misfolded proteins. And then that allows us to do all of the things that you would ordinarily do in a drug program. We can do virtual screening campaigns against that target. We can utilize that information to identify additional features that are important for other kinds of modalities, such as PROTAC, and just generally utilize rational drug design enabled by this in silico approach. I'm trying to advance the slide, but it's not doing anything.
So you may need to click back on the slide and then you will find that it will advance. If you just click on it.
I am trying to do so, but it does not advance.
So when you hover over to the bottom right hand, you can see those two advancing arrows.
Over to the bottom.
Actually, the very bottom of your screen, it's almost grayed out. It's the bottom left-hand side. Yep, keep going down. There's actually some little arrows there.
Yeah, in my aspect ratio, I can't see them. Can you manually move past the slide? Then I can probably control from there.
Let's see. I don't think I can.
Yes, I can. I'm trying to control it right now. Just 1 second.
Okay. Thanks for your patience, everyone. Okay, I believe this will work now. Excellent. So CCM allows for this kind of work. I think in particular it's relevant to how it is that we address the therapeutic hypothesis, which is really driven by the fact that if you are designing a compound that directly targets this epitope, then you would expect that that's going to have certain kinds of downstream impacts on the overall population of these kinds of misfolded species. You're going to turn off the production of new oligomers. This isn't going to change the amount of plaque or tangle that's already laid down. It shouldn't change the concentration of monomer that exists in the overall feedstock. But what it does do over the time scale of elimination by the proteasome, which varies from weeks to months depending on the protein you're talking about, is that you establish a new steady state concentration of oligomers. And so in these approaches, this means that you ought to be able to design a compound which has disease modifying effects by halting synaptotoxicity, neurotoxicity, and transmissibility of these kinds of misfolded species in a neuroprotein misfolding disease in particular. So in our tau and A beta program, which is the one that we've made the most progress in, we've actually shown this by making novel brain penetrant drug-like small molecules and demonstrating dose-dependent reduction in vivo of oligomer synthesis for both A beta and tau with the same chemical matter. And we can do that at pharmacologically relevant concentrations. So we're working towards building out an IND package for this with our lead TRV301, and in fast followers throughout this year as we bring that to candidate declaration. The potential area of applicability of this kind of approach is really quite large, apologies, I can't advance anymore.
So
when I click on the screen, it doesn't advance anymore.
Well, have you tried the arrow keys on your keyboard? That's usually foolproof.
Okay.
the last.
Sorry, sorry, I hadn't realized that I could even do that in this software. All right. Okay. All right. So, in the interest of time, of course, and because that's really the focus of today, I'm going to focus mostly on the ALS program. So in our ALS program, which we're working together with a number of collaborators on, we are really focusing on TDP isoforms. So the literature findings of this, particularly in sporadic ALS, is that TDP-43 is relevant to many such patients. It's also relevant to patients with frontotemporal dementia, and in fact, it appears about half and half frontotemporal dementia versus tau in terms of the overall impact of the pathology in those diseases. So much as we've done in tau, our intention is to develop small molecules that inhibit the misfolding of TDP isoform oligomers, TDP aggregates, through broad and precise targeting of early misfolding events. And these events, when I say early misfolding, are dynamically occurring throughout the disease. We're simply talking about being able to block the formation of new seeds of particular proteins, in this case TDP. And you know, particularly in this area, we're talking about that we want to design something which impacts on the common intrinsically disordered domain present in biologically relevant isoforms, so that we can impact on even aggregates which are heterogeneic. There's a lot of literature that suggests that the ectopic irreversible aggregates found in the vicinity of deceased motor neurons in ALS models are actually composed of both TDP-43 and 35, and that potentially 25 plays a role as well. So I think the ability to inhibit misfolding across isoforms may be quite important. Our early work in this area used compounds from our CCM design library of anti-misfolding small molecules in collaboration with ALS and zebrafish expert Dr. Pierre Drapeau. And we collected extensive data in zebrafish and C. elegans models to screen these CCM-enabled compounds. And we identified a tool compound that we call TRVA-242 that performed well on all of these studies. TRVA-242 rescues early motor behavior and locomotor deficits in this particular mutant TDP gain of function model in zebrafish. And you can see that this compound rescues wild type swimming phenomena. The phenotype is rescued in terms of swimming distance, swimming duration, and velocity with this compound. We've also been able to show positive data in electrophysiology, specifically looking at synaptic deficits at the neuromuscular junction that would normally be found for this mutant TDP zebrafish, but was rescued in the presence of Treventis compound. And we have extensive other data, which is in the paper if you'd like to look at it, looking at synaptic developmental morphology as well as axonal length that was likewise rescued for this model. So that early work built the ground for our hit to lead work that we have been doing throughout the last year in building out cell-free systems and making new compounds in 7 different chemotypes to demonstrate that we can have this impact in a general sense. We've been doing this work in conjunction with our SAB member, Dr. Janice Robertson, who's well known for her expertise in ALS biology. And so we've developed compounds in this program that have in vitro potency in anti-misfolding cell-free models. There's selectivity for TDP over tau, which is important for development of distinct chemical matter for the different diseases. And we have, through this GFP tag TDP cell model, a method of showing rescue of motor neurons and reduction of ALS-related aggregates in the presence of compounds, as well as showing that in an arsenite experiment for the same cells, you can show effects that do not obtain in regards to normal reversible stress granules. So that's an important counter screen and demonstrates that making a compound that affects misfolded proteins doesn't have to affect normal folding or even normal aggregation processes that we might see in biology. So we're really on course for doing an accelerated LO around this. We have many of the, if not most of the experimental capabilities in place now, and really our capstone in terms of being able to demonstrate that we have a lead compound in this program will come through the later part of this year through a proprietary mouse model that's being developed to demonstrate that we, like in the tau models, can impact on the formation of TDP oligomers and then downstream aggregates in the presence of Traventis molecules. So to summarize the approach in ALS, we're targeting multiple forms of TDP with the same chemical matter. We're building out a robust preclinical package in a number of different chemotypes, looking in systems cell-free, cell-based, ex vivo, in vivo, and we are pushing through to be able to produce a candidate targeting the latter half of next year. Traventis has a strong team, a strong base of researchers and entrepreneurs working for over a quarter of a century, in some cases in neurodegeneration, and a lot of support from prestigious granting councils as well as investors in this area. And when we look at the people involved at Traventis, there's myself in the computer-aided design area and serial entrepreneur. But I'd also like to point out Dr. Don Weaver, who's a medicinal chemist as well as a neurologist, and one of the inventors of tramiprosate, which is currently in phase 3 clinical trials with Alzion for Alzheimer's disease. So we have quite a bit of expertise across the continuum of drug design and development within Traventis. So Traventis is really trying to stop protein misfolding diseases as possible. And so we're poised to become a clinical leader in this space. It's a space which is a new area that cuts across therapeutic indications. And there are other companies that are working in here, and for time reasons didn't get into that, but certainly as a small molecule company, we have a particular modality in mind here, which obviously is well suited for delivery into target tissue of CNS. And we are seeking throughout the next 2 quarters to have conversations with people either on support of this program or support of the company in general through investment in the platform of CCM and what we're doing here. And I thank you all for your attention.
All right. Thank you, Chris. We appreciate that. You've gotten us almost back on schedule, so that's wonderful.
I aim to please.
So I don't see any questions up right now. I think in the interest of keeping us on schedule, we can move on. But if you want to follow up with Chris, his contact information is in the PDF compendium. So thank you very much. Okay, so we're going to move on, and next we will hear from Irving Sochalecki, and if I've massacred that, I apologize to you.
Sochalecki.
Sochalecki, it was pretty close. Matrix metalloproteinase inhibitors for the treatment of amyotrophic lateral sclerosis. So from Aquilis Pharmaceuticals, go ahead.
Am I coming in clear?
You are.
Okay. First, I would like to thank the Muscular Dystrophy Association and Liz for inviting me to speak to you today. I'm going to go through our presentation rather quickly to allow for ample time to answer as many questions as possible. Our company, Aquilis Pharmaceuticals, is looking for support to advance a late preclinical stage therapeutic for the treatment of amyotrophic lateral sclerosis. I will not spend too much time describing the disease, since many of the previous speakers have already done a fairly good job. However, for those that may have come in late or missed some of the presentations, I will summarize some of the key points. Let me see here. Let me see if I can move forward. All right. All right, that's good. ALS is a neurodegenerative disease that affects about 30,000 people in the US, with roughly 5,600 new cases diagnosed each year. There are 2 forms let me see if I can get here. There are two forms that encompass of the disease. There's the familiar form that accounts about 5 to 10%, and there is the sporadic form that encompasses about 90 to 95%. A very unique and scientifically interesting aspect of the disease is that it begins within fast-twitch fascicule, or FF motor neurons. These are the motor neurons responsible for running, jumping, speaking, breathing, and swallowing. But the disease does not affect slow twitch, or S motor neurons, which are the cells that are responsible for eye movement. Matrix metalloproteases, or MMPs for short, are a family of about 24 metalloenzymes that are responsible for digesting the various structural components of the extracellular matrix. Around 2008, various researchers such as Professor Veronika Shubayev, who is at the University of California at San Diego, and Professor Rurong Ji, who was at the time at Harvard Medical School and is currently at Duke Medical School, discovered that a subset of MMPs consisting of MMP2 and MMP9 were upregulated in both glial and astrocyte cells during the initial stages of nerve damage and inflammation. For example, during the initial stages of nerve damage, it was discovered that MMP9 can break down myelin basic protein, which is a key component of the outer sheath of axons that extend from motor neurons. It was discovered that the myelin basic protein fragments elicited an almost immunogenic response, causing neuroinflammation, leading to elevated caspase levels and apoptosis of neuronal cells. MMP9 and MMP2 have also been observed during nerve damage to cause the initial stages of inflammatory cytokine activation. For example, both MMP2 and MMP9 can convert inactive IL-1 beta to its active form. There have been several studies that have shown elevated levels of MMP2 and MMP9 in the skin, cerebral spinal fluid, and blood of people with ALS or PALS. Microarray profiling studies in SOD1 overexpressed mice have shown that MMP9 is one of the few genes of approximately 18 that were overexpressed by greater than 10-fold, specifically in FF motor neurons, as compared to the S motor neurons. And that when you do reduction via knockdown of MMP9 or through MMP9 inhibition, it caused a significant delay in muscle denervation and an overall enhancement in lifespan by about 25%. Okay. Currently, there are no FDA-approved MMP inhibitors. Aquilis, over the years, has developed an orally bioavailable, dual-active MMP2 and MMP9 inhibitor that has exhibited a very good safety profile in mice, rats, and dogs, and has been shown to be efficacious at reducing neuronal damage in several rodent animal models of neuropathic pain. In a key rodent model of nerve injury, we have been able to show that oral dosing with AQ118 was able to reduce caspase-3 protein levels in the dorsal root ganglia, leading to a reduction in neuronal cell apoptosis and a reduction in neuropathic pain symptoms. As part of our PK studies, we have been able to directly measure high nanogram levels of compound in the spinal cord as a result of oral dosing. We're able to increase those levels by increasing the oral dosing. Aquilis has tested AQ118 orally and in other models, in particular in the TDP43 and SOD1 overexpressed fruit fly model, and found that it enhanced larval locomotion to a statistically significant extent as compared to vehicle controls. We then tested the compound orally in the SOD1 overexpressed mouse model. at disease onset and observed an increase in mean survival of up to 6%. Lastly, and most importantly, we performed a series of biomarker studies in the serum of a group of PALs and found that they had a greater than 7-fold increase in their total MMP9 protein level as compared to healthy age-matched controls, which was around 57 years of age. We then compared the PALs group to a sample set comprised of a group of age-matched type 1 and type 2 diabetics having been diagnosed with peripheral diabetic neuropathy. We did this because we believe that diabetic neuropathy is an inflammatory disease that is derived through the exposure of oxidative stress and chronic low levels of inflammation leading to peripheral nerve damage. To our surprise, rather than observing similar levels of MMP9, the PALs group had a 5-fold higher MMP9 level as compared to type 1 and 2 diabetics with diabetic neuropathy. In our opinion, these biomarker results are compelling evidence of the relevance that MMP9 has to this disease. Aquilis believes that ALS is a result of a systemic-wide inflammatory process initiated through some misfolding event that triggers an overexpression and production of MMP9 in FF motor neurons. Whether the increase in MMP9 is a result of the trigger of these misfolded proteins inducing an inflammatory response, or as a way of activating the process of autophagy, which is a natural mechanism the body uses as a way to break down the misfolded proteins such as SOD or TDP-43, is not known. Whichever the case, we believe that MMP9 levels rise uncontrollably, resulting in the breakdown of various vital CNS proteins, such as myelin basic protein and neurofilament, and as well the activation of various inflammatory cytokines such as IL-1 beta, IL-6, TNF alpha, as well as caspases, all leading to motor neuron death. We believe that introducing an MMP9 MMP2 inhibitor, such as AQ118, has a good chance of impacting disease progression. We have completed the bulk of our animal studies on AQ118, which so far has shown a very good safety profile in mice, rats, and dogs. We only need one rat and dog study and a formulation stability study to complete our IND enabling package, which we estimate to cost about $750K. AQU118 represents unique composition of matter IP, whose patents are fully issued in the US and abroad. Lastly, I'd like to go over our management team, which comprises myself. I have 24 years of industrial experience as a pharmaceutical researcher working in various therapeutic lead generation and optimization programs. We have Dr. Daryl Nix, our Vice President of R&D, who has 18 years of experience in the pharmaceutical industry in both preclinically and clinical programs. Dr. Roy Suchaleki, our Vice President of Clinical Development, which I'm not sure if he's listening in. I gave him the link. And who's our Chief Medical Officer and who is a board-certified practicing neurologist and staff member at Northwestern Medicine Central DuPage Hospital. Lastly, for those interested in receiving a copy of the presentation, please email me. I would be happy to send it out to you. I also would like to send out an invitation to those researchers that are interested in testing our compound, especially those that may have a TDP-43 overexpressed rodent model. We're very interested in seeing how the compound impacts that in that model. And lastly, I'd like to thank you all for your attention, and hopefully with enough remaining time, I can answer as many questions as possible.
Okay, well, thank you very much. I'll give it a minute. We have a little bit of time, so I'll give it a minute for some questions to come in. But, you know, while we're waiting, it occurs to me that this is a pretty different approach to ALS than I've seen, you know, looking over the field. And I wonder if you've thought about the potential for combining this with other types of approaches that would hit different pathways, and what you think of.
Well, the main hurdle that we found has been generally there has been in trying to approach and trying to raise funds, there's been kind of a hesitancy because people felt that the target is somewhat not as well known or validated. So I think if one of the things that we've been interested in is trying to get the compound into the clinic as soon as possible, especially into PALS. We have a pretty high confidence in terms of the safety of the compound. The key issue, I think, is timing. So as far as in combination with other medications, because we have such a high confidence on the safety of the compound, I think there's nothing wrong with trying to do that. And I would encourage, if anyone is interested, we'd be happy to work with you in seeing how the compound would work in combination with, say, other strategies.
Right. And so obviously you just showing proof of concept with your compound alone would be the priority. It was just sort of a thought that it has the potential to be a good combinatorial product as well. So I don't see any additional questions coming in right now, but if you have anything else you want to bring up or maybe address, maybe people could follow up offline. If you have questions that you would like to put to the audience.
Well, only that right now we're interested in trying to look at TDP-43 animal models. And if someone is currently actively involved with that, we'd be interested in collaborating and seeing how the compound behaves. But at this point, we're so close to an IND that, frankly, the most important thing for us is to get into the clinic and to basically try to see how, in terms of biomarker profile, how this behaves. We have a pretty good strategy in which we'll look at the MMP9 engagement through inhibition, and then we'll be monitoring on the levels of myelin basic protein and neurofilament to see how it impacts in terms of its mechanism of action.
Do you feel pretty good? Do you have a clinical strategy mapped out already then?
Yes, we do, but obviously the one thing that's missing is funds. But we've spent quite a long time interacting with various thought leaders, and clearly one of the things we've thought heavily is on the use of biomarkers as a way to monitor both engagement and to see how the inhibition is affecting other compounds that are being targeted. Obviously, if you're trying to impede the MMP2, MMP9, and inhibiting them, then we're going to carefully look at neurofilament, myelin basic protein, and IL-1 beta levels to see basically if we're impacting that. In addition to obviously motor function.
Well, I know your contact information is in the compendium under Aquilis Pharma, so people can reach out to you if they want to follow up afterwards and have any further thoughts or have a mouse model that might be of interest. So,
thank you very much.
All right, thank you. Appreciate that. So we will go on now to our next speaker, who is going to be Dr. Akinori Hishiya.
Yes, right. So Akinori Hishiya. And first, I'd like to thank you so much for this great opportunity. This kind of opportunity is very important for small biotech companies like Sora. Okay. Okay, so first I'd like to introduce about Sora Biosciences very briefly before we move to scientific parts. So Sora Biosciences is self-funded biotech companies and still young.
Oh, it looks like Dr. Hashia is frozen. Let's give him a moment. Okay, we will try to reach out to Dr. Hashia to see if we can reestablish its connection. So just give us a moment, please. And I apologize that we don't have some nice chamber music to play while we're on standby. We'll know this for next time. Hopefully it will only be a moment though. Okay, we're just going to give it one more minute. If we can't get Dr. Hashia back, then we will go ahead and move on with Dr. Lingapa. So let's just one more minute. But Dr. Lingapa, please be standing by. Just in the nick of time. Can you hear us?
Yes. Can I start again or should I wait?
I think she's going to pull your presentation up again.
All right, sorry.
So it'll be just a moment, no worries. It happens. I'm sure we're just loading up that presentation.
All right, sorry. So where were we? Actually, it's the very first part. So actually, this is a threat management team. And then the next slide is, can we move to the next slide?
So if you click on the presentation and then use the arrow keys on your keyboard, that should move it forward.
It doesn't work. Oh yeah. So and this is our sub-members, and Jerry is experienced at many clinical trials for rare diseases, and Clotilde is from the Cleveland lab at the UCSD, and she has her own laboratory at MGH now for ALS research. And Miguel is a specialist for the AAV gene therapies, especially capsid designing. All right, so now we know that many diseases are actually caused with protein folding issues. And this kind of disease is so-called conformational disease, such as ALS and Alzheimer's disease. And for instance, Alzheimer's disease is characterized by a senile plaque in which the misfolded and aggregated amyloid beta peptides have been found. And neurofibrillar tangles, of which major component is hyperphosphorated aggregated tau proteins. So the current hypothesis is these misfolding from proteins start misfolding with age, and at some point they exhibit the neurotoxicity leading to a neurodegeneration. So the suppression of misfolding or elimination of already misfolded the disease-causing proteins can be a fundamental treatment for many neurodegenerative diseases such as ALS. And this kind of intervention is called chaperone therapies. Chaperone therapy is mainly categorized into three strategies. The first strategy is a small compound called chemical chaperones. And the second strategy is like HSP90 inhibitor to induce the expression of set of chaperone proteins. And the third strategy is gene therapy to directly manipulate the expression of chaperone proteins. However, the chaperone therapy has not been established yet. The SORA also tested several chaperone therapies before, but the results were inconsistent. And even if we found some effects, the effect was very weak. Our hypothesis is chaperone therapy had a very weak activities due to the lack of specificity. Suppose we activate or express chaperone proteins in a cell. These chaperone proteins can theoretically work for any kind of proteins. Therefore, the direct effect on disease-causing protein is limited. Besides, off-target effects have been concerned. So our idea is if we can convert chaperone activity onto a disease-causing proteins only, we would see a very strong and potent activities with minimizing the off-target effects. With this idea in mind, we started developing these platform therapies. We first conducted screening to find the protein fragment, protein domains, which has protein folding activities, and we found the J-domain sequence. J-domain sequence exists in J-domain proteins, and the J-domain protein is a critical factor for HSP70 chaperone systems. HSP70 chaperone proteins binds to misfolded proteins, and through the structural change of HSP70, misfolded proteins are unfolded. Therefore, unfolded protein has another chance for protein folding. HSP70 chaperone system is also coupled with degradation systems such as ellipsin proteasome system or chaperone-mediated autophagy systems. And some of the misfolded proteins are directly transferred to degradation systems. Therefore, HSP70 chaperone system plays a pivotal role in both protein folding and degradations. HSP70 does not work alone. It needs assistant proteins called co-chaperone proteins, and the J-domain protein is most important co-chaperone proteins. The function of J-domain sequence is first, it binds to HSP70, and second, it activates the HSP70 chaperone systems. Okay. So what we want to do is to control chaperone systems specifically to disease-causing proteins. And this is how we do. The J-domain sequence, again, J-domain binds to and activates HSP70. This J-domain sequence was conjugated to a targeting domains. Targeting domains binds to a disease-causing proteins. Let's say we want to target tau proteins. Then targeting domains has affinity for tau molecules. And we designed fusion molecules named targeting J-domain proteins. This fusion protein is expressed in a cell using these gene therapies. And in the cell, the fusion protein first binds to disease-causing proteins through targeting domains. In parallel, J-domain brings HSP70 to a complex. The next step is especially very important. Only if disease-causing protein is misfolded, disease-causing protein is transferred to HSP70 because HSP70 recognizes only misfolded proteins. And once disease-causing protein is transferred to HSP70, J-domain activates the chaperone systems. Therefore, disease-causing protein is folded or disaggregated or even degraded. So the next is how we design. The J-domain sequence is derived from natural J-domain proteins. And we have already optimized the sequences, so we don't need to touch the sequence. What we do is to design targeting domains. Targeting domains can be a peptide sequence which is screened through a phage displays, or can be an antibody-like sequence such as single-chain variable fragments. For instance, Huntington disease is caused by expanded polyglutamine repeats in Huntington proteins. Therefore, TOR175 carries binding sequence for polyglutamine storage. And the Parkinson disease is characterized by Ruby bodies, and the major component of Ruby body is misfolded alpha-synuclein proteins. Therefore, this compound has alpha-synuclein binding sequence. So from this slide, I'd like to focus on ALS. And I don't need to go into details about ALS, but quickly, ALS is devastating neurodegenerative disease affecting both upper and lower motor neurons. And there is no treatment for ALS. Surprisingly, more than 90% of ALS patients have a protein aggregation in its nerve cells. And the detailed analysis of aggregates revealed one protein called TDP43. The TDP43 is DNA RNA binding proteins with versatile functions such as RNA splicing, transport, and stability. In the aggregates, TDP43 is misfolded, phosphorylated, ubiquitinated, and even proteolyzed. These misfolded TDP43 are highly toxic to nerve cells. Therefore, TDP43 targeting therapies can be a fundamental treatment applicable to many or most ALS patients. However, this therapeutic has not been realized yet. The main reason is TDP43 is essential for CNS functions. The suppression of TDP43 expression causes neurodegeneration. Conversely, the false expression of TDP43 also causes neurodegeneration. Therefore, direct manipulation of TDP43 expression is not feasible. The Biogen is developing antisense oligonucleotide targeting ataxin-2, and ataxin-2 is believed to modulate TDP43 aggregation through stress granules formation. When we think about the difference between a functional TDP43 and toxic TDP43, the biggest difference is protein folding status. The functional TDP43 is folded, while toxic TDP43 is misfolded. And our therapeutics can control protein folding and degradation, specifically disease-causing proteins like TDP43. Besides, the effect is totally dependent on protein folding status. And we can manipulate only misfolded TDP43 by these therapeutics. So we designed our fusion molecules for TDP43. The J-domain sequence, again, J-domain sequence binds to and activates HSP70. This J-domain sequence was conjugated to a TDP43 binding sequence. And we named the fusion proteins SOL257. When normal type of TDP43 is expressed in a cell, the TDP43 is dominantly localized in nuclei. However, pathogenic TDP43 are mainly localized in cytoplasm and eventually form the aggregation. Very interestingly, we found that SOL257 has two main activities. The first activity is SOL257 strongly suppress the aggregation formation of TDP43 like here. And the second activity is SOL257 also accelerated the degradation of TDP43 through autophagy. And importantly, SOL257 does not have a significant effect on a normal type of TDP43. So SOL257 successfully distinguishes pathogenic TDP43 from normal type of TDP43. Right. And when pathogenic TDP43 is expressed in cells, the pathogenic TDP43 is phosphorylated and aggregated. So this data is fractionation assay. So we fractionated this lysate into a soluble fraction and insoluble fractions. And this data shows that the pathogenic TDP43 is phosphorylated, and the phosphorylated species are dominantly fractionated into insoluble fractions. And SOL257 strongly eliminated the phosphorylated and insoluble TDP43. Yeah. So in advanced clinical trials, we are going through 4 stages. The stage 1 is the production of AAV. And the stage 2 is to confirm the expression of SOL257 in vitro. We use the primary mouse cortical neurons and we confirm the expression. And in stage 3, we injected AAV into wild type mouse to confirm the safety and expression. We injected SOL257 AAV through intrathecal. And the data shows that the SOL257 administration did not cause any abnormalities in body weight and behavior. Also, we injected SOL257 via ICV, but ICV also shows that the ICV administration did not cause any abnormalities. And we also confirmed the expression of SOL257 in motor neurons in spinal cord. And this data is from ICV administration, and we observed very strong immunoreactive signals. The expression can be sustained at least for 8 weeks. And in stage 4, we injected the SOL257 AAV into ALS mouse models. This mouse model expresses pathogenic TDP-43 after removing the doxycycline from their food. So this is inducible systems. After removal of doxycyclines, the mice develop TDP-43 pathologies such as phosphorylation and mislocalization and aggregation of TDP-43, and neurodegeneration and gliosis, and eventually die within 5 to 6 weeks. We injected our SOL257 AAV into the mice through ICV first. And we are very surprised because this is very aggressive mouse models. But however, this mice develop TDP-43 pathology and lose their body weight, but SOL257 treatment attenuated body weight loss. The orange line shows that the control AAV treated mice, and the gray line shows that the SOL257 treated mice. The blue line shows that the mice fed with doxycycline, so the no TDP-43 was induced. And we are very pleased to show this data for the first time. Actually, we got this data in this mouse, so very new data. Also, all mice treated with control AAV died within 5 weeks. However, very surprisingly, the SOL257 treated mice survived 100%. This is very surprising data. And the study was conducted at Psychogenics, the very well-known CRO companies dedicated to in vivo disease mouse models studies. And then we talked with them, and they say that they have been running the studies with these mouse models, but they had never seen the result this good. So this definitely encouraged us to repeat the studies with more mice and the different administration route and the timings. But now we are analyzing the histology from these mice. Okay. So this is my last slide. So actually, ALS is a very brutal disease, and we can't imagine how scary the ALS and advanced form TLS is. But there's no treatment. This is unacceptable. We need to deliver hope and treatment to a patient as soon as possible. To accelerate the programs and the development of SOL257, SORA definitely needs the financial support or partners or collaborators. So if you'd like to discuss more or you'd like to know more about our products, please feel free to contact me. So we'd be very happy to discuss in more detail. Okay, so thank you very much. And I would be happy to take any questions.
Okay, thank you. It looks like we have a couple of questions that have come in. One question is, how many mice were treated in the TDP-43 NLS experiment?
So actually we used 10 mice. 10 mice were treated with SOL-257. So the mouse number is very limited to this time, but for the next study, we definitely want to increase the mice to 16.
Okay, and then also, does this therapy result in the return of TDP-43 to the nucleus? Since the real toxicity of TDP-43 in humans is not the aggregates, but the loss of its nuclear localization and function. And then also, does it allow for normal TDP-43 RNA metabolism? In other words, eliminate the altered RNA splicing, now widely accepted as the main dysfunction of TDP-43 loss of function?
Yeah, this is very important questions. And this is exactly what we want to do. And but we got this result very, very recently, just 2 or 3 weeks ago. So now we are analyzing the histology to carefully examine the TDP-43 localization. And also we have the frozen samples, so we are analyzing the RNA metabolism as well. So this question is very, very important for us.
And one follow-up from me, are you also, when you do your bigger study, are you planning to let some of those mice survive for longer, if they will survive? To see the extent.
So actually this time with the necropsy was done at 5 weeks after doxycycline removal. The reason is first we discussed with the study coordinator of psychogenics, and they said that this mice develop the pathology very quickly, so you wouldn't see any efficacy. Therefore, we decided to go with the small number of mice, and we decided to focus on histology. So the necropsy was done at 5 weeks, but for next studies, we definitely want to check how long the SOL-257 treatment can improve its survival.
Great, it's a really interesting result. Thank you so much.
Yes, thank you very much.
Okay, so if you're interested in getting in touch then with Dr. Hishiya, you can find his contact information in the compendium. So now we'll move on to Dr. Lingappa, who will talk about small molecule assembly modulators, a novel approach to ALS therapeutics for Prosetta Biosciences.
Thanks so much. Can you hear me?
We can hear you.
Great. Okay. So since I think most people aren't familiar with assembly modulators, I'm going to start with a couple of background slides on that approach before turning to our ALS program. Biochemical pathways are quite complex and have been classically viewed as governed by the individual enzymes of each step, thought of as the individual proteins originally identified with those activities. But there's now a growing appreciation that proteins don't act as individuals, but rather as parts of multi-protein complexes. And there's also the appreciation that allosteric sites, which are the control panels by which particular enzymes are controlled, are going to be more complex in a multi-protein complex. Now, we started focusing on a somewhat different question that has come full circle to this issue. And that question is, wait a minute, a multi-protein complex, you have a number of different proteins that are together, but they're made individually on ribosomes, and the cell is an extremely crowded place. How do they form? Is that formation spontaneous or is it catalyzed? And given the crowding in the cytoplasm, the spontaneous explanation, which has been historically the one people have assumed, seemed implausible. But then why has nobody found these enzymes of assembly? Well, we've found them, and we now understand why they've been missed for several reasons. First of all, they act co-translationally while the chain being added to the multi-protein complex is nascent. And that's a minuscule portion of that protein's life cycle, making it very hard to detect. Second, those assembly machines, as we call them, those enzymes of assembly, are very transient in their action. They catalyze formation and then fall apart. It's how they maintain homeostasis, most likely. But in any case, it makes them difficult to detect. Furthermore, they're energy dependent not only in their action, but in their formation. So classical approaches, biochemical approaches, won't detect them. And then finally, there's a growing appreciation for protein heterogeneity. And it's not just the moonlighting functions, multiple functions that people are finding, but perhaps just as important, that heterogeneity of any given gene product's role in many different multi-protein complexes makes the one function you want to look at in this particular multi-protein complex assembly machine difficult to detect. So now add to the problem of finding an allosteric site rather than the active sites that most drugs today target, which is not physiological, not homeostatic, finding among the many allosteric sites in a multi-protein complex, which is the one you want to target. And we've been working largely under the radar to do three things. One, develop the tools and assays needed to drug the enzymes of assembly, as we call them, assembly machines. Find those drugs, which we've done, advance them, and preclinically validate them through a target product profile. And finally, better understand this very different way of thinking about mechanism of action in health and disease. Okay, so here is the conclusion of a lot of work we've done in a number of therapeutic areas before I turn to ALS specifically. There's a normal assembly machine. It is transiently coming together and then falling apart. What's it doing? It's building physiological structures that maintain homeostasis. Along comes a disease-triggering insult of many different sorts, and the net effect is an aberrant assembly machine. Some of the components are lost, different ones are added, and you get an assembly machine that doesn't do what it's supposed to do. And we have found two ways to fix this problem. One is to identify a small molecule that tweaks the same allosteric site that caused the disease and drive it back to normal. Another approach is to drive this aberrant assembly machine into autophagy and let homeostasis repopulate with the normal. And we started out by establishing this paradigm for viruses and then moved on to non-viral disease. And with regards to neurodegeneration, there's an extensive literature on the enigmatic role of viruses in neurodegeneration. They don't cause neurodegeneration generally, but there is a connection, and that connection appears to be that viruses manipulate host assembly machines in ways that mimic the pathophysiologic mechanisms involved in neurodegeneration. And initially, to our surprise, what we found is that assembly modulators are strikingly therapeutic in a variety of neurodegenerative disease models. And in particular, our influenza active small molecules were effective against alpha-synuclein aggregation. Our herpes active compounds were effective against tau hyperphosphorylation and A beta oligomerization. And point of this discussion, our retroviral active small molecules targeting host assembly machines that are repurposed by retroviruses for their needs are effective against motor neuron degeneration. So, cellular models for TDP-43, there's a nucleocytoplasmic localization assay. If you look at normal cells, you find the TDP-43 in the nucleus where it belongs. If you look at ALS-derived, patient-derived cells, you find often the TDP-43 leaked out into the cytoplasm to different degrees, but to one extent or another. And when you treat with our advanced assembly modulators, it restores everything to normal. Alternatively, there's a stress-induced aggregation of TDP-43 assay where you see TDP-43 aggregates in stress granules. The same assembly modulators fix that. So you have one class of compounds effective on both pathophysiological hallmarks. Now, if you look at sporadic ALS patients, however, you see a remarkable heterogeneity. By the nucleocytoplasmic assays, some patients have massive mislocalization, others modest mislocalization, and some no obvious mislocalization. Everybody that has the mislocalization, however, is corrected by assembly modulators. Those same patients looked at by the stress granule assay, their cells show that, in fact, the one that showed no mislocalization has massive stress-induced aggregation. The others show that to a greater or lesser extent, and all are treatable by the same assembly modulators. So I don't have time, so I'll summarize further cellular and animal validation we've done over the last several years in iPSC-derived motor neurons. Initially, we worked in the C. elegans transgenic model, showing striking efficacy at every parameter looked at. Then in the transgenic fly with the C9 ORF72 repeat expansion model, showing striking rescue of multiple parameters. There's just a pretty picture that speaks to that. And then in the SAW G93A mouse, showing by every criteria, biochemical, neurophysiological, and clinical efficacy. And then the pharmacokinetics are very reasonable of these compounds with excellent brain exposure. And we have now achieved biochemical evidence of target engagement. And the way we've done that is with one of these new tools I alluded to, which is energy-dependent drug resin affinity chromatography, which we use the acronym EDRAC. And I don't think anyone's ever done this before, but it's quite striking in that if you take the brains of the mice in the study I just alluded to, you find that RAN-GTPase, this is now looking at the normal versus aberrant assembly machine that has bound to the drug resin. The normals don't have it. It gets worse, gets dramatically increased with disease and returns to normal with treatment, both for RAN-GTPase and TDP-43. And again, we're looking at a tiny subset of total cellular RAN-GTPase and TDP-43 that's in the normal versus aberrant assembly machine. The same response to treatment is seen in the patient-derived fibroblasts. And by photocross-linking, one can show that the normal patient-derived cells do not show the RAN-GTPase cross-link as the direct binding partner of the drug, but both sporadic and familial ALS do. So we then asked, could that EDRAC signature be used as a peripheral biomarker? And strikingly, in normals, you see none of the post-translationally modified RAN-GTPase that is seen in essentially every ALS patient we've looked at. 13 of the first 14 patients to date. And in all of them, the signature gets worse with progression of disease. And patient 78 was striking to us because she walked into the clinic immediately after her diagnosis and still driving, just using a cane, and already we could detect her signature. And by the time she was disabled, it was massive. Meanwhile, P62, a regulator of autophagy, which is highly present in the normal assembly machine, basically the reset button, if you get the colored wheel of doom on your, to reset your operating system, that is lost in ALS, much depleted and lost with time as ALS progresses. Whereas the same patient-derived fibroblasts that the drugs show efficacy on restore the autophagic markers as they return to the normal assembly machine. So this raises the possibility of detection of ALS early, before onset of disability, by using the same drug as a ligand for detection, a way to select patients for trial enrollment, monitoring therapeutic efficacy, and I suppose ultimately monitoring the entire Medicare population to identify patients as early as possible before onset of disability. So I've already alluded to this. Why have others overlooked assembly machines? Because they're very hard to detect. You need the tools. We've spent an enormous amount of time developing to detect them. You need to target the right allosteric sites. And we turn the viruses into truffle hounds to tell us what is the most effective allosteric site to go after. That's what our drugs are targeting. And I would say genomic-centric thinking overlooks the implications of biogenesis, co-translational assembly, protein heterogeneity, and multifunctionality, which makes it almost impossible to detect what we've been able to detect. And if you can't detect it, you can't drug it. We've drugged it and driven the SAR to therapeutic efficacy. So the conventional view, I would say, is that normal proteins misfolded are either degraded or form aggregates, and the aggregates cause disease. For the reasons I've summarized, we don't believe that model. We think instead there's an assembly pathway that builds what? Physiological structures, synapses, what have you, and along comes a CNS disease triggered. You get an aberrant assembly machine that pathological signaling complex causes disease. Oh, and yes, there's aggregates too, but that is the wreckage after a car crash. If you want to prevent the car crash, don't call more tow trucks. Instead, put in a stop sign to prevent the aberrant assembly machine. That's what we believe our drugs are doing. So, in summary, a very novel orthogonal approach to drug discovery applied to ALS, encompassing both stress granule dysfunction and nucleocytoplasmic dysregulation, effective on both familial and sporadic ALS in cellular models and in the available animal models, and a range of ALS-relevant mutations with target engagement established, promising early patient peripheral EDRAC signatures as biomarkers, managing the pathophysiological and clinical heterogeneity of the disease, pretty much at target product profile, with diagnostics and therapeutics integrated, and in search of funders interested in a completely different approach to ALS and other difficult diseases. Maybe I'll stop there and open it for questions. Thank you very much.
Okay, so thank you. We have one question. So you referred to protein assembly. Does this agent work to reassemble the altered nuclear pore seen in C9 ALS and sporadic ALS, which could explain the normalization of the RAN-GTPase?
We've not looked at the nuclear pore complex per se. On the other hand, the presence of RAN GTPase in our assembly machines makes that hypothesis a very appealing one. I mean, again, we have not had the bandwidth to do more than the minimum we needed to do to confirm our way of thinking about this disease.
Okay. Well, that's a very interesting, it's interesting to hear something so different. We appreciate that presentation. Next, we'll hear from Dr. Antonella Favit Van Pelt, who is the president and chairwoman of the board of Thera Neuropharma, who will talk about treating incurable neurodegenerative diseases with an integrated technology platform and a synergistic effect. Yeah.
All right, thank you very much for the opportunity to present. Great presentations. I'm very impressed by everybody who had presented their data. Surely the field is moving very encouraging to a treatment or to potential treatment. And my presentation is going to be slightly different than others. We have not made our scientific data public, so I'm going to give you more of a strategic view of our company. So if I can just move my slides, is there something I need to do to move my slides?
Yeah, so if you will click onto the slide itself, and then you should be able to use your keyboard arrows to move it.
Yeah, okay. I am clicking to the slide, but it's not happening.
You click the slide and then use your keyboard arrows. That usually works.
Oh, okay, so we just, oops, hold on 1 second. Okay, here we are. All right, so we, Thera Neuropharma is the research and development arm of Synerion Therapeutics, which is our holding corporation. And we are a company focusing on delivering therapeutic treatments for neurodegenerative diseases. Our vision is to treat neurodegenerative diseases with an integrated technology platform that has potential for synergistic approach. And both platforms are positioned to leverage the brain's ability to compensate and restructure while silencing or mitigating the pathogenic causes. So we believe that the company, has a good investment proposition. We are targeting neurodegenerative diseases, which are normally multi-billion healthcare burden. We have two patented technology integrated platforms. So one is a small molecule regenerative therapy, and the other one is a SOD1 protein targeted self-delivering RNA interference. And this technology are complementary positioned to be developed in multiple indications, though our intention is not to develop them together, but separately, and then find opportunity to integrate them. We believe that we will have a good probability of success because in addition to our therapeutic platform, we also have, we're exploring also the potential to companion a biomarker. We have very positive preclinical data and animal proof of concept. We have also identified a clinical candidate. The market size, as as everybody believe, I believe know, it's a multi-billion dollars worldwide market, and we are positioned to value inflection at the latest 3 to 4 years, and that will be when we complete our phase 1 trial. But we could be positioned for an earlier inflection point within a couple of years once we submit R&D for a clinical candidate. So our technology platform, as I mentioned, has the comprised of small molecules and RNA technologies directly at the SOD1 protein. And we have a very strong patent position. So we have the exclusive worldwide rights for CNS for both the small molecule technology licensed by from Southern Research and the RNA interference from biopharmaceuticals. And within those exclusive rights, we also have the rights to any new IP deriving from these two platforms. The smart tissue, the smart molecules have been issued patents to treat ALS and memory and learning impairment in neurodegenerative disorders. And they were issued in Taiwan and in US and in Taiwan. And the RNA interference technology has US and international patent protection through 2029. And we have new patent that has been issued to file, to which we have access in 2015. And the current, integrating synergistic technology, I don't know if you could see my mouse, my pointer. Synergistic technology has been just granted patent protection in combination with Thera, so Phio and Thera together in 2021. So we have basically positioned our technology for long-term sustainability. Our smart technology comprises of 18 small molecule library with 11 neuron selective compounds, and we will see later why that is important. And we also have more than 120 second generation molecules with potential composition of matter and IP protection. Out of this program that we run in collaboration with Southern Research, funded by the Muscular Dystrophy Association, we have identified two very promising second generation compounds that have oral bioavailability. As I mentioned, unfortunately, our data have not been disclosed, and we're still, keeping them confidential. But if anybody is interested in learning more about our medicinal chemistry program, I'd be happy to liaise with them separately. And then our SOD1 RNA interference, we have established an engine for production of target-specific RNA or oligonucleotides, and specifically, we are now working in optimize a number of variants that to replicate the data that we already have on the SOD1 G93A animal model. So the mechanism of action of our technology impacts multiple pathogenic targets. They are common to NDD, and therefore, positioning our technology for development in multiple therapeutic areas and providing good market differentiation. So the therapeutic objective of both our technologies platform is to keep the patient functional, and especially to minimize, and manageable, and manage disability, and increase life expectancy. So the way we believe that our technology delivers this, this objective is through their small molecule that maintain brain cell viability and regenerate neuronal network, and the RNA interference that decrease and stop damaged proteins production. So both treatments will induce potential disease-modifying effect, and as I mentioned, are uniquely positioned for either be complementary or synergistic. And what's important about our approach and our technology is that it might cover the target diseases across all clinical presentation, and especially in ALS. So just to talk a little bit about our, our technology, transcription deficit are associated with key ALS pathogenic mechanism. They're involved in motor neuron degeneration, including neurotrophin deficit, mitochondrial malady, and protein transcription deficit or misfolding damage, as we heard from many of the presenters today. So I believe that there's consensus that effective therapeutics must aim at controlling simultaneously key pathogenic causes, leveraging brain pathway that engage compensatory mechanism and promote regenerative processes. So our technology is positioned, synergically to fight the mitochondrial damage and the neuronal death, as well as the defective proteins, by blocking further progression of the disease. And I think okay, here we go. So our smart molecules, especially our clinical candidate, engage a pathway, the nuclear factor kappa B, known to upregulate mechanism key to cell viability, protein function, and neuronal network integrity. Our molecules activate NF-kappa B by nuclear translocation of the P65 subunit in brain cells. So we also, as I mentioned earlier, we have 11 compounds there neuronal, in which this translocation occurs exclusively in neuron, including our clinical candidate. And the importance of this activation is that these compounds translocate P65 in neurons without triggering inflammatory effects from cytokine release, and promote transcription induction mechanism that regulate cellular correlates of long-term potentiation, long-term depression, hence leading to neuroprotective, and neurotrophic effect, but also increase release of neurotrophins, and therefore they modulate neurotrophic mediated effect of neuroregeneration in the neurite network. Our compounds, by activating NF-kappa B P65, also promote increased expression of the manganese superoxide dismutase, known also as SO2, resulting in reduction of reactive oxidative species and improved mitochondrial viability. When we go to and look at oops, I can't, looks like I can't do this anymore, can't advance anymore, sorry. Okay, so when we look at the defective proteins, and therefore we're looking at more comprehensive pathogenic effect, our small molecules also promote increase of intracellular expression of chaperones, and therefore promote protein, SOD1 protein folding, increasing functionality. I will have a next slide showing a little bit in more details. And on the other hand, our RNA interference technology, with obviously targeting SOD1, is positioned to mitigate the expression of defective proteins through protein silencing, and therefore, impacting the protein-related pathogenesis of ALS. So as you can see from here, our proposition, our therapeutic proposition, it really provides a very comprehensive targeting of the ALS pathogenic, and because upregulation of neurotrophic and neuroprotective and compensatory mechanisms triggered by NF-kappa B is common to a number of neurodegenerative diseases, we believe that this synergic approach can be effective in diseases like traumatic brain injury and Alzheimer's disease. So next we have, okay, so the biomarker, as I mentioned, we have a biomarker program that is coupled. And specifically, we wanted to leverage the ability of our clinical candidate to increase expression of chaperones proteins and DNAs. And we will partner with Dr. Cashman, British Columbia in Canada, to utilize some of the new antibodies against the misfolded SOD1 protein to couple with our clinical candidates in our preclinical and clinical trials. And in addition, we're also looking at our library of RNA variants to identify the potential protein-based biomarkers. So, and I trust you could see my slides. Okay, so our pipeline, we are focusing our lead program to advance in ALS. As I mentioned, we have a small molecule clinical candidate that we have identified, SYN1. The compound is at a stage where we feel comfortable that we could move to an IND towards the mid-2023 or end of 2023, and then start a phase 1, phase 2B, phase 1B, phase 2A trials shortly thereafter, with potential, viability for return on investment at the end of the phase 1 study. We also have right now a lead selection ongoing for our RNA interference program. And we also have a discovery program, as I mentioned, with the compounds that we have identified from our medicinal chemistry program. And we believe that we can position these compounds for discovery in traumatic brain injury and Alzheimer's disease. So our small molecule, our SMART technology, as I mentioned, we have completed the discovery and key preclinical program. So we have established the structure-activity relationship, generating additional compounds. We have identified the mechanism and target of effect. We have established the activity and efficacy in the ALS symptomatic animal models. Actually, we tested 2 models. And we have profiled the PK, the in vitro pharmacology, and in vivo bioanalytic studies. And we established acute and long-term safety. And we have shown that in 2 animal models, daily treatment for over 6 months with our small molecules did not have present any evident toxicity. And then from a CMC manufacturing standpoint, we have pretty much defined our synthesis and lab scale compounds production. We have optimized our formulation activities, and to that, that's confidential, but there's some very promising development there. And our API has a very low cost for production. So we have tested our compounds in SOD1 1992 animals at 96 doses.
So these are our projected revenues or pro formas. These are based.
Thank you. And we have maintained functionality and delayed progression of gait and posture impairment. Especially, we have shown that we use a significant weight loss reduction, preserving muscle strength by over 50%. And we also have, and I think this is the most important part of our trial, our animal trial, that we have delayed the progression to more severe stages of the disease by keeping the animals at the milder stages, namely the NS1 and NS2. And we have prolonged survival in this animal model. I wanted to point out that these studies were done in a blinded fashion. So we have prolonged survival by 50%, and I'm showing here only the male animals. With the females at the same, profile. So with our SOD RNA interference technology, we have optimized our cell delivery mechanisms. We have, with some of the variants already proven efficacy in the SOD1 G93 animal, and we have identified the variants that we're now exploring as leads for the SOD1 protein. And for CMC, we have performed, backbone chemistry on our library, and we have established a dose range for our variants. And now, right now, we're doing some chemistry optimization to start our validation in the animal, in the ALS animal model. So from a funding standpoint, we are going to need, as a seed, we estimate that we need about $5 million to progress both programs in parallel. The SMART technology is more advanced, so we anticipate needing approximately between 0.5 and 0.7 millions to complete the pre-IND enabling activity, and then move to the program, the IND program in non-mice and non-rat animals. And then for the RNA, we need approximately between $300,000 and $400,000 to complete our lead variant optimization, and then approximately $700,000 for the animal studies, and then the additional money to the IND. And we believe that, in truth, we would be able to establish a good value inflection by the, as I mentioned, by the second half of 2023. When we're positioned to file the IND, obviously for orphan drug for ALS. And then we believe that, once we are able to start our phase 1b/2a study, we might be able to position the company for liquidity event in second quarter of 2025. So far we've been funded by non-dilutive funds. So we had, as I mentioned, in collaboration with Southern Research, when we licensed the technology from them, $300,000 from the Muscular Dystrophy Association that has allowed Southern Research to conduct the medicinal chemistry program. We also have been granted a grant from the Flemish Innovation Agency for Innovation Entrepreneurship in Belgium. And then the ASLA grant that we received in 2017 has allowed us to test the animals in another animal model, but most importantly, to identify SYN1 as our clinical candidate. So we're grateful to the ALS Association for funding us. These are some of our collaboration. Our team, myself, I am a neurologist, a still practicing neurologist. I'm a specialist in neuromuscular disorders, and I still treat patients with ALS and rare neuromuscular disorders. Mr. Maestre is our co-founder and chief operating officer. Dr. Vincent is our head of preclinical and CMC, and obviously we have our general counsel. And we present with a significant expertise. We have over 25 years of pharmaceutical and drug development experience. But we are looking for partners that can help us identify talent, execute the strategy, and achieve target inflection milestones. So I leave you with this, slide.
We're gonna have to end here because we're several minutes over now. If you like
so
one.
This was the last slide that showed that we do have we're positioned promisingly positioned for disease modifying therapeutic approach to ALS. Thank you.
Great, thank you. So I think in the interest of time, we're going to need to move along. But as Dr. Favit Van Pelt mentioned, people can contact her for additional information if you want to find out more about those molecules. And I'm sure a CDA would probably be very helpful in getting more information. So now I'd like to introduce Dr. AJ Arohali, who's the founder of Rhea Therapeutics. We'll talk about a combination therapy approach to treating ALS.
Can you hear me?
I can hear you. It's great.
So, I'm just trying to change the slide.
If you click on the slide, yep, got it.
Perfect. Okay, great. So thank you very much for attending and watching the presentation. My name is Anjan Arali, or AJ for short, and I'm the co-founder of Raya Therapeutic, a company focused on developing multiple clinical stage compounds to treat ALS both on its own and in combination. So the company is actually named after my father, who unfortunately died of ALS in 2011. It's based in Montreal, Canada, with a presence in Belgium. It's currently privately held and self-financed, and it's, as you can imagine, virtual, with a reliance on expert external consultants. And of course, we will add full-time employees as the company matures and raises additional capital. We're focused on developing drugs to treat all ALS, including sporadic. And our 5-year mission is to have clinical proof of concept data in pivotal trials with at least 5 different drugs with at least 5 different mechanisms of action. So it's pretty ambitious. And we're currently raising seed round to help with regulatory filings, manufacturing clinical supplies, as well as orphan drug filings and preclinical development. So again, we'll talk about that in greater detail. So I'm not going to cover the disease because I think it's been well covered thus far today. What I will talk about is just motor neuron degeneration in general. Basically, there are multiple disease mechanisms that contribute to motor neuron degeneration in ALS. And Raya is focusing on targeting those mechanisms that contribute to the loss of motor neurons first. And so the image that you're seeing here depicts a number of disease mechanisms that contribute to motor neuron loss, and I believe some of them have actually been talked about earlier today from various companies. And so Raya's compounds are expected to target either directly or indirectly about 50% of the mechanisms of action that you see here. So let's talk a little bit about all the molecules under consideration. So Raya has currently licensed one compound, and that is RT-1972. So we have a worldwide license for all indications, and I'll get back to that in a second. The other 4 compounds are currently either under term sheets or exclusive negotiation agreements. Each of the compounds have clinical data. These compounds were shelved by biotechs or pharma companies. They have anywhere between 200 to almost 1,000 patients worth of data for each one. And they were shelved not for toxicity reasons, but for a variety of other reasons. None of them were originally developed for ALS. And so if we look at each of the compounds in a little more detail, you can see the first column actually provides information on the level of support at the molecule level. And when we say support in terms of preclinical support, we're talking about SOD1 data or primary neuronal cultures. And so that's the kind of preclinical support. So you've got 3 compounds that have direct support for that molecule. A couple of the compounds don't have preclinical data for the molecule, but they have it for the target. There's plenty of that. And then basically all of them also have potential in other indications, neurodegenerative indications. And so they're all orally bioavailable. They're all small molecule. The CMC is somewhat straightforward and not too expensive. They all penetrate the blood-brain barrier. And so just getting back to RT-1972, which is the one compound that we have already licensed, it also has potential in a very rare pediatric neurodegenerative disease. And the company also received rare pediatric designation from the FDA for that particular indication. And again, I'll talk about that later. So there's actually a lot of value in shelved compounds. And it's actually had a bit of a resurgence in the last year or so. And I put on this slide a number of different examples of shelved compounds that have been resurrected, if you will. And so you can see some of them go back as far as 2006 in terms of when they entered the clinic. But also the bottom right corner is a company that recently raised almost $200 million for a compound that had been shelved a few years ago, and it was in development in cardiovascular disease. So this fundraise was perhaps the largest Series A raise in the history of European biotech. So it was certainly something notable. In terms of IP, of the 5 compounds, 3 of them already have IP. However, all of them will be able to benefit from orphan drug designation. And of course, that comes with anywhere between 7 and 10 years of exclusivity data. As well, each of the 5 compounds will qualify for a new chemical entity or new active substance designation, and that comes with exclusivity and data protection as well. And 1972, because of its potential in the rare pediatric disease, will benefit from an additional 2 years of data protection on top of the orphan drug designation here in the US. And so for those of you that don't know, how these data protection mechanisms work, they effectively act like patents and provide market exclusivity for the holder. So there are quite a few KOLs that have made comments about the heterogeneous nature of this disease. And actually some of the presenters before me have made the same comment. And I put on this slide a couple of quotes, one from Dr. Hyman Patterson and another from Dr. Rosenfeld, both of whom are very well known in ALS circles. And they talk about the heterogeneous nature of the disease. And so one of the things that Riad has done is actually we have gone to the largest ALS clinical consortium in Europe, and they're called TRICALS. And we asked them to look at all of these 5 compounds under confidentiality. And they did. They did a fair amount of diligence on these compounds. And we asked them what they thought, and they were actually extremely supportive, and they provided us with a written endorsement, call it a letter of support. And I just want to point out the last sentence here in red. In that letter of support, it says the ability to test multiple drugs on their own and in combination will be an unprecedented effort in the history of ALS research. So why is this unique? Why is this combination strategy unique? Well, if you look back at the history of ALS research, it has been primarily one drug, one company. You have not seen, with perhaps one exception being Biogen and Ionis, you have not seen a company with a pipeline of compounds or a focus to treat ALS, and that's really what makes the company unique. And as part of that, so how are we going to move the compounds forward in terms of testing them for combinations? So the first thing that we're going to do is we're going to test each of the compounds on their own in iPSC motor neurons that were taken from ALS patients, both sporadic and familial. And this work is actually going to be done at the Montreal Neurological Institute. So we will test these compounds on their own, and that will provide effectively benchmarking data, performance data. And then we will take each compound on its own, and then we will test them in combination in a simple 1+1 matrix, as you can see here. And then we will take the top combinations in terms of performance. It's a survival assay. And we're then going to combine it with the standard of care, which is edaravone and riluzole. And then we're going to look at the performance with the standards of care. And then we're going to take the best combinations, and it's effectively a filtering process. And then we're going to move towards either going to an animal model or directly into phase 1B. We've already talked with a couple of KOLs about this. They're pretty comfortable, given all the safety data that is already available with these compounds, that we can in fact move directly into a small phase 1B trial. And as I mentioned, this is a fairly unique approach. We are not really aware of any biotechs that that have a comprehensive approach to combination therapy in ALS. So we're certainly pretty excited about it. We have talked about this approach with numerous ALS clinicians, and they too are pretty excited and hopeful about it. Okay. And I know this has been mentioned before, but it's certainly worth repeating. ALS does provide for a streamlined drug development pathway. It's possible to get a waiver on chronic tox studies until after phase 1 on a case-by-case basis. Carcinogenicity studies are typically only required as a post-approval requirement for ALS companies. And importantly, there's a possibility to file an NDA, for example, just based on the positive single phase 3 trial. And in some cases, we've seen, I guess, really small trials, not even perhaps could be designated as phase 3. And I think what's really important lately is the creation of 2 new platform trials, the Healy trial in the US and the TRICALS trial in Europe. Both have been developed to, created to develop ALS drugs more rapidly through pivotal trials. And with both the FDA and EMA have indicated in the past that they're willing to be flexible and as accommodating as possible given the deadly nature of the disease. And actually, the FDA made this statement just last month. I've put it on the screen here. I'm not going to read it out, but effectively, it just reinforces their commitment to try and help companies advance therapies as quickly as possible. So if we take a step back and look at how this is all going to work, what would happen is an ALS patient would get enrolled into the platform trial, and we're planning to do this in collaboration with TRICALS, and they would get enrolled into one of the 4 arms, and we would have a 5th arm probably starting anywhere between 6 to 12 months later, and then they would get randomized again into either the drug arm or the placebo arm. And if you remember, there's a whole lot of combination work that's happening, preclinical, and then a phase 1b trial. So after the phase 1b trial, ideally, we're going to have at least 1 candidate from the phase 1b trial that shows some potential as a combination therapy, and that will get added to the combination, that combination program will get added to the platform trial at a later time period. Okay. So, if we look again back at the history of drug development in ALS, it's littered with failures. We've only got 2 drugs approved in the US and 1 in Europe. And so with that in mind, I created this company. I founded this company with the thought of trying to build it so that it could withstand a number of clinical development delays or even failures and still survive. And so when we think about having 5 drugs and testing them in ALS, you're talking about 5 separate shots on goal, an additional shot on goal for a pediatric indication, and possibly, maybe even 2 shots on goal for combination therapies. So, and when I say overtime, what I mean by that is, again, going back to, you only need 1 positive phase 3 trial to move forward with filing. And so really any one of these, for example, 8 shots on goal, only 1 positive phase 3 would be necessary for us to move forward with filing. So just taking a look at the corporate timelines, we have just started our preclinical work at the Montreal Neurological Institute. This literally started just about a month and a half ago. And the entire preclinical program is actually going to be supported in part by an $800,000 US grant that we just finalized this week. So we're pretty happy with that. And so this will actually offset a majority of our preclinical budget. And so, like I said, that work has already started. We have started some in vitro work on that pediatric neurodegenerative disease as well. We have talked, just a word on that, we have talked with 3 of the top KOLs in that neurodegenerative pediatric indication. And they've indicated that the development pathway is relatively straightforward. And they're also pretty excited with the compound that we are hoping to move forward for that disease. We're currently raising a seed round. We hope to have a Series A investor. It's actually a corporate or a strategic investor by the end of the summer. We have negotiations ongoing. And we're going to be raising a large Series B of anywhere between $60 to $80 million, either at the end of this year or early next year. I've been building this company and thinking about it for the last 2 years. And over the course of that 24 months, I have actually approached probably 24 pharma companies and large biotech companies about the program, about the strategy. And, if all goes well, and that's certainly a big if in biotech, but if all goes well, we hope to have by the end of next year 4 of the compounds enrolled in the pivotal platform trial. By the middle of 2023, we actually hope to have 5 drugs enrolled in 6 phase 3 trials. And so I can tell you, based on all my discussions with these strategics, is that a company that has 5 drugs in phase 3 in six phase 3 trials by the middle of 2023 would be of a high degree of interest. So I think there's multiple potential inflection points along the way, but certainly there is definitely going to be a potential for an exit as soon as about 24 months and perhaps up to 36 months from now. Okay. I know there's a lot to absorb when a startup company is talking about developing 5 drugs in phase 3, but I just want to step back for 1 second and say that if we just take 1972, which is the compound that we have developed, and just look at that as a standalone, the company is a pretty compelling investment opportunity. There's a substantial amount of literature and KOL support for this target and this compound. There's data in 4 phase 1s and 1 phase 2 in over 200 patients. As mentioned, there is the potential in at least 1, possibly even up to 3 rare pediatric diseases. We've got RPD. The development pathway is relatively straightforward. And for those of you that don't know, when you have a rare pediatric designation, it actually qualifies you for something called a priority review voucher. And this is something that a company can actually sell to a large pharma, and it provides the large pharma with an expedited review process when they give it to the FDA when they filed their NDA. And so there's actually a market for these priority review vouchers, and the last 4 have sold for about 100 million apiece. So there's certainly a lot of value there in the priority review voucher. So a little bit about the team, about myself. Like I said, I'm currently working as a venture partner for a venture fund up in Montreal, although I live in the Princeton, New Jersey area. I've got a lot of big pharma and biotech experience. I have quite an extensive amount of nonprofit experience in the ALS space, both with the MDA, Wings Over Wall Street in New York, Prize for Life in Boston in the past, and with the Packard Center for ALS Research at Johns Hopkins. And Dr. Angela Genge is a very well-known ALS clinician. She's based in Montreal, and she's the executive director of the clinical research unit there. And my partner in crime throughout this entire process has been Dr. Kim Statz. She has published extensively in ALS and has actually been educated in 4 different countries, which is rather unique. And this is my last slide. I'm going to end here. And basically everything here has been covered. I just wanted to mention that this is a mission-focused company. And it's an ambitious project, but it is realistic. And I think this is going to be the direction that is needed to find, to be able to turn a deadly disease into a chronic disease and ultimately a cure. And as the famous expression goes, it takes a village. And we're a highly collaborative company, and we're actually already engaging in a number of different discussions with a variety of companies, pharma, biotechs, AI companies, to again, try and work together to find a way. Thank you very much.
All right, thank you. I think we're not going to have time for questions because we're really running a little bit late, but I do want to commend you for taking on this combinatorial approach, because I think it's sorely needed. And we've seen in so many other fields where combinations of drugs are effective and cocktails are commonplace, and it just hasn't really been done in ALS. So it's a really interesting approach.
Thank you.
All right, thank you very much. So we're going to go to our next speaker. It's Dr. David Innist of Origent Data Sciences. I will turn it over to you now, Dr. Innist.
Thank you. We got our start in 2012 when statisticians at our parent company, Centrana, a Washington, D.C.-based marketing analytics firm, participated in the Dream Phil Bowen ALS prize prediction, prize for life challenge to develop an ALS disease prediction model. This was published in Nature Biotechnology. Since that time, we've become leading experts in neurodegenerative clinical trial optimization using predictive analytics. We have a lean core team of 5 FTEs, and we're a very asset-light business. We have a strong reputation among ALS key opinion leaders, and we're building our reputation in Parkinson's, Huntington's, and Alzheimer's. We got our start with a $1.45 million angel investment, and we've grown our revenues on a service basis to about $1.4 to $1.5 million. We've had a number of NIH SBIR grants, including a phase 2 grant, and a number of ALS Association grants as well. And here you see a number of the clients that we've had over the years. What we're doing is pivoting from a service business to a business that can rescue drugs and benefit directly from revenues from sales of the drugs. The idea is that we will increase our growth by doing this pivot. So here's our team. I postdoced at the National Institutes of Health in Bethesda, Maryland. I went to work at the first gene therapy company, Genetic Therapy Incorporated, which was acquired by Novartis. So I've had the biotech and also the large pharma experience. My job was, as a group leader, was to translate drugs from gene therapies from the preclinical lab into early clinical testing. Danielle Bullier is our Chief Data Science Officer. She's a MPH biostatistician with over 10 years experience analyzing clinical trials. Albert Taylor is a PhD neuroscientist who similarly has a wide experience in clinical trial biostatistics. We have a strong board of directors, including Fabian Rosado and Mike Kiemer, who incubated Origent in its early days, and Sid Mansour is the CEO of our parent company. The problem that we're addressing is the $22 billion that are written off each year due to failed drug programs. And the leading failure in phase 3 trials is inability to show efficacy. And as we know, CNS clinical trials are especially susceptible to false negative results. And we see as a root cause is heterogeneity in patient disease progression. Here's the spaghetti plot in ALS. On the y-axis, we have a revised functional score, and with time going on the x-axis. And you see, really, these are actually patients who participated in ALS clinical trials. You see some patients are very slow progressors, other patients decline remarkably rapidly, and then some patients decline at what's widely seen as the average rate of a point per month. This wide range in different disease progressions causes a lot of noise in the statistical analysis and making it very difficult to detect any therapeutic effects. We have developed a number and validated a number of ALS disease progression models. The ones that are most useful in clinical trials are the total functional score, percent expected vital capacity, the survival model, and the time to 50% expected vital capacity. But that's just the place where we start. We take those models and develop tools from them that help us increase the clinical trial efficiency. And this includes the development of virtual control arms, enriching patients for patients who will progress at a predetermined possible rate. But probably the key tool that we have is the ability to stratify patients. And this, in a publication that we had with James Ferry at MGH, when we randomize and do covariate adjustment using the predictions, we can boost power of a study by as much as 10% or lower the sample size on the order of 20%. These are all tools that we use to increase the clinical trial efficiency. But the tool that I want to talk about today, and the one that we see as the most valuable one, is a novel method of subgroup analysis that we call detectable effect cluster analysis. And what it allows us to do, and the application that I want to talk the most about, is it allows us to rescue failed trials so that we discover patient subgroups that could have formed the basis for a successful clinical trial. And so what we do is use machine learning to rejuvenate drug programs that failed to show efficacy in late-stage clinical trials. The drug project has been shelved and has been lying dormant, and we're rejuvenating and rescuing it in ALS. So we've developed a platform that has a rough screen where we go to the published literature and various databases such as Citeline and clinicaltrials.gov, identify trials for possible in-licensing, and then we go to the owners of the license and negotiate terms, obtain as much information about the drug program as they're willing to share at this point. But the key is that we do get the clinical trial data sets, which we put through DEC analysis, the detectable effect cluster analysis. And this helps us find those hotspots. We identify the thresholds of predicted disease progression for a particular drug where patients formed a positive subgroup, and then propose a clinical trial plan that we then package and out-license to either other drug developers, or we complete the development of the drug with we do a co-development with the originator. So what we do is de-risk the drug candidates by identifying patient subgroups with decreased statistical noise and demonstrated efficacy in failed late-stage trials, and then we use those patients to enroll the subsequent late-phase registration trials. Again, what we're doing is rescuing drugs in the same indication, in ALS in this case. And so here's a deeper look into the heart of our platform, DEC analysis. This is a patent pending method and system, a novel way of subgroup analysis. We start with a training database. In the case of ALS, we use the PROACT ALS database. We have a number of developed pre-existing models. And then we run trial data through the model, develop predictions for all the patients in the trial using just their baseline information. And then we put them in order from the slowest progressors to the fastest progressors. And then we form very small subgroups of the most similar patients, and then systematically expand the size of each subgroup until we get to the full analysis set. And this is what a matrix of all those subgroups looks like. Each of these squares represents one of the subgroups in the analysis. The smallest squares here are along the diagonal, and the full analysis set is the tippy-top square in the upper left corner. This is a failed trial where there was no hotspot. There's just some random noise in very small trials. This is what we're looking for. A failed trial, the full analysis set in the upper left corner did not have a p less than 0.05, but we find this broad region of activity of patients where we did find a p less than 0.05. And so we've validated this approach both in the commercial and scientific setting. We work with MT Pharma on their Edaravone phase 3 trial, which, as several speakers have already mentioned, has recently, in the last few years, been registered for ALS. We worked with their failed trials to see if we could identify a broader group of patients who seem to respond with a detectable effect to Edaravone. I'm happy to say that this study is now in press, and we identified a fairly broad group of patients who seem to form a detectable cluster. And then in 2019, we closed our first drug rescue deal with a company that had a failed trial. This is a successful kind of validation of the business approach in that Neuraltis was willing to trade equity for our results. Unfortunately, we did not go forward with that because we did not find a hotspot. But then just recently, we completed an NIH SBIR grant with 2 studies, ceftriaxone ALS and topiramate ALS. I spoke with KOLs in the field, folks who had been PIs in these studies, and they both told me that the ceftriaxone study, they thought some of their patients responded positively, and they both agreed that the patients did not respond to topiramate. So we analyzed these 2 studies using the topiramate as a negative control. We confirmed that, so we don't randomly find hotspots. And then the ceftriaxone ALS analysis is this on the right. And what you see overall, the upper left corner has a p greater than 0.05, but we selected this subgroup here that appeared to have the highest treatment effect, the lowest variance, the highest power, and we simulated the power. So we threw a number of additional metrics at it, and we're able to select that particular set of patients as the ones that we would suggest going forward if this were a viable program. So where do we get the drugs from? We have a number of databases we look through with a number of criteria for a primary screen. In Citeline, we were able to find 1,700 failed programs. Of those, our initial targets are ALS, Huntington's, and Parkinson's. And from these, we have distilled down to 118 programs that we're actively screening. We have begun discussing a number of these programs with the current holders of the licenses and are deep into negotiations for these drugs, for some of the drugs. What's this going to cost us in terms of upfront payments? At the low end, it's a $30,000 upfront. At the high end, it's $11 million upfront. Most deals, upfront payments are in the $1 to $5 million, and this is the range that we've modeled for our pro formas. Competition, we look at it twofold. On the right, these are companies that are using AI to repurpose and also to find common molecular pathways. And going back to early development, we see that as a way, pretty much of once a drug is identified, it enters a pretty traditional drug development course of 10 to 15 years down the road. We're focusing on late phase trials and getting them immediately back into late phase clinical testing in the original disease. So we're rescuing it rather than repurposing trials. And what we have are a number of approaches. There's a technological, biological, or a digital approach. We're in the digital end of it. The biological approaches tend to find common biomarkers between subgroups. And so in our upper right corner, where we have a digital biotech approach, there's a company, Roivant, which has become quite a large pharma company, but very little other competition in that area. And so we've created strong barriers to entry. We have a disease modeling foundation, and on that we have a number of of strong pillars, including scientific reputation among KOLs. We enjoy first mover advantage in mining for the drugs. And we also have filed a patent protection on the method and system. But then in addition, when we find the thresholds for individual drugs, we will file the drug combination with the thresholds for additional, for a new patent protection on the drug. So our business model, as I mentioned, we negotiate terms, obtain the clinical trial data sets. If it's a drug we want to go after, we finalize terms, and then we either package up the de-risked asset for out-licensing, or we work with the original developer in a co-development type of an arrangement. For the out-licensing, we plan to out-license at 3 times our upfront costs. So that's where a lot of our initial revenue will come from. And here are our pro formas. You see 2020, this is our last full year as a service company. We're pivoting this year to become a drug rescue company in neurodegenerative diseases, starting with ALS. And you see our first revenues from the out-licensing will come in 2022. And then by 2024, we should have $40 to $50 million in revenue. We see this as a valuation of conservatively threefold. This would be about $150 million valuation in 2024. And we see our current valuation at about 1/6 of that. So any investment today will yield a 6-fold return. What we're seeking is $8.5 million to license our first drug programs over the next 1.5 to 3 years. That will include $3.5 million for operations. We will increase our scientific staff so that we can fully engage ALS as well as Huntington's, Parkinson's, and Alzheimer's disease. In addition, we'll need increased business development staff. We want to hire 2 FTEs who will handle the in-licensing and out-licensing functions. And then we'll also need $5 million for our first upfront payments, which we will then turn into out-licensing deals. We're seeking a SAFE or a convertible debt instrument. And we're also open to a price round if that's the preferred vehicle. So with that, I want to close and take any questions.
Okay, thank you very much. That's an interesting model. Can I ask, do you typically expect to end up with a narrower label in this?
That's a really good question. And if you look at the MT Pharma experience, I think the answer would be that the FDA gave MT Pharma a license for all of ALS based on a registration trial that was fairly narrow in its enrollment. So that's something that we would certainly discuss with the FDA. And ultimately that might be a risk, but then we've also rescued a drug and brought it to market and gotten it into patients' hands as well. And if it's a narrow group of patients, that's fine. We'll keep going after additional drugs for different types of patients.
Okay. Let me see if there's a question. Would you anticipate this model would work in a genetic ASO context or primarily small molecule biologic?
Yeah, I think this model will work in all settings. And so we don't have any reason to believe that it wouldn't work, say, in a rapidly progressing ALS genetic patients. So no, we believe that the modeling works in all patient types.
Great.
I should say that it's been validated both in with clinical trial data sets as well as clinic data sets that have not gone any trial selection.
Okay. All right, thank you very much. I think we're going to move on now.
Great, thank you.
Thank you. Our next to last speaker, and that's Dr. Nader Yacoubi, who will talk about a novel non-invasive neuromodulation approach to treatment of ALS using neuronal hyperexcitability suppression from Pathmaker Neurosystems.
Great, thank you, Sharon. Very pleased to be here today and be able to tell you about what we're doing at Pathmaker Neurosystems. My name is Nader Yacoubi, and I'm president, CEO, and co-founder of the company. Compared to some of the interesting pharmacological and gene therapy approaches we've been hearing about today, I'm going to be talking about something different based on neuromodulation. So we wanted to start by introducing our investigational non-invasive neuromodulation device, MyoRegulator. This will be the world's first device for the non-invasive treatment of muscle spasticity. This is a microprocessor-controlled device that delivers our proprietary double stim technology using disposable single-use electrodes. It has a rechargeable battery and is portable. We've been showing the device at several conferences over the last year and a half. We expect to have CE mark clearance in the next 12 months and to begin European sales actually next year. And we're working with the contract manufacturer to actually produce the device. Our platform is based on our proprietary neuronal hyperexcitability suppression technology. This technology allows us to non-invasively modulate the excitability of spinal motor neurons. It combines spinal stimulation done using anodal TSDCS, which is transspinal direct current stimulation, together with peripheral stimulation done using direct current stimulation. And when you combine them together, we can actually suppress the firing of hyperexcitable spinal motor neurons by driving current across the spinal cord and down the afflicted limb. The science has been published in leading journals, and we have over 40 issued patents protecting the technology. We've now completed 2 clinical trials with the MyoRegulator device, and we're launching our US multi-center pivotal trial next month, working with Spalding here in Boston. And this trial is fully funded by NIH on the basis of a $5 million grant. We've so far treated approximately 70 patients. We've seen effective reduction of spasticity, and the treatment has had, it's been safe with no significant adverse events reported. Our company has an office and employees based at the Paris Brain Institute at PTA Salpêtrière, which is where Professor Charcot actually first described ALS. Our company did not start out focused on ALS, but the science has now led us directly to ALS based on the mechanisms by which our technology works. As we've been learning about the disease, we think it's relevant that, first of all, you see spasticity in about 80% of ALS patients. There are numerous publications talking about motor neuron hyperexcitability appearing as an intrinsic part of the ALS disease process. And of course, there's a massive loss of spinal motor neurons that lead to an inability to control muscles and functions. So we have a technology that allows us to non-invasively suppress motor neuron hyperexcitability. And we think this provides an entirely new therapeutic strategy for treating ALS. Over the last 2 years, we've been working to put together a robust preclinical data package in SOD1 G93A mice. And this includes data on electrophysiology, motor function, biochemistry, and survival studies. Our scientific founder, Professor Zaghloul Ahmed, he's established a colony of SOD1 G93A mice at his institution at CUNY CSI in New York. These mice developed tremor, paralysis, and premature death, and are a gold standard preclinical model of ALS. On the left, you see the multi-site DCS setup that we use to stimulate the animals. And you see spinal electrodes positioned along the spinal column and peripheral electrodes positioned along the sciatic nerve. And there is a abdominal return electrode, which is a cathode. On the right, you see the mouse positioned in a mouse holder. And this allows us to both apply stimulation as well as elicit reflexes and measure various parameters. These animals develop disease in Dr. Ahmed's lab typically at day 113. And after disease onset, what you see is increased muscle activity. That's apparent on the left. Once we turn on anodal multi-site DCS, you see immediate reduction in spike amplitude. When you treat the animals with 3 consecutive sessions of stimulation done 1 hour a day, you see that after 3 stimulations, you have a decreased frequency and decreased amplitude seen on the EMG trace. So we have methods that allow us to look at tremors in these ALS mice as well. And we use both a microgoniometer as well as a force transducer. And on the left in the shaded area, you see the tremors that exist in the animals prior to stimulation. And on the right, when stimulation is turned on, you see that there's a significant decrease in the tremor that is witnessed. We have 2 videos that I'd like to just show just to give a more visual sense of this. The first video shows a twitch tremor in one of the SOD1 G93A mice. The next video shows during stimulation. So beyond looking at tremor, we've also been doing motor function studies and done grid walking. What we wanted to show here is just, again, some video that shows the difference between mice that have been stimulated and mice that have not been stimulated. Here, these are siblings. So these are mice born in the same litter to the same mother. And on the left, this is cohort 1. One of the animals has a black tail and did not receive stimulation. Then the other 2 animals received 8 days of stimulation, 60 minutes per day. And you see the 2 stimulated mice also have some scars on their back. So what we saw is that the stimulated animals are able to, they have better motor function than the unstimulated animals. And here on the right is a second cohort done more recently. Here, the unshaven mice have not been stimulated, and the 2 mice that have been shaved have been stimulated with 5 days of stimulation. Again, these are sibling studies, and what we're seeing is significantly improved motor function in the stimulated animals. So we published in 2019 a paper that showed the effects of stimulation on NKCC1. And NKCC1 is a sodium potassium chloride cotransporter found on spinal motor neurons, and it's involved in maintaining the chloride gradient. We found that in situations where there is disease or injury to the spinal cord, there is overexpression of NKCC1, which leads to high intracellular chloride, which then leads to GABA-A receptor activation becoming excitatory. And we found that when we treat the animals with our double stim technology, NKCC1 levels are suppressed, leading to normalization of the chloride gradient and GABA-A receptor activation, again, becoming inhibitory. So we've recently looked at NKCC1 in the SOD1 G93A mouse model. And what we saw here is that NKCC1 is overexpressed in carrier mice. And it's actually overexpressed about 5x. I'm not sure if this is coming across fully, but it's overexpressed 5x compared to non-carrier animals. And we found that when you stimulate, you get a reduction in NKCC1 levels. And that reduction is confined to the site of stimulation. In this case, the lumbar region is where the stimulation occurred, and the cervical region did not get stimulation. So all the data we've been generating over the last 2 years is actually going to be presented next month at NCALS. And beyond the few data slides I went through, we're going to be presenting more extensive biochemical data, looking at HSP70, SOD1, LC3B, also additional data on motor function, motor neuron counts, and survival studies. And just in the last few months, our survival studies have gotten to about 22 animals per group. So the next step for our ALS work really is to continue on with the mouse studies. We've been working with some experts here in Boston to develop our first clinical feasibility and safety study for ALS, and we're looking to get that underway. There's some modifications to the device that we could do to enable bilateral stimulation, and we'd like to get to that over the next year. And the clinical feasibility study will, of course, inform the design of our pivotal trials for this device. So on the IP side, we really pioneered the field of multi-site direct current stimulation. We have over 40 issued US and foreign patents. We own the space, we have freedom to operate, and we have exclusive rights to all of this IP. We have a veteran management team that has been instrumental to being able to move this technology forward. Our Chief Technology Officer has brought 7 medical devices to market. Our VP of Regulatory and Clinical Affairs used to work at FDA and was in charge of global regulatory at Boston Scientific. And our Chief IP Officer is a patent lawyer for the last 30 years. Our board of directors consists of the former president of Medtronic, the diabetes group there, and also the chief innovation officer at GE Healthcare, who have joined us. We are very grateful for some of the recognition we've received over the last few years, and we greatly appreciate the support that we've had. So in summary, our team has developed a novel, non-invasive approach to treating ALS using neuronal hyperexcitability suppression. We've characterized the effects at molecular, cellular, and functional levels. We have preclinical studies that have been completed that show improved motor function and increased survival in SOD1 G93 in mice. We've completed 2 clinical trials on this device showing that it's safe and effective in treating spasticity. And now we have designed a safety and feasibility trial for ALS in collaboration with ALS experts. This device has been labeled by FDA as a breakthrough device and is ready for immediate commencement of trials. So we appreciate the interest and support, and for those who might be interested to talk further and get to some of the data I alluded to, please reach out. My email is listed here. Thank you so much.
Thank you, Dr. Yaghoubi. And what are you looking for now from investors?
Yes, what we're working on is our Series B that takes the company to the point where we have CE mark clearance on the MyRegulator device, and we're selling product in Europe, and we have completed the ALS feasibility study that I talked about. At the same time, we have our US pivotal study getting underway next month, so that will also be moving forward.
Okay. Looks like we don't have any additional questions now. So I think people have your contact information and know how to get a hold of you. I think it's a nice compliment to hear a device in the mix, and after so many drugs, it certainly, the data are intriguing for sure.
Thank you, Sharon.
Okay, so we will move to our final speaker. It's Dr. Gino Cortopassi from Ixchel Pharma, who will talk about IX109, the first drug to rescue multiple animal models of mitochondrial orphan disease. And I believe, Dr. Cortopassi, you're using your own slides, right?
If that works, are you able to hear me, Sharon?
I am. I think we're just switching to your presentation or allowing you to put your presentation up.
Okay. I will go hit the share button right now. And you should now be seeing my screen.
We
And
do.
I will put it on presentation mode. And I'm guessing that you can see that.
We can. That looks great.
Okay.
Thanks.
Hello, everybody. The residual, those with strong heart and spirit that are here. I'm Gino Cortopassi. I'm a professor at the University of California, Davis. And I'm also CEO of Ixchel Pharma, where we're bringing mitochondrial therapeutics to life. So I will tell you, we are developing small molecule therapeutics for mitochondrial diseases. And I'll tell you some of the value points of Ixchel Pharma. We have our lead molecule has superior pharmacokinetics to billion-dollar marketed monomethyl fumarate prodrugs, Tecfidera, Vumerity, and Bafiertam. We uniquely target this very important mitochondrial transcriptional activator named TFAM as our mechanism of action to rescue lethality and mitochondrial function in animals, and we believe humans, with multiple mitochondrial diseases. And I'll show you data today for 2 different animal models of Friedreich's ataxia and also an animal model of Lee's syndrome, the most common mitochondrial disease. Our molecules have an accelerated regulatory path as they're based on a prodrugging technology. And because they deal with a pediatric disease, there's the opportunity for a priority review voucher. We have an experienced science, pharma, regulatory, and clinical team that can bring the drug to the clinic. And we are finalizing our preclinical data package, filing in Q4 of this year. And we are currently seeking investment and development partners. So these are the foundations of our value. This is my Ixchel Pharma email address, and that's our website. Guess I'm advancing this. Yeah, so there's just a little bit about Friedreich's. It's an orphan neurodegenerative and cardiodegenerative disease. Forgot to start the timer. It's triplet repeat disease, and it is caused by the suppression, not the elimination, of a single mitochondrial gene called frataxin. And this results, this puts kids in wheelchairs in their teen years, but the lethal part of the disease is the cardiomyopathy. So this illustrates where we came from and where we're going to. So we started off 11 years ago screening with the really catalytic support of MDA, I should say, on the university side, was screening, doing a repurposing screen of 1,600 medicines to find ones that were increasing mitochondrial protein like frataxin. And then as we worked down our screening funnel, found one in particular that was increasing mitochondrial proteins, including frataxin, in the brain. Then we carried out a clinical trial to see on-label with dimethyl fumarate Tecfidera in MS patients, so on-label in Italy, and found that it had these same properties to increase mitochondrial proteins, including frataxin, in people. But there were liabilities around that molecule listed here. And so we decided to transition to a new molecule and started that project 3 years ago, where we are, so this is the payload, the monomethyl fumarate that is increasing mitochondrial transcription and function. And this is the R group. When modified, this modifies the PK of the drug. And so working through several different R groups, we identified finally IXC109, which is our lead, which has better PK, lower GI tox, it's a better brain delivery, and it's also patented. And so this is just showing you the improved PK over these marketed $100 million and billion-dollar MMF prodrugs that have different R groups here. And that's showing you that we have about a twofold greater AUC, area under the curve, meaning that we can dose at a lower level with less GI side effects, et cetera, and get higher efficacy. These are some of the comp sales of related molecules with other R groups. So in Friedreich's, the thing that drives the whole disease is the reduction of this protein frataxin in the mitochondria. And what I'm showing you here is that our molecule dosed in mice is dose-dependently increasing mitochondrial frataxin and also mitochondrial cytochrome oxidase 4, but not tubulin. I remind you again that this is not just an artifact of dosing in mice with a related monomethyl fumarate releasing molecule in humans. Those mitochondrial proteins are also increased. And why is that important? That's important because in Friedreich's, everything is related to frataxin level. This is the amount of frataxin in blood regressed against the median age of onset. And what you'll see is that as you have, so normal people have about 20 nanograms per mil of frataxin, but Friedreich's patients have about 10% of that, or sometimes less, but the amount of frataxin is related to the age of onset, the clinical severity, and the age at death. So these patients will die much sooner than these patients. And so we are increasing this mitochondrial protein frataxin to a clinically meaningful extent, and when regressed against the age of onset would allow for more than a 10-year delay in the disease. So does the molecule work in the best mouse model of disease? Yes, it does. This is Rotarod study of the ataxia. And this is showing that the wild type mice stay on the Rotarod. The Friedreich's mice fall off sooner. An existing molecule that I told you about, dimethyl fumarate or Tecfidera, Biogen's Tecfidera, does not rescue them, but and it's delivered at the very same isomolar concentration of monomethyl fumarate, but ours significantly does. It's the first small molecule to approve ataxia and Friedreich's mouse models. Now this is a bit sciencey, but I'm doing fine on time, so I will tell you a little bit more about this. So this is in Friedreich's, as I told you before, the patients go into wheelchair because of the neurodegenerative properties of frataxin deficiency, but they die because of the cardiomyopathy. And it's a very classic, stereotypical cardiomyopathy in Friedreich's. So this is showing that our molecule, the IXC109, is increasing mitochondrial function, both complex 1 and complex 2 in the heart. It's showing that it's also doing so more than an isomolar dosed Tecfidera or DMF, Biogen's molecule. So it's also showing you that we're rescuing the mitochondrial enzyme aconitase, it's also showing you that we're rescuing frataxin itself. And then what does that mean for the heart? The stereotypical cardiomyopathy is the result of thickening of the ventricular walls. This is echocardiography of these mice. And it's just like what happens in the patients, that's thickening of the ventricular wall and then a shrinkage of the intraventricular diameter that is reducing all the cardiac output and cardiac function. So in the Friedreich's mice, they have a thicker ventricle. The Tecfidera does not reduce that thickening, but the 109 significantly does. And then the consequences of that is that there's a shrinkage of the intraventricular space in the Friedreich's mice. But when treated with our molecule, it goes back to wild type. And however, that does not happen with the DMF Tecfidera. And that's resulting in an increased stroke volume and increased cardiac output. So we're rescuing something that is lethal in Friedreich's patients. So here's another test of that. And this is now a different Friedreich's mouse model where their frataxin is very, very low in the heart. And so that, and it's like an end stage Friedreich's cardiomyopathy. And you see that these are the vehicle mice. These are the mice treated with the Tecfidera, and these are the mice treated with 40 milligrams per kilogram of the 109 molecule. They extend the life by about 13%, which is equivalent to 9 years, which is significant, clinically meaningful extension of lifespan. There are other drugs in the market that only extend lifespan by 5 months, and they're FDA approved. What we think is really special about this molecule is that it is in some way a general solution to mitochondrial disease. And why do I say that? The mechanism of action is that it increases the most important mitochondrial transcriptional activator, which is called TFAM. And other groups, completely separate from us, academic groups, like Neil Göran Larsson's have shown that if you overexpress TFAM, you rescue mitochondrial diseases of many different types caused by many different mutations. So how could that ever work? Well, it turns out that because mitochondrial proteins are in big complexes, and these are just a listing of the number of different mitochondrial diseases in the US, resulting in about 65,000 patients. Multiple mutations reduce mitochondrial function, but you can overcome that reduction in mitochondrial function by transcriptionally increasing the amount of mitochondrial genes and proteins. And this was a very surprising result. It works with different mitochondrial loss of function mutations, as Niels Göran showed here, and also even heteroplasmic mutations. And then what we showed here is that this molecule, the monomethyl fumarate-based molecule, is increasing the expression of all of these. And so potentially there's a total addressable market for an orphan disease of about 65,000. Now, that's all very nice to say. Is there any proof of that? We also treated Leigh syndrome. This is the most common mitochondrial disease and is very severe. Mice with Leigh syndrome, its features are shown here. And what we see is, once again, the Tecfidera does nothing over background, but the 109 molecule at a lower concentration than the Tecfidera is dose-dependently increasing their longevity to a clinically meaningful extent. So another point is that these this molecule, the 109, I'm not showing you the R group, but as it hits the gut, it's cleaved at this ester group to make monomethyl fumarate, which has been in humans for 1 million patient years. And it makes this vitamin-like molecule that has been in humans, an equivalent amount of humans. So these molecules have been in many, many, many humans. So we believe that that will allow us to have an accelerated regulatory response. And because we're dealing with 2 rare pediatric diseases, that getting a priority review voucher is a possibility. We do have a experienced team. I have spent my whole career for 30 years in developing understanding mitochondrial pathophysiology and developing mitochondrial therapeutics. And we have experienced business people, pharmacology people, regulatory people, and most importantly, clinical people that are very expert in the clinical development of molecules. So that's then just a restatement of the first slide. Our molecule has superior pharmacokinetics. It has a unique mechanism of action. It's got an accelerated regulatory path, and we have an experienced team to bring the drug to the clinic. We're finalizing our preclinical data package, and we are seeking investment and development partners. And then we have been lucky to get support, as I said before, on the university side from MDA, Grace Science Foundation, UMDF, and and the FARA, Friedreich's Ataxia Research Alliance. Thanks very much. And I'm available for questions, and I think I'm done about 5 minutes early.
That's great. Thank you very much. I don't think we've had any new questions come in, but I guess I'm wondering, I'm pretty familiar with this project and, thinking more about the mitochondrial applications, the mitochondrial myopathies. A disease like Leigh syndrome, I'm wondering if you might have a faster path to get to the clinic. I know there have been a couple of cases where people have been granted a sort of individual INDs and other, faster paths to the clinic, if you can do that safely, and wondered if you had thought about that, since that disease does have such high mortality.
Yeah, and I'd be happy to talk to other people. So I'm a little bit more familiar with the clinical path in Friedreich's, and there's sort of a road to hoe. That's probably not the right way, but people know what the FDA likes in terms of their endpoints to move and all that, and so it's basically we can pull a clinical trial design off the shelf for that because I've participated in that before, but I'm not aware that is as available in Leigh syndrome. But one of the earlier speakers was saying, or it was from the panel, I guess, be bold, be the first. And it's got more risk, but potentially more reward. Yes.
Well, certainly I think a good outcome might be very clear cut in that disease. It's, typically lethal at fairly young age. So if you were able to move the needle there, again, there's a variability, but it's just a thought.
No, I like it. It's been, I've been back and forth. Should we be doing Leigh's rather than Friedreich's? It's a hard thing for me to decide.
We don't talk you out of Friedreich's either. So.
Say it again.
I don't want to talk you out of Friedreich's either. So. Okay.
Yeah, I mean, the other thing is because we're seeing very potent effects on mitochondria in the muscle, not only the heart, but also skeletal muscle. We're thinking about other diseases, maybe more aligned with MDA's main line, like Duchenne's, and we're going to be starting to test it in a MDX mouse in the next few weeks. So, but yeah, one needs to be focused with one's clinical plan, and so that's the way we've directed it so far. Yeah.
Okay, well, thank you very much, Dr. Cortopassi. I'm not seeing any additional questions. Well, thank you all. I appreciate it.